Welcome, Dr. Stefan. The floor is yours.
Well, good morning, everybody. It's a great pleasure to be here. Thank you for the kind introduction. Can we go to the start of the presentation? I mean, it's nice to, or maybe I just, so this is a little preview for you. Just go it backwards and then we're done straight away. Okay, early morning. Here we go. Right. Thank you for the invitation to share the work we've been doing for the last 12 years, pretty much. So I'm a computational biologist by training, who slowly morphed over time into a genetics person. I'm working in this lovely place here in the south of Cambridge. You can see we've got some, night sky images in Cambridge as well. So this is the city centre. We're in the south of Cambridge, and it's this biomedical campus that has seen in the past years buildings coming out of the soil like mushrooms, really. This is the Victor Philipp Daedalus Hardenlang Research Institute where I am. We're right next to the Papworth Hospital that was relocated into Cambridge a couple of years ago. And in the background here, you can see the new headquarters of AstraZeneca. So we're right in the middle of everything where you have to be. Now, in the next few minutes, I would like to take you on a short journey through the past, present, and future of genetics in PAH. Like to highlight discoveries, achievements, and also limitations, which then really trigger us to identify those opportunities for translation into the clinic, in particular for clinical genetics, but also for future global research. So if we go back 25 years, a quarter of a century, Richard Trembath, Bill Nichols, and team have identified heterozygous germline mutation in BMPR2 encoding the bone morphogenetic protein receptor type 2, and these mutations are associated with pulmonary hypertension. You can see on this graph here that on the left-hand side, I'm plotting, the number of genes as we go along, and on the right-hand side, we start from the top at 100% unexplained cases down to 0. And you can see with the discovery of BMPR2, we were able to explain about 15 to 20% of idiopathic PH patients. Following these discoveries, candidate gene approaches mainly have then identified, in particular, genes around the TGF-beta signaling pathway. But then going forwards, you can see also that the step curve has started, the frequency of additional genes got smaller here, and that was caused by the very disruptive next-generation sequencing technology that came available at the time. However, if you see the red curve again, you see the number of cases we're explaining is significantly smaller as we go along. Now, the next-generation sequencing has given us the opportunity to really look deeper, and so technologies were developed, and depending on which regions are covered, we are able to now look at either the whole genome, which is here at the bottom, which we also call WGS, or just the protein coding fraction of the genome, which is called the exome. And in clinical practice, very often we these days find a gene panel testing where just the diseased genes are represented on these arrays, and you can see here, indicated by the star, variants that might be identified in there. Having this technology of next generation sequencing, we then started back in 2012 a project internationally set up, but led in the UK and by the UK to do population-scale sequencing in the healthcare system with the aim to streamline genetic diagnosis in the national healthcare system. And if there was no diagnostic rate gene identified, this went into the research arm of the study, where we tried to uncover missing genetic heritability in both the protein coding and the non-coding regions of the genome. Now, let's just do a quick example on what we can actually read out of a genome if we sequence it. We do get about 3 to 5 million variants, and I mean, this is clearly indicating straight away where the challenge is in terms of how to find potential causal mutation in there. So if we take just the protein coding fraction of the genome, this is then getting us down to just 30,000 variants. Only 1 or 2 of these, if we think about Mendelian disease, might be disease relevant. So we need many lines of evidence to really hone down in here. The most powerful discriminator is the allele frequency, and this is defined as the proportion of alleles, 2 per individual, that carry a particular variant at a specific location in the genome. Now, it is very important to have an idea what the genetic architecture looks like, so that you can choose the optimal approach to discover causal variants. And so if we plot the allele frequency on the x-axis and the effect size on the y-axis, what I've just described on the previous slide, we have now identified the majority of monogenic causes, which are rare variants, mainly or predominantly in the protein coding space, which have a very large effect size. However, for many, many years, probably 50 years or so, with the advent of SNP arrays, we already were able to just probe somewhere between 500,000 to 2 million positions in the genome and undertake common variant analysis, where we then identified common variants with a smaller effect size. Now, it is important where those variants reside in the genome, and on the right-hand side you can see an illustration how the effect of a variant on the protein depends on the location and affects either the structure or the dosage or somewhere in between. Very important information to analyze the data. Now, we've had 13,000 genomes here in the end that we sequenced. It is important to have the right reference populations to do these analyses. However, this is a fast-forward view of a recent publication here, and what you can see is that we clearly have large efforts already done in America and Europe, and also some in the Asian area. However, there is a lot of gray area where we don't have actually reference genomes, and that is an unmet need which we really need to address going forwards. Now, back to pulmonary hypertension. You can see now here the plot has completed at this point. So when we started, we did a rare variant analysis and identified 4 genes here. And at the same time, we also did a genome-wide association study. And to our very surprise, we identified both rare coding variants in the protein coding space in the protein coding body of SOC17, a very important transcription factor, and we're able to demonstrate segregation of the phenotype, as well as signals in the enhancer of SOC17, which is covered by common variants. The size of the cohort you analyze plays an important role, and so we joined forces with our friends and colleagues in the US at the PH Biobank, and we were able to float up further variants. Now, if you don't have the opportunity to increase your size of cohort, you can also start trying to do much smarter approaches. And one of these we've applied to identify KDR, and I just want to highlight this approach a little bit more. So this was a Bayesian framework that we used here to encode rare variant association. We started with the phenotypes, where we simply labeled our individuals in this case control analysis. We had the genotypes, which we, with a focus on rare variants, filtered according to pathogenicity scores and labeled those individuals that were already explained with pathogenic mutations. These went together into an analysis framework that was developed by one of our PhD students in the lab from the NHR bioresource called BeviMet. And this approach estimates the probability of association between a given set of variants by gene units, a case control label, which we've assigned through using the phenotypes. It considers the mode of inheritance, and it then calculates a probability that each individual variant is pathogenic. This plot here, which is a pseudo Manhattan plot, it really shows the result. So we were able to recapitulate pretty much, as you can see on the top, all the, at the time, known genes that were up to then associated with PAH. We then identified also KDR mutations, and in particular, you can see that here, these were mutations of, that were loss of function, so protein truncating mutations, which we already saw in the initial analysis here, but because the significance level wasn't big enough, we discarded it in the first place. So we identified that loss of function mutations in KDR are associated with a significantly reduced transfer coefficient, similarly to individuals that are diagnosed with PVOD that bear a biallelic mutation in IF2AK4. However, if we compare further, you can see that the loss of function mutation carriers clearly have a much later onset compared to, for example, the biallelic IF2AK4 mutation carriers. In fact, we were able to show also segregation of the phenotypes in one family. We identified 4 individuals in our cohort. We validated that in the US Biobank, where another 4 individuals were identified, and our colleagues in France also identified through panel sequencing another 2 individuals. So there was some strong evidence here that something was going on. Now, if you look at this tree, and this is important for, where's the pointer gone? For clinical genetics, really. So we have this lady here who was diagnosed with PAH shortly after her father died. Now, that tree clearly indicates that both her sisters are at risk, as well as the offsprings that needs genetic counseling here in this case and follow-up. Good. So if you look at this, we still only explained about 1/3 of individuals, so there's much more to do, and the question obviously remains, is there still further genetics we need to identify, or are there other factors that we need to look out for? I won't go into more detail here at that point right now, and I'm pretty sure we'll see it. So let me just quickly summarize part 1. I think significant advantages in next-generation sequencing enable genetic studies at much higher resolution. Clearly, the statistical power for genetic discovery increases with the increasing size of the cohort. We've demonstrated quite interestingly that, in particular, standardized and harmonized phenotypes with genetic data provides further advances. However, the lack of samples, as I mentioned, the lack of sample diversity is an unmet need. So, because of this, we put together, and in actual fact, Nick Morell initiated that back in 2016 in San Francisco at the ATS. We put together the International Consortium for Genetic Studies in PAH to pool resources and expertise across the globe. In our first symposium, the result was that we formed 19 working groups, and I'm just highlighting 2 of them here because they're important, I think, and almost like flagship exercises from that group. So the TBX4 focused working group, for example, has now morphed into a really big initiative, also driven by a family member of an affected young boy, and Eric Austin and Anton Mokin are both running this now for several years, and they've had a massive success with this going forwards. I will speak a bit more about our efforts in terms of the clinical genome and the ClinGen initiative in the next few slides. I just also want to mention that a subgroup of us from the consortium were asked to form the Task Force 3 in Genetics and Genomics, and you can read the recommendations in the ERJ manuscript. So looking at the clinical genetics, so how can we translate this? The purpose of the ClinGen initiative is clinical validation. So what we want to do is we want to evaluate the strength of evidence supporting gene disease associations, and then communicate these results to the community and in particular the clinical community, so that they have some guidelines and some lead. You can see it's a big effort. A lot of people have joined in and working on this. We're working on at 2 levels, and I'll explain that quickly to you now, because I'm worried I'm running out of time. So we started at the gene level, and for BMPR2 we've now gone down to variant level. We started off with formulating a consensus statement on genetics and genetic counseling and testing, as you can see on the right side, which we now updated, slightly as part of the 7th World Symposium and the task force work we've been doing. In the first instance, we looked at curating the genes, and as you can see, we had to define a scope of work, did extensive literature research, then work out what the exact scoring is in terms of identifying from the literature both genetic and experimental evidence, perform the classification. And you can see the result here on the right-hand side. 12 genes came into the top group, which are considered as definitive. There were 3 in the moderate group, another few with limited evidence, and we've got the non-relevant and disputed genes on the far right. However, I want you to just remember BMPR1A, because we'll get back to this later on. But here is the guideline. So if you start genetic testing, I think these are this is the list of genes you should definitely start looking at. In a further step, we went down and looked at variant curation. We adapted the BMPR2, the ACMG guidelines specific to BMPR2. You get 5 categories based on the likelihood of pathogenicity, and we did a little pilot where we systematically curated variants from ClinVar, and the results are shown here on the right-hand side, where we were able to resolve conflicting entries in ClinVar for 28 selected variants in the first instance. This manuscript is already a preprint on medRxiv and currently under review. So to conclude, this second part is that the international collaborative efforts provide an empowering platform to engage patients and their families, evidenced by the TBX4 effort. I would like to encourage everyone who is interested in joining the International Consortium to get in touch. We can put you on the mailing list, and you will receive regular updates, and we have monthly meetings. Genetic validation through the ClinGen curation expert panel obviously increases confidence in gene disease association down to the variant level. We've already demonstrated now that over time, variants of uncertain significance will be reclassified, and every 3 years we revisit our gene curations to see whether new evidence has emerged in the literature. Genetic testing should be offered to most group 1 conditions, and pediatric group 3, which is the update, from the World Symposium. However, there's a caveat there because so far clinical guidelines only exist for some of the genetic conditions. So, and with this, I would like to sum up. Ideally, I think what's already become clear is we need these registries that really have a much wider scope. They should be population scale, diverse and global in its characteristics, ideally longitudinal. We need to look at not just affected, also unaffected family members and get an idea through the follow-up what's happening. And from a data science and computational point of view, we need to make sure we all speak the same language so we can federate data. With this in mind, we've put out a call to action at the World Symposium from our task force, and you can scan this QR code here and provide your information if you have samples and or data you are willing to contribute to this for further future genetic and genomic efforts. Or please just come and speak to me after the presentation. This is a little warning. There will be some advertisement, as in any good podcast, you know. So here we go. Now, going forwards, this effort will be taken on by the international consortium. Obviously, we made a deliberate decision to also align this with PVRI Go Deep, who has a meta-registry, and many of you have probably heard about this already. So I've borrowed this slide from Werner Seeger, who presented this at the PVRI in Rio 2 weeks ago. And I mean, it's an astonishing effort and massive achievement. You can see there is now 35,000 enrollees in this database, and further people are poised to hook up with this. Now, they also decided that, you know, we keep the phenotype data, the clinical information, separate from the genetics, just also for a you know, trust and security purpose, I think, to convince the regulators. But we have to start, you know, thinking strategically how we can push the boundaries to get data sharing going across the world, and that is a big challenge. Last bit I want to mention is PVRI Go Deep themselves have decided now to also take care about imaging data, and this is something that is coming up and is in development. And the reason why this is so important, and this is the last, pretty much the last slide, we've recently undertaken an interesting study in, you know, one of the reference databases, UK Biobank, where we analyzed CMR data with the existing genetic data from SNP arrays. We undertook a sex-stratified genome-wide association study and identified common variants that are sex-specific candidate loci in 2 genes, and here we go, BMPR1A as a sudden turns up again. These associated with 2 particular parameters from the MR images, which is the right ventricular end-systolic volume and the right ventricular ejection fraction. We were then able to take forwards BMPR1A and show that the minor allele located on the promoter region of BMPR1A is associated with increased cardiac index in female patients in our UK cohort. Clearly indicating that, you know, again, combining the genetic data with really high-quality, valuable data is extremely helpful. However, there is limitations that apply. And with that, I want to summarize. Knowledge gaps for precision medicine require larger global and more diverse sample sizes, as I've explained. Leveraging clinical information in association studies adds significant statistical power. Standardization and harmonization of resources will be key. We need to speak the same language, simply saying. I think it's absolutely clear we need to be much more careful when we do the analysis and perform these analyses sex-aware and clearly integrated, we really need to make sure that genetic testing for PH patients is integrated in clinical care. There is a long list of next steps, but I think I've pretty much run out of time. So I'll let this up in a second. Just want to quickly acknowledge clearly the patients, volunteers, research nurses, and coordinators that help getting these works done. It would be a very full slide if I name everybody individually, but then also, you know, the geneticists, biologists, computer, and clinician scientists here who, in part, have joined the PH Icon initiative. Particular thanks go to my colleagues and friends from the Task Force 3 and obviously the funding bodies. And with that, I thank you for your attention and leave that up again.
Thank you, Doctor. Please join us for questions at the end of the session. And now our next speaker is Professor Paul Yu from Harvard Medical School. He's giving a talk today about, sorry, the title is not here. Please, Dr. Paul, the floor is yours. Thank you.
Thanks so much for the introduction, and thanks for the invitation to join this really great symposium. I'm Paul Yu. I'm from Mass General in Boston, and these are some of my disclosures, and I just wanted to note that this is a pilot study of a nuclear imaging modality for pulmonary hypertension. This was supported by Gossamer. So why do we need to develop new imaging modalities? I think that we have seen the power of CMR, of CT as part of our standard of care, but we're hoping to gain new aspects of characterization and phenotype in our patients. And PET imaging, nuclear imaging using molecular imaging gives us a number of important advantages. Molecular imaging with PET gives us very high sensitivity and high signal to noise for very low abundance signals, rare cell types, rare proteins. It gives you an opportunity to look not only for these rare molecules, but also metabolites. And you can actually assess different cellular processes with this kind of imaging. Dynamic imaging is a refinement of PET imaging that increases our specificity and looks at the uptake in time with some of our probes beyond just the static uptake values at a given time point. And then many of the probes that are being used, coincidentally, are drugs that have been modified to have a nuclear label on them, and that provides proof of target engagement and tissue specificity. There are some disadvantages of PET imaging. PET imaging, even though it's better than some of the SPECT imaging that preceded it, it still has less spatial resolution than what we're used to in CT or MR. There's a need for an exposure to ionizing radiation. This is generally 1 to 5 millisieverts per study in addition to the CT. This is being combined with PET imaging now, so that can reduce one aspect of the radiation dose. And then another limitation is that for some of these probes, you actually need to have a cyclotron nearby and a nuclear pharmacy to generate these. There are commercial probes that are easier to obtain, but many of the experimental investigational probes need to be made on site because they're not commercially available and they have extremely short half-lives. And so these are the isotopes that we typically use for PET imaging. We use 18 fluoride, which has a intermediate but fairly short life and emits beta particles. We have 68 gallium, and this is what we used in our study. It is a pretty high energy emitter, so it can get resolution into some very deep structures, but its half-life is pretty short. And then we have zirconium-89, which is best used for tracers that are going to be residing for days rather than hours. And this is a very high energy emitter as well. Okay, so some efforts preceded our effort. This is the group in Canada that looked at pulmobine, which is a labeled adrenomedullin receptor targeting probe. And this actually used SPECT, not PET. And what they actually found wasn't so much increased uptake in pulmonary hypertension, but an increased degree of heterogeneity. They used this measure called the Activity Distribution Index, and while there might be some modestly increased uptake in their pulmonary hypertension, and that's group 2 pH, and then group 1 pH, as well as CTEPH, there was more of an association with this heterogeneity, but it wasn't true in every case. This is a study that came out of a collaboration from Harm Bogaard's group in the Netherlands and Lanzhou in Imperial College. And what they used was this 18-fluorine molecule that I told you about, but this is substituted onto thymidine. So thymidine obviously is a nucleotide which would be taken up by mitotic cells. And the hypothesis that had been advanced was that pulmonary hypertension is a hyperproliferative disease, and so using a tracer for DNA replication might detect enhanced proliferation in the vasculature. And in this small pilot study, in fact, that's what they found. These studies are necessarily small pilot studies because you're trying to get proof of concept for justifying the dose of radiation for these patients. The group actually pursued a follow-up after this initial positive study. They recruited more patients from both sites. Unfortunately, the initial results were not replicated in the follow-up study. There wasn't necessarily an increased uptake of thymidine in their PAH patients. Even in the original pilot study, there weren't huge increases in the relative uptake of thymidine. I think what we can take from this is that small studies like this are always prone to having encouraging but maybe misleading findings. So in need of replication in larger studies, but also in contrast to what had been supposed, maybe pulmonary hypertension isn't a cancer-like state. It isn't so hyperproliferative in the way that a cancer probe such as 18-fluorine thymidine might detect. Okay, and this study used a different approach. They used a labeled cell. This is a human endothelial progenitor cell that had been isolated and tagged with this long-acting, this long half-life probe, 89 zirconium oxine. And what they found was that there was slightly increased uptake over time in monocrotaline-induced pulmonary hypertension treated rats. And again, the margins are a bit small, maybe 2.5 to 3-fold, but sort of persistent over a couple of days after injection. This study from Korea took a different approach. This is a human study, and they used manosylated albumin, and that was labeled with 68 gallium, again, the high-energy short half-life probe. And what they found was that in pulmonary hypertension, there was a small increase in uptake. Again, a very small pilot study with just a handful of patients. And I think the contrast is a little bit enhanced to show you the effect in column D down there with pulmonary hypertension, but it does look like there's some modest increase. So what does this reveal? This is a saccharide that is bound by macrophages, which have the mannose receptor, and so this could suggest that there's an inflammatory phenotype in the vasculature. So we took some of these preliminary studies as a guide to how to design our study. In this case, we chose to use gallium dotatate, which is a commercially approved probe. This is used for diagnosing carcinoid and certain other neuroendocrine tumors, such as thyroid cancer. And what this is it's actually somatostatin analog DOTA labeled with 68 gallium, and it will bind to SSTR2, the somatostatin type 2 receptor. And you can see it's quite good for detecting the local and metastatic presence of these neuroendocrine tumors. It's been, the same probe has been applied to detect vascular inflammation in acute coronary syndromes and in acute carotid syndromes associated with stroke, and also for detecting myocarditis. It's taken up by macrophages that infiltrate to these sites. And the histological studies support that this is in fact what's being labeled. So we hypothesize, since macrophages are thought to be an important component in pulmonary hypertension-associated vascular inflammation, that dotatate might be able to detect this aspect of the disease. And so we enrolled 7 patients with group 1 pulmonary hypertension, 3 controls that were actually enrolled, and then we took advantage of a large data set of several hundred patients who had undergone gallium dotatate scanning at our center, but were negative for metastatic disease in the chest. We looked at a number of clinical parameters, and we used a couple of different metrics for quantifying the scans. We normalized against the blood pool, the uptake in the proximal aorta, and then we had volumes of interest drawn by 2 radiologists, and specific uptake values were normalized against the blood pool. So the values that I'm going to show you are the net enrichment over the blood. And this is the characteristics of our cohort. This is supplemented with some of our historical controls scanned on the same hardware using the same probe, same protocol, fairly matched for gender, BMI, and comorbidities. And this is a sample scan. What appeared to be the case is that in some of the patients who received the dotatate with group 1 PAH, this is associated with connective tissue disease patient, there was an increase in the lung parenchymal uptake of the probe. We had our radiologists look at the scans in a blinded fashion and score them qualitatively. So this was their subjective impression without knowing the diagnosis. And in every case, all of the positive scans were from our cohort of patients and not in any of the controls. But then we quantified the SUVs in this way based on taking the uptake in the lungs and dividing it by the blood pool in the ascending aorta. And so these are some of the clinical values here. So what did we see? Out of the 7 patients, 4 of the patients had extremely high uptake values. This is a 3- to 4-fold enrichment versus the blood, but probably a 5- to 6-fold enrichment versus the mean values in the controls. And then the other 3 were totally normal. They broke evenly between idiopathic and connective tissue disease. We tried to enroll a bit of both, hypothesizing this would be more associated with connective tissue disease. Didn't seem to be specific for that. As a test for pulmonary hypertension, I wouldn't rely on this, but it turned out to have a pretty decent area under the curve with a sensitivity of 86% and specificity of 83% at this cutoff of 0.8. There is a trend towards association with some clinical parameters, including echocardiographic RVSP. What we did note was that this seemed to be higher in patients who are on multi-drug regimens. Unclear if this is due to an effect of the regimens themselves or due to their greater severity. There is a trend with higher reveal scores among the patients who had elevated uptake. And interestingly, SSTR2 expression was noted to be enhanced in a subset of maybe 15 to 20% of patients in a cohort of 55 explanted lungs with pulmonary hypertension. This is from the Lafayette group. And what we found was that these individuals who had had high SSTR2 expression broke evenly between idiopathic and connective tissue disease. And in this cohort, it looked like the patients who were in this tail were enriched for patients receiving triple drug therapy or prostacyclin-containing regimens. Again, raising the possibility of being a more severe population or having prostacyclin therapy as their common element. We did some histology on explanted lungs these are patients that were transplanted at Cambridge University. These were from Nick Morell and Mark Southwood. What we found was that not in every patient, but in some patients, there was increased SSTR staining that corresponded to a population of CD68 macrophages. I'll say that this is not a universal finding, but it perhaps goes with the sense that vascular inflammation is a sporadic finding on histopathology. Unclear if that's due to sampling error or due to an actual variation in phenotypes. Based on our scanning study, we wonder if this is actually reflecting a difference in pulmonary vascular inflammation throughout the lungs. So to summarize, gallium dotatate SUVs were dichotomous in pulmonary hypertension patients. We think this is kind of noteworthy that the patients who were positive weren't just a little bit positive, but they were 5 to 6-fold elevated versus the controls. There was a trend with PVR and EVRSP. It correlated with a number of pH-specific medications, and there is supporting data from histopathology and from expression profiling that SSTR2 is a sporadic feature of possibly increased inflammation in a subset of patients. So we wonder then if dotatate might be used to track efforts that target inflammation in pulmonary hypertension and might signify an inflammatory endophenotype. And with that, I just want to thank our colleagues who enabled this study. This is the nuclear medicine physician, Heather Jacine, who helped with our analysis. Aaron Waxman helped us with recruiting our patients, our nuclear medicine director, and Larry Zisman actually gave us a lot of scientific support for this study. And with that, I'll conclude. Thank you.
Thank you, Professor Paul.
It's my pleasure now to introduce our next speaker who's joining us virtually from Gießen. This is Soni Pulimseti, a colleague of ours who's a professor at Justus Liebig University, and she's going to speak to us on epigenomics of pulmonary hypertension and translation.
Thanks a lot, Paul. Can you hear me?
Yes. We can hear you.
Okay. Thanks a lot for the invitation to Abdullah and also for Paul for the introduction. So I'd like to read a little bit piece to the talk title to Epigenomics and the Metabolic Pathways in PH. And I think as Paul was already mentioning, I'm from Gießen, Germany, which is in the middle of Germany, located where you can see here. Can you also see my arrow?
Yes, we can see.
Okay, good. So in the middle of there, and then my group is majorly located. Give me a second. My group is majorly located to understand the interplay between the epigenetics and the transcription factors in chronic lung diseases. And of course, majorly focusing on pulmonary hypertension. But we are also working on some of these relevant aspects in lung fibrosis and COPD. With pulmonary hypertension, when you now look at the cellular and different molecular mechanisms, so the pulmonary vascular remodeling in pulmonary hypertension, it is driven by various interconnected things. So we have due to a genetic or the environmental factors, we have endothelial dysfunction. This may be leading to smooth muscle cell fibroblast proliferation or transdifferentiation, and the inflammation and immunity plays an important role, which we also heard already from Paul's talk. So this is all leading to pulmonary vascular remodeling. And the most important and highly studied pathways is, of course, is about the BMP, TGF-beta, and activin. Paul, I was expecting that you will be talking about activin, so that's why I didn't cover that. But you already know about the importance of the activin signaling, the studies from Paul Yu's group, and also sotatercept in the clinic. how functional it is. So, but today I think I'll be talking more about the metabolic and the epigenetic and the transcription factor networks that are integrated and how they will be driving the pulmonary vascular remodeling changes. Why? At first, I would like to briefly overview about epigenetic changes, why we think it is important, because we also know in the what Stefan was Graf was mentioning, and also from all our work from the genetics, that not every patient who has mutations are susceptible to the disease or exhibits the disease phenotype. So this means that they are, like, the epigenetics plays an important role, how you are influenced by your environmental stimuli, the age factors or the drugs and so forth. So, and this plays an important role. And then when you look at the epigenetic mechanisms, so they are, may have changes at the higher chromatin organization, but at a base level, then it could be changes in the DNA methylation. So whether DNA can be methylated, and the histones, which are the main proteins around these are also modified, like acetylation can be happening, or the methylation of the histones, and also some of the non-coding regions can also plays an important role. What do they do, all these changes? So they are important in turning on or turning off the gene expression. So you can see here, so it can also lead to a condensed switching of the state with the increased DNA methylation status, for example, a histone methylation, whereas the histone acetylation changes can open up the chromatin. When you open up the chromatin, so then you have the transcription factors, then the whole transcriptional machinery can bind, and the transcription, the gene expression can go on. So when this kind of, like, to understand, so how the epigenetic landscape is altered in the pH and vascular cells, the first efforts were given by Merlin's group and also our group. So we try to work on more on which I will be explaining, on the smooth muscle cells and fibroblast, and Merlin's group has shown in endothelial cells which are isolated from the pH patient lungs. So they did the RNA sequencing to identify the changes in the transcriptome, but also the chia PET. So in that, combining these two data sets, they can able to show how are the enhancers, that means far away elements, can regulate the gene expression. But what they have found, there is an extensive remodeling of these enhancers and the gene regulatory networks, but however, there is a very little connection to the transcriptional changes, because maybe they are waiting for the ligands and the disease-specific situations that comes, and then these enhancers are active and regulates the gene expression. And we did our efforts to identify the epigenetic landscape from the fibroblasts, which are adventitial fibroblasts that are isolated from the pH lungs. And we know these fibroblasts, they have hyperproliferative nature, they're pro-inflammatory nature, apoptosis resistant, and this phenotype is maintained for several passages. So from these ones, we did the transcriptome analysis, like we're doing RNA-seq, and the epigenome analysis of doing the chip sequencing, the chromatin immunoproliferation sequencing of 12 different histone marks. So in a nutshell, when we combine these data of the RNA sequencing and also the chip-seq data sets, and integrated these data, we got like two different networks. So one is like where the gene expression is, increased gene expression is associated with some of the histone marks, with increased histone acetylation marks, and decreased expression is associated with an increased H3K27 trimethylation marks. And looking further deep into that, what we have identified is we observed that more than 2,000 genes are differentially regulated, 4,000 enhancers are altered, and what we observed mainly is that we see more than 30% of the genes are related to the developmental genes. So what we believe is, like, we are awakening the developmental pathways and transcription factors in a wrong time of your life cycle. So there may be a reactivation of the developmental transcription factors and genes, which is going on, could be happening there, and we are further pursuing this developmental theory of pH. And the next aspect I think I would like to say is that also the studies confirms this, because you have changes in the histone acetylation, but also this means that this histone acetylation marks has to be written by the enzymes which are adding the acetylation groups that are called writers, and the removing the histone deacetylases, and then they are read by the enzymes called bromodomain. So what we observe is that these histone acetylation signatures are increased in the human pulmonary hypertension, and the enzymes which writes these ones also which removes, both of them are increased in the PH vascular cells. And then further trying to block these signaling pathways, because we see an increased histone acetylation and increased histone acetylation associated transcriptional changes. We try to block the histone acetyl transferases using two different compounds, the CCS1477, CBP via nebulization, and we applied this in 3 animal models, fawn-hooded rats, monocrotaline, and also hypoxia zoo. In all the 3 conditions, both of these drugs, you can see they decrease the right ventricle systolic pressure and then improves cardiac output. And further, we also check this for the human relevance, because then for that we established the pH precision cut lung slices, the living slices. So you can make them and culture them for 14 days and can pre-treat with the different compounds. So what you can observe is, like in this human pH precision cut lung slices, which you obtained from the transplanted lungs, treatment with these 2 different compounds, they decreases the vascular remodeling. And similarly, the group from also from Amsterdam group, and also from Sebastian Bonis from Quebec, they have showed that the blocking of readers, the bromodomain inhibitors, are also in a different animal models were shown to block the bromodomain inhibitors also improves decreases the right ventricle systolic pressure and improves the cardiac output. And further progressing this, they have done a pilot study with the bromodomain inhibitors, epabetalone, approach A study, so which you can see here in a very handful of patients. But then I think you see a decrease in the pulmonary vascular resistance and an improvement in the cardiac output. And the phase 2 clinical trial with approach 2 is planned. But also found that as I told you, that it's not only histone acetylation, HDAC inhibitors are also, HDACs are also upregulated, and blocking these class 1 HDAC inhibitors, there was a CS1, which is a controlled result formulation of valproic acid, which blocks selectively class 1 HDAC inhibitors. And this was checked in the phase 2, and it shows that a positive impact on the revealed risk score, functional class, and mean pulmonary arterial pressures. So maybe this is maybe used as a compassionate use for the pH. So this suggests that the epigenetics alterations are happening in pH vasculature, and the initial clinical trials may give you a positive hope towards this. Another aspect which I want to cover is about the metabolic changes that are happening. So we all know from previous work from Stephen Archer's team and so forth that there is a lot of Michelager's team, that there is a lot of changes that are happening in the pyruvate, the glycolysis pathway, so the pyruvate to the acetyl-CoA and so on. But also the TCA cycle changes. Apart from the TCA cycle changes, there is a recent literature suggesting in pH that a lot of changes in the lipid metabolism and fatty acid oxidation, which at the end leads to changes in the mitochondrial, as well as not only the mitochondrial changes of polarization, but these changes can also influence again because also the epigenetic changes. Because for example, if you have changes in the acetyl-CoA, they are the co-molecules that are important for the histone acetylation. Alpha-ketoglutarate is important for the histone demethylation. So in that way, so there is like this crosstalk of the epigenetics and the metabolic changes is an important player. In this regard, the work from Marlene's group has shown when there is a loss of BMPR2, this increases an epigenetic enzyme, ALDH1A3, and this increases the acetyl-CoA levels. In that way, it opens up the chromatin and coordinates the regulation of energy metabolism, nucleotide synthesis, and the cell cycle. So now this clearly shows that the acetyl-CoA, which is coming from the TCA cycle, is an important player in that. And in this direction, recently Sebastian Bonnet's group from Quebec also showed that the S-type-CoA, one of the ATP citrate which produces S-type-CoA, and could be an important player in driving one side the lipid synthesis and another side the epigenetic changes by regulating the proliferative genes. So blocking this ATP citrate lyase, so what they can do is they can block on one side this lipid synthesis changes, another side is the proliferative genes. And this blocking of this ACLY with the BMS compound, they can show that there is a decrease in mean pulmonary arterial pressures, pulmonary vascular resistance, and also in the precision cut lung slices, there is a decrease in the medial wall thickness. So suggesting that the changes in the metabolic changes go hand in hand also in the regulation of the epigenetic changes, and this interaction plays an important role. And another recent study from Stephen Chan's group from Pittsburgh is pretty interesting, because what they try to check is that metabolic in a huge cohort, they did the metabolic changes and they identified there is an increase in the oxysterol and the bile acids. And further doing the genomic studies, they have identified there is a SNP in one of the molecule called nuclear co-receptor A7 and CoA7, and this SNP leads to loss of function of the NCoA7 or dysfunction, and this is associated with the survival and the 6-minute walk distance and the mortality of these patients. And looking at the mechanism, what they have identified is this NCoA7, which can have an impact on the lysosomal dysfunction, because this is an important player in the lysosomal dysfunction and can be regulating again like the changes in the oxysterol and the phenotype of the endothelial cells. So I think in the normal conditions, what will be this NCoA7 is doing is that this is trying to bind with the EV ATPase, which is important for the lysosome, and it's trying to control the sterol metabolism. So what is happening is you have sterols and they are redirected to fatty acids and fatty acid oxidation in the mitochondrial thing, acetyl-CoA, and then you have epigenetic changes and so forth. There is a hemostatic. But in pH conditions, you have a deficiency of NCoA7, either because of the genetic or because of the IL-6 or whatever these pro-inflammatory cytokines, they can also leads to decreased expression of this NCoA7. And this leads to less acidification in the lysosome. That means more sterols are produced, and they leads to a more chemokine and cytokine release, and which causes the pro-inflammatory phenotype of the endothelial cells, but this could be also applicable to other cells. So you have a total reduction of fatty acid beta oxidation, and this is also then influencing the mitochondrial changes and also the nuclear epigenetic changes. So this seems that we have an abrupted cellular communication going on in pH, like you have different organelles are affected, and then we need to more and more understand how this cumulative effect is going to influence these changes. And this could be one example of that. So in conclusion, so I think the epigenetics and the metabolic changes and also the transcription factor networks plays an important role in driving the pH pathogenesis. And there is studies going on at the preclinical level, and also, more towards, some of them move towards the clinic. For example, with regard to the epigenetics, the histone HTAG inhibitors and the BRD inhibitors, they were in the phase 1 and phase 2 clinical trials. And the HAT inhibitors we will be checking further on. But then when you now look at the metabolic state, metabolism, so there is like ACLY inhibitors is there, the GLS1, serine metabolic changes. So there is a lot of them which is proved to be efficacy in the preclinical, and we need to see what comes up. And also with the transcription factors like FOXOs and PPAGA activators and so forth. But is that the end? No, because I think the problem with this is like you have a lot of changes that are happening, and these are all interconnected. So that's why I think what we need to understand is more what are the nodes which critically integrates several of these changes which are happening in the compartment of the nucleus and the metabolic changes and the transcription. So what could be the critical nodes that are changing the most impact, and those ones we need to identify and we need to target those ones. This is still an open area, and because the metabolism, this fatty acid TCA coming up, but we don't know how is the other metabolic pathways are regulated and what could be critical integrators of this. And the second thing with epigenetics specifically is that seems to be clinically maybe some working good, but we also know from the cancer field that there will be definitely off-target effects because they do such a broad changes. So that's why I think what is important is because we need to control their impact. Instead of across many genes, we need to have on a specific, then we need to go on with precision epigenomic therapies, and we need to find a ways to detect the responders and the long-term outcomes, because that's why I think it is important to identify more and more where we want to have these epigenetic alterations to be targeted. So we need to have very much more specific things. This improved specificity is an important issue. Also for the metabolic modulators, this is the same thing. And the second thing is how closely we can bring all these epigenetic and metabolism alterations and so on, how the omics can be brought much closer to the clinic. So previous work from us from the right ventricle side, when we try to do from the RV transcriptomics, from the human compensated and decompensated, and from the animal models, we identified some molecules and then combined this with the Olink data from the plasma proteomics and identified 5 biomarkers. And a few of them is, at least in our study, but numbers are low, seems to have a better predictability than BNP. But I think the main thing is that they allow you to identify a better stratification of the patients from the early to late decompensation phase. And we combined this data with the imaging data. And so I think this is maybe the way. So we need to have the biology. This biology has to lead us to identify the biomarkers, to diagnose as well as to stratify the PH patients. This is something which omics can also be exploited, and we need to do much more efforts there. So one thing which we have recently done in this direction is we took in our PH patients, we tried to do two things. One is we tried to do the RNA sequencing and also the attack sequencing, which can tell us about open chromatin regions and the closed chromatin regions, and then this could be combined with the transcriptional changes. So what we identified from the PBMCs, so from the patients, and then I think we can able to achieve enhancer landscape. So how these enhancers are altered by specific drugs when we try to intervene and so on. So this kind of work is what we are trying to do. So to identify therapeutic responses and so forth, to bring more closer the omics to the clinic. With this, I would like to thank all the people from my group who contributed and collaboration partners. Thank you for your time. And with that, I close this talk.
For questions for all the speakers.
Hello.
Yes, go ahead.
Ayman Farghali from Egypt. My question for Dr. Hu. Actually, it's very interesting to have new modalities for imaging. And when we think about introducing new things like the PET scan, I would like to have some practical points in order to add. So you have concluded that it can differentiate between inflammatory phenotypes. And I didn't find any specification in your control group. What kind of control group? Because I would imagine that it can differentiate between a very important issue like idiopathic pulmonary hypertension and the HFpEF, for example. But I didn't understand this from your description about what is the type of your control group. Did you include such very important patients that we need to differentiate from idiopathic inflammatory types?
No, thanks for that feedback. Yeah, no, we didn't intentionally include patients with group 2 PH or HFpEF or heart failure. These were patients who were referred to us through a cancer center. You raise a good point. We tried to match comorbidities for diabetes, for obesity, for obstructive sleep apnea, but in many cases we didn't actually have cardiac imaging. We didn't actually have catheterization data for maybe 1 or 2, echoes on maybe 2 or 3. So I think a follow-up would definitely want to compare pulmonary hypertension against cardiorespiratory disease in general. That's an excellent point. Anton.
Yes, thank you, Paul, Stefan, and Sony, for 3 excellent presentations. I want to challenge you about all 3, and that is that we are mainly studying more established diseases. And established disease because that's when the diagnosis is made, and then the pulmonary vascular resistance is already high, and the pulmonary vascular damage is done, and then we are doing the PET scanning, and we're looking to epigenetics, and so on. And we don't know whether it's the consequence of what already happened in the pulmonary vasculature, because there is a rarefaction of vessels, and it's quite a hostile lung at that moment, or it is just a consequence of the diseases, and we are looking for treatment targets. So, and it might also probably explain sometimes the differences, for instance, in the thymidine PET imaging, as you showed, between the institutes. So my question is, how, the challenge is, how can we get the early developmental biology imaging and know whether it is the consequence or the cause of the diseases what we are studying? And I can imagine that it's very difficult for the epigenetics, but can you a little bit give your thoughts, all 3 of you, to how to be sure that it's the consequence of disease rather the cause, that the biology we are studying is the causing of the disease instead of the consequence?
That's a great question. Let Stefan and Sony start. Do you want to repeat the question?
So, for instance, for Stefan, genetic mimicking in animals might be something that we can study the early biology better? Is that possible? We do know now that the BMP2 rats.
Yes, I would say that is a good opportunity to do this. There is obviously limitations to this. In particular, when it comes to genetics in mice, I know, and when we look at transcriptomic, so there is differences between humans and mice, for example, that can be picked up, which you need to be aware of. I think Nick Morell has in the past very well also shown that nice cell models, so blood outgrowth endothelial cells, for example, and smooth muscle cells provide some really interesting insights into the mechanistic understanding. And I mean, if I go a little bit further forwards, this is exactly what we're trying to do now with large population scale data sets, the imaging. We've been funded an international consortium from the heart foundations in Germany, the Netherlands, and the UK, as you know. And there's 3 work packages. One is really addressing the genetics, but then all the findings we get from the genetics will be further analyzed for epigenetic changes and the systems there in Sony Pulamsetti's lab in various mouse models, genetic mouse models. And your colleagues in Amsterdam have taken on the challenge to also functionally investigate this further. And then when it comes to the actual clinical genetics at that point, this is the evidence we need to generate to be confident about what we're diagnosing in the patients.
Sony, go ahead.
Yeah. Anton, that's a beautiful question, and indeed it's a challenging one. So, but like from the right ventricles, what we are trying to do is to take RV biopsies, for example, when they come from the before pulmonary thromboidectomy, and then try to correlate according to ERSCS guidelines and based on the severity. So to think about like what could be the mechanisms that could be very early, like not that severe, and to severe, what could be changes. But when it comes to the pulmonary vasculature, so isolation of these endothelial cells from the catheter is one option, but we don't get so many cells and people are culturing this. So that's the problem. So extrapolating the things from the animal models, this is what we are trying to do. But I think this need to be combined. This need to be combined, for example, with the precision cut lung slices, but they are exposed to different growth factors and so on. because the human is much more complex, and we know a lot of regulatory genes in the intergenic regions and so on. This is more strong, more present non-coding parts and so forth. They are present in the human, they're not present in the animals. So that's why we cannot take one-to-one from the animals, but I think this needs to be combined. So this is what I think is with the iPSC's work, or with the precision cut lung slices from the human, and to expose them to hypoxia or to different conditions, and to have an integrated view. So that's the only option I can think more at this moment, because we don't have a direct access to the biopsies other than RV biopsies, but it's still very small amount and so on.
Speakers. So two quick questions. One is for Sony because she's far, and then for Stefan. So Sony, with this complex interaction between the metabolism, transcriptions, epigenetics, and many other levels of changes, what do we think the ideal drug to target which level that will probably be enough to tackle the problem? Like, I think having failed interventions many times, because any single development of a molecule targeting one problem failed because of this complex interaction. So what do you think the best ideal intervention will be at what level?
So again, it's also a pretty interesting question and complicated answer, because I think, I mean, as I was mentioning, so we need to go on and to target the molecules which are really integrating several of the signaling pathways. So this definitely comes to transcription factors or the epigenetic molecules and so on. So this is, but the only thing is we just need to find a way how to deliver them and how long to give them. So to have really to get rid of the side effects. So this is only problem for me, but otherwise we need to really target on those nodes which are most integrated of the signaling pathways. So that's one thing. And the metabolic modulators for me is maybe still, I think maybe these are more for the preventive approaches. Yeah. So to really think, if we know they are causing, so because a lot of metabolic modulators can be given early, so with not so many side effects, so could be that. But yeah, as I said, so we need to find this epigenetic or the transcription factors, but they're not that druggable. We need to find ways how to find a good to target those transcription factors, but they are the critical nodes. And then targeting them, we can really address multiple pathologies of the pH and also what's happening in the right ventricle. So this combination approach would be making sense.
We have not recently, over the last probably 4 or 5 years, identified a cluster of families from the same tribe, north of Saudi Arabia, positive for GDF2 mutation, but none of them has any feature of HHT. So it looks like it's a unique clinical phenotype with this gene mutation at some level, causing only severe pulmonary hypertension and no changes related to hemorrhagic hereditary telangiectasia. So have you seen something like this? And in simple genetic terms, because these are our only clinicians, how do you explain this?
Question. Probably in that very moment I will not be able to explain that because I need to see the data. So very important here would be to just simply, first of all, look at the pedigree, so the family tree, and see where the information is, where the affected individuals are, how these relate to each other, whether there is consanguinity and, you know, cousins, potentially, you know, relationships there that gave birth to offspring. And so the other comment I would make straight away is there is a question about penetrance and having mutations and whether, you know, what the penetrance is in terms of, you know, developing symptoms or not. And so I think we would need to look at this particular tribe and really try to work out, is there a founder mutation somewhere further upstream that could be identified, which most of the time is quite difficult because you don't have samples that can be genetically tested, for example. So I would be very interested to look together with you at this. One of your colleagues already mentioned that tribe to me as well, and it would be I think also an interesting exercise then to start potentially identifying mutation carriers that haven't got symptoms and follow this asymptomatic individuals in that family over time to gather more information about this. But as you say, this is a very difficult one because HHT and PAH, given the same pathway is affected, we are still not 100% sure, and we need to do much more work on this. They are very rare mutations. We only identified very few in our large cohort.
There are actually several families, and some of those who have the disease are homozygous and some actually heterozygous.
You see, so here we go. So that's further important information that needs to be looked at. Thank you. Thank you for that question. Very interesting.
Thank you, everyone, for this informative session. And sorry, we have one more question, and then we'll go for a break.
Thank you for giving me the chance to ask a question. Thanks for the session. My question is now, in the recent years, we are looking at the new classes of medications that are remodeling by nature. And through the remodeling process, they work through the BMBR2 pathway to remodel it and make changes. When we looked back into the genetic makeup of the patients, responders and non-responders, there was no difference. Although maybe the pathobiology of how the medications work involve the pathway for the BMBR2. How can we put this into clinical practice and the important work that you do? How can we translate it into a therapeutic area other than a single gene mutation that probably will not be the case, that will be translated even to better patient outcome?
Yeah, thank you for this very interesting question. And actually, from the statistical genomics point of view, we're starting to look at this. So this touches towards approaches like Mendelian randomization, where we'll try to really tackle the question: can we find additional variation in the genome that is responsible and predisposes for response to exposures that come from the outside. And one exposure could even be vasodilators, and whether people respond or not then depends on a different location in the genome, whether they actually respond to this therapy. And I think this is where we're now adjusting the statistical models. I can just put this out here. So there's something called, which is variance QTLs. And the theory behind that is we need to now use the statistical models to identify those mutations that allow us to make some statements about what the modulation is, and have the potential to better help us understand the differences in penetrance.
Thank you.
Thank you, everyone, for this fruitful discussion. And we can continue our discussion during the break. We'll have the break, and we'll be back at 10 o'clock for our next session. Thank you, everyone.
Assalamu alaikum, everyone, and we'd like to welcome you to this session entitled Definition, Classification, and Diagnosis. I'd like to introduce my co-chair and dear friend, Dr. Hussam Sakija, who needs no introduction in this group. So our first speaker, who will be presenting virtually but will be available for questions and answers in the panel discussion, is Dr. David Montani from France. So he will be talking about the pH definition and classification, evidence that led to the update at the 7th World Symposium. And Dr. Montani, if we can play the video, please.
Well, thank you very much for the invitation. I'm very proud to be with you to discuss about definition and classification of pulmonary hypertension from the 7th World Symposium on pH. So my name is David Montani. I work in the French Referral Center for Pulmonary Hypertension. These are my conflict of interest. And only one slide to begin to show you the journey of the World Symposium on pH from the first meeting in 1960 to the last meeting in Barcelona in 2024. And to say one word, say that all proceedings of each task force have been published in European Respiratory Journal. So there is a task force dedicated to definition, classification, and diagnosis of pH. I was honored to chair the Task Force 5 on definition, classification, and diagnosis with my close colleagues Gabor Kovacs from Austria and David Badej from United States, and all very sympathetic team. And so we have reviewed the definition, the classification, and diagnosis, but we do not talk about diagnosis today. So on definition first, as you know, there are a lot of changes in the definition of pH on hemodynamic criteria to define pH from the previous ESC/RS guidelines or World Symposium. In 2019, pH was defined by an increase of mean pulmonary arterial pressure higher than 25 millimeters of mercury. The precapillary pH was only defined by a decrease of a normal wedge pressure without inclusion of PVR in the definition of precapillary pH, and there is a very complicated for non-expert definition of the combined post and precapillary pH based on DPG and PVR. So progressively we shift to a more simple classification based on three criteria: mean PAP, wedge pressure, and PVR. And the threshold changed during the different World Symposium and the ESC/RS guidelines. And in the last one, the precapillary pH is defined by an increase in mean PAP higher than 20 millimeters of mercury, wedge pressure lower than 15, and PVR higher than 2. And so we discuss all this definition in our task force. First of all, the threshold of mean PAP above higher than 20 millimeters of mercury was the first question. And you know that we have changed. Since 2018, we shift from a threshold of 25 millimeters of mercury to 20, based on a different paper, but mainly on this very beautiful paper published by Gabor Kovacs on healthy subject. And you can see that the normal mean PP is around 13 millimeters of mercury. And when you use 25 millimeters of mercury of threshold, that means a free standard deviation. So progressively we shift to a more classical definition of abnormality for physiological parameters, and we shift to 20 millimeters of mercury that represent 2 standard deviation from the normal. And so now the threshold was 20. And this definition was reinforced by this paper published by Maron in Lancet Respiratory Medicine and from other papers showing that in a large population of more than 20,000 of patients, there is an increased mortality after when mean PAP was higher than 20. the increase began at 20. Of course, it's a large population, not well-defined population. We do not know exactly why the patient had an increase of mean PAP, but clearly we have a correlation with the level of mean PAP and prognosis. So it reinforces the good threshold that could be mean PAP of 20. The second point is about PVR. And the PVR on 2, you know that the normal PVR is around 1 Wood unit. And also in this paper from Brad Maron and other paper, we can see that clearly there is a shift of prognosis when you have more than PVR more than 2.2 Wood units in a large population of 40,000 of patients. So in the same way, we are not sure what is the explanation of increase of PVR, but when you have an increased PVR higher than 2, probably it's associated with a worse prognosis. And more recently, in 2023, this paper from UK look at the prognosis according to PVR and mean PAP in a large population. And you can see that in patients with mean PAP between 21 and 24, that means moderate pH, PVR is associated, higher than 2 is associated with a worse prognosis than patients with normal PVR. Of course, if you have more than 3, you have very severe prognosis in blue. If you have between 2 and 3, you are in green, with a decreased prognosis as compared to patients with normal PVR lower than 2. Exactly the same analysis if you look on patients with in the gray zone with PVR between 2 and 3, and you look on the impact of mean PAP on prognosis, you can see that the patient with a mean PAP between 21 and 24 had a worse prognosis as compared to patients with normal mean PAP lower than 20. It's independent of age, sex, and comorbidities. So clearly, all these papers reinforce the hypothesis that to have an increase of PVR higher than 2 and a mean PAP higher than 20 had a significant impact on prognosis. And it is clearly what we want to identify in this definition to define patients who can require specific management. Regarding wedge pressure, it's more tricky, and I think you have a presentation dedicated to group 2 pH, and I will be not too long on this part, but clearly if you have less than 12 of wedge pressure, 12 milliliters of mercury, clearly you have a precapillary pH. If you have more than 18 at baseline, it's also very likely that you have a post-capillary pH. Between 12 and 18, there is a zone of uncertainty, gray zone, and clearly in this patient, you should add some clinical data, history of the patient, some score may be helpful, echo parameters to better characterize the patients. And also, probably if you are between 12 and 18, it could be of interest to have a provocation test with exercise or free challenge to better characterize the patient. But to date, all the parameters and all clinical study was based on the definition of 15. That means that patients with pH and less than 15 milliliters of mercury may be included in clinical study, clinical trial for pH therapy. And in this paper, you can see, I think the most important point is on the right part. If you look on individual data or forest plot of patient and response to treatment according to the wedge pressure in different clinical trials, you can see that the patient who have between 13 and 15 at baseline of wedge pressure and pH respond in the same way that patients with wedge pressure lower than 12. That doesn't mean that the patient have no left heart disease. That just mean that patient with less than 15 may respond to treatment. So to date, clearly there is no robust data to change the threshold of wedge pressure. And based on the clinical classification, the aim of the clinical classification is to determine the best treatment for the patient, and to that to have precapillary pH, and wedge pressure lower than 15. We can say from the clinical trial that this patient may respond to treatment. So finally, we kept the threshold of 15 in the definition. One word on exercise pH. Exercise pH, we kept the same definition, mean PAP on cardiac output slope, higher than 3 millimeters of mercury by liters by minute, define what is exercise pH. And it is clearly associated with a worse prognosis as compared to patients with normal hemodynamics at rest and exercise. Two major limitation from this definition. It's not an easy way, it's a slope, so you need to calculate the slope, and so it's more for very expert centers. And the second major limitation is that you have not, with this definition, the possibility to determine what is the mechanism of exercise pH. That means that if you have a post-capillary pH at exercise or pre-capillary pH at exercise, the patient had an increase of this slope. So we need to have more data to better characterize pre-capillary and post-capillary pH at exercise. There is some interesting parameters. I think one of the most interesting is wedge pressure and cardiac output slope, and that could be helpful to determine if it's a post or pre-capillary pH. With another limitation is that it's very difficult to have good wedge pressure during hemodynamic at exercise. So clearly we need to work more to define what is an pre-capillary pH at exercise, but to date we have no robust data on this. So finally, we decided to keep all the same definition of pulmonary hypertension with 3 criteria: mean PP and then the threshold of 20, wedge pressure threshold of 15, and PVR with a threshold of 2, and exactly the same definition of the exercise pH, but this definition cannot be able to define precapillary and postcapillary pH at exercise. So we move to the classification. You know this classification from the guidelines: 5 group, group 1 pH, rare disease; group 2, a very frequent condition, pH associated with left heart disease; group 3, another frequent cause of pH associated with lung disease. So group 4 is dedicated to pulmonary artery obstruction and mainly chronic thromboembolic pulmonary hypertension. And the group 5 is pH due to a disease with unclear and or multifactorial mechanism. So the first question we have in the task force: should we keep the same structure for the classification? To remind you, the general purpose of the clinical classification is to characterize clinical condition associated with pH according to similar pathophysiological mechanism, clinical presentation, hemodynamic characteristic, and finally, therapeutic management. The 6th World Symposium and 2022 ESCRS guidelines propose a very simplified classification, including children and adults in 5 groups, and we discuss about if we need to change this classification based on genetics, on pathophysiological mechanism that leads to pH, but to date there is clearly an absence of sufficient robust data to propose a classification based on pathophysiology or mechanism. In the future, if we are able to propose a personalized medicine, probably we need to identify some specific mechanisms, some specific phenotype, and specific treatment, but to date we are not in the field of personalized medicine in pulmonary hypertension. So we suggest to retain the core structure of the clinical classification in 5 groups. Some specific discussion we have on classification. If we look very carefully on the 6th World Symposium and 2022 ESURS guideline, you can see that for response to acute or long-term responders, there is discrepancy between these two classifications. In the 6th World Symposium, we propose to include for the first time a subgroup that is PH with long-term response to calcium channel blockers. And in 2020, it was changed, and there is a subgroup in idiopathic based not based on the long-term response to calcium channel blocker, but based on the acute response. All these two proposals have some interest, but also have some limitation. If we use acute response in the classification, the positive point is that with a positive acute response, predict a potential long-term response to calcium channel blockers and may be helpful to define management. You have an acute response, you propose calcium channel blockers. But not all patients with acute response demonstrate sustained clinical and hemodynamic improvement on CCBs alone, and it's defined management rather than a specific phenotype, and in all other parts of the classification it defines phenotype. If we use long-term responders to calcium channel blockers, the good point is that long-term response is a specific entity with a significant better prognosis, a specific management, probably different pathophysiology, but classification may change over time, and it may take 1 year to define if it's a long-term or not response to calcium channel blockers. Recently, this paper published in 2024 showed very interesting data on patients with acute response. You can see the prognosis of all acute responders, and the prognosis according to the long-term response to CCB or no long-term response to CCB in these acute responders. And you can see that in patients with long-term response to CCB, you have a normal survival, and it's really, I think, a specific phenotype. If we look on patients who have acute response but no long-term response to CCB, the prognosis was very worse and very close of what we observe in idiopathic PH. So that means that patients with acute response and no long-term response are probably not a specific phenotype. And in this paper, they show that mainly long-term responders are mainly idiopathic PH patients. And in a paper we have published in 2012, we also show that it's not only idiopathic, but you can have long-term response in the same proportion in patients with appetite suppressant associated PH. So it's mainly idiopathic, and for some patients, anorexigen associated PH. So this is our proposal from the task force. We propose to keep and to use a subgroup in idiopathic that is called long-term responders to calcium channel blockers. That means if you are, whatever was your response, you are acute response, you are an idiopathic PH. If you have a long-term response to calcium channel blockers after 1 year, you can be considered on the subgroup of idiopathic PH, long-term responders to CCB. And this patient have a specific phenotype, and we want to identify this subgroup because we think that we should work on this to understand what is the mechanism that leads to long-term response to CCB, specific pathophysiology, whatever. And we put also mark to say that long-term response may be also observed rarely in irritable, but also in anorexigen associated PH. The second part of the classification was based on drugs and toxins, And we define definite association when there is an outbreak, epidemiological or pharmacovigilance study or large multi-center study, and a possible association if there is some multiple case theory or case with drugs having similar mechanism of action. We had some factors. We identify the reversibility of pH, but not for all drugs. It was well known in pH. Existence of preclinical experimental data, if we have pathological findings, and if we have common mechanism or potential mechanism that leads to pH. And that will be very short, but we review all drugs to define which drugs may be classified as definite or possible. And I give you one example of a drug that is moved from possible to definite. It's carfilzomib, a proteasome inhibitors. We have data from registry, large registry, pharmacovigilance registry, international pharmacovigilance registry. We have also data from disproportionality analysis. We have also some data from labs, and so we have all the criteria to put them in definite association with pH. And we do exactly the same work for all drugs, and this is a list of drugs with possible association. And this is an update list of drugs and toxins. Carfilzomib and mitomycin was moved from possible to definite, and we had two drugs in the possible drugs associated with pH, bevacizumab and bortezomib, that is another proteasome inhibitors. I will be short to finish because it will be discussed. We better classify pH associated with Lufter disease with specific subgroups to better identify some entity. For group 3, very important point, we change from the obstructive or restrictive pattern to disease, COPD, interstitial lung disease, combined pulmonary fibrosis and emphysema, and non-parenchymal restrictive disease. Very important shift because group 3 was the only group not defined on disease, and now with the new classification we have disease for all these subgroups. So group 4 was not changed, and the group 5 we keep sarcoidosis in this group 5 because we consider that it's clearly multifactorial. Also some histiocytosis because it was well demonstrated that it's a chronic respiratory disease but with specific vascular involvement, and in the same way neurofibromatosis type 1 that has also a specific vascular involvement. So this 3 respiratory disease was kept in group 5. Only one changes, another changes is that we include complex congenital heart disease in group 5. So in conclusion, we do not change the definition of pulmonary hypertension based on mean PAP, wedge pressure, and PVR. And for the classification, we discuss all these points, we included long-term response to calcium channel blockers, some mild modification in pH group 2, major changes in group 3 to include disease and not obstructive or restrictive pattern, and only very mild changes in group 5. We include complex congenital heart disease. Thank you for your attention.
Kranz from University of Cologne, Germany, to take us into the journey of an update, the 7th World Symposium on the diagnosis of pulmonary hypertension. Stefan.
Good morning, everyone. And first of all, thank you very much for the invitation to SAPH2025, which happens to be another excellent conference here. So I'm very happy to be here again. So my task now is to focus on the diagnosis of pulmonary hypertension and the update from the 7th World Symposium in Barcelona. Hope this works here. Here we go. Conflicts of interest, as always, the start. And then we have to look at diagnosis. If we focus on the World Symposium, I'm going to do the same thing as David just did in his first talk to also relate this to the updated ESC-ERS guidelines, so obviously there is a connection between the two. And I want to start with the diagnostic algorithm that we put together for the ESC-ERS guidelines, and most of you will have seen this. Just a couple of important points here. So 3 steps are listed here. Step number 1 is the suspicion of pulmonary hypertension. I guess we all still face a significant delay from symptom onset to the establishment of a diagnosis of PH. And that's why this algorithm also follows, if you wish, the path of the patient through the medical system, starting at the GP, and this is where suspicion comes into play, then to specialists, be cardiologists or pulmonologists, where we need to detect pulmonary hypertension, and then finally at the center, confirmation by right heart catheterization, but also subclassification, as we have just heard from David, according to the clinical classification. And I put a red box there in the middle, because what we highlighted also in this algorithm of the guidelines was that we probably need rapid cross-referral, particularly between pulmonologists and cardiologists in a disease where we frequently have lung and heart disease combined when we talk about pulmonary hypertension. And finally also, and that was not new, there is a recommendation for a fast-track referral in patients with suspected PAH or CTEPH, or those who have warning signs of right ventricular failure. So that's the guidelines algorithm. And then we have heard in detail from David about the updated clinical classification, and we have heard that the 5 main groups have remained. There were some subtle changes, but when we talk about diagnosis of pulmonary hypertension, it has 2 aspects. One is to diagnose PH in general, but then also to classify the patients according to the clinical classification. So in that context, the diagnostic workup requires a lot of detail to make this subclassification. So here is the diagnostic algorithm from the World Symposium, and as you can see in the middle now, there are 5 steps to follow. It goes from step 1, medical history, symptoms and signs of PH. Step 2 then is the simple non-invasive tools that will help us to detect PH, but also to detect potential underlying causes. Then crucial is step 3, where cardiac assessment by echocardiography, but also lung assessment come into play. Then step 4 to either detect or rule out CTEPH by perfusion imaging, then additional testing, which includes assessment of exercise capacity, potentially cardiac MRI, and then finally step 5 at the center, right heart catheterization. And the recommendation here is really to perform right heart catheterization at the centers, because quality of hemodynamic assessment remains a problem outside of the centers. So in the guidelines, we kind of visualized the symptoms and signs of pulmonary hypertension. We see the symptoms here on the left at different stages of pulmonary hypertension, from early to late, as you can see. Obviously, the symptoms that we have are not specific for pulmonary hypertension, but they may be suggestive of pulmonary hypertension. And the white box at the bottom lists rare symptoms which may occur due to pulmonary artery dilation, including exertional chest pain, which may be due to dynamic compression of the left main coronary artery in patients with PH who have an enlarged PA. Then on the right, we see clinical signs of pulmonary hypertension, clinical signs of right ventricular backward or forward failure that may be found in these patients, and then finally also signs pointing towards underlying causes of pulmonary hypertension, be it venous thromboembolism, be it scleroderma or connective tissue disease, be it signs of congenital heart disease that we clinically can detect in some of the patients. Then obviously a key aspect in a diagnostic workup is cardiac assessment by echocardiography. And here we need a structured approach to the assessment of the right heart. And the guidelines provided those two images on the left, a schematic highlighting where we should measure, what we should measure, and what is important to get information about the right ventricle, but also the right atrium. Then on the right, we see the recommendations on how to utilize echocardiography in order to establish the echocardiographic probability for the presence of pulmonary hypertension, which can be low, intermediate, or high. And the first step at the top here is to look at the peak tricuspid regurgitation velocity. And despite the changes in hemodynamic definitions, those measures or those cutoffs have remained the same in the guidelines. Then we would also consider additional echo signs that may be suggestive of pH, and then in order to make a decision for invasive assessment, we would also consider additional risk factors that are associated particularly with PAH or CTEPH. And one of the things that was newly introduced in the guidelines was to combine or to integrate right ventricular function and right ventricular afterload, if you wish. And here we suggested to look at the TAPSE over SPAP ratio as a surrogate for what we would call RV-PA coupling. And that was based on a nice study that was carried out by Rodretello from Gießen, who validated this simple to calculate echo ratio to conductance catheterization being the gold standard to assess RV-PA coupling. And that was a nice study that had different aspects. So it related various echo single parameters and then also ratios to the standard, gold standard conductance catheterization. And it also highlighted that the TAPSE-SPAP ratio is predictive of survival in patients with pulmonary hypertension. So something easy to calculate and something to consider in the patients with suspected PH, but also for PAH. And back to the World Symposium of Pulmonary Hypertension, these are the images from the paper on diagnosis of PH, and it lists also the different echo parameters at the left and how they should be measured. And they also list the additional echocardiographic signs that may be suggestive of PH. That's the table on the bottom right. And I highlighted that also there the TAPSE-SPAP ratio, among other parameters, has been added with a cutoff of 0.5 millimeters per millimeter mercury. And after the guidelines have been published, this study came out looking at the prognostic impact of the TAPSE over SPAP ratio in the context of risk stratification in patients with PAH. And that study has nicely shown that yes, if you add this to the risk assessment parameters, it works in terms of prognostication, and importantly, the cutoff to define a low mortality risk in this study was 0.33, and that is very similar to what was shown in the guidelines. So this is important for diagnosis, but also for risk stratification in the end, and we'll hear more about that in the later talks. So also then to establish the diagnosis, obviously right heart catheterization is the tool to use, and there are a couple of recommendations here for right heart catheterization in the guidelines, and the last one may seem trivial, to get the complete hemodynamic or the complete set of hemodynamics. The guidelines list this table of right heart cath variables that are directly measured or calculated, and it's important to note that we need all of this information to properly define a patient's hemodynamic status. And we've already heard in the last talk by David about the changes in the definitions, and you're all aware of this. And we've also heard that in terms of the diagnostic workup, no further changes for the hemodynamic definitions have been made at the World Symposium. And I want to highlight again, and this study was already shown by David, that, and I view this as very important, after the guidelines were published, additional studies, and this is just one example, confirmed the prognostic relevance of the updated definitions. That's true of both the mean pulmonary artery pressure and pulmonary vascular resistance. And finally, the wedge, also that was mentioned by David, cutoff of 15 remained the same at the World Symposium for good reasons, and I listed the two main reasons here. Again, on the left, we see the upper limit of normal in apparently healthy adults, and that was 13 millimeters of mercury, so there could be an argument to actually lower the cutoff, but the study on the right, that's an important one, because that has shown the therapeutic efficacy of PAH drugs related to the wedge pressure, and here the efficacy was there to at least 15 millimeters of mercury. At the same time, I want to make the point here that we shouldn't overestimate the impact of the wedge pressure in the diagnostic workup, and then also the classification of the patients with pulmonary hypertension. But we have to interpret that in the clinical context and also related to specifically the echocardiographic phenotype of our patients. Then, if we focus on the updated clinical classification of PAH, I want to also briefly focus on the what's called here long-term responders to calcium channel blockers. And in the ESC guidelines, this subgroup of group 1 was classified a bit differently. Here, it's not about the long-term responders, but the acute responders at vasoreactivity testing. So both of these have advantages or disadvantages. If you consider the World Symposium classification, you don't know at the time of diagnosis whether a patient would be a long-term responder to a calcium channel blocker, but you have to write down a diagnosis. So from that perspective, it would be probably better to have the term acute vasoresponder, because that's what you get during your diagnostic workup. At the same time, we have learned that not all acute vasoresponders are long-term responders to calcium channel blockers, so that would argue for the term in the Barcelona manuscript. And we have specific recommendations for acute vasoreactivity testing during the diagnostic workup in the guidelines, and I want to keep this short here. We have the criteria that most of you will be aware of. Criteria for an acute vasoresponder: drop in mean PA pressure by more than 10 to below 40 without a decrease in cardiac output. And then the therapeutic consequence of that would be to treat these patients with calcium channel blockers. But what is also mentioned here is that we need a close follow-up of these patients. It's not that patients are vasoresponders, you give them calcium channel blockers and everything is good. David had already quoted this paper about vasoreactivity testing and acute vasoresponders, and he has shown already that depending on the patients under treatment with calcium channel blockers meet the criteria for a long-term response or not, that has impact on survival. So what can we learn in terms of initial diagnosis in this context? Here in this study, we also looked at pulmonary arterial compliance, and we see that a PAC, and the cutoff here was 2.5, was, in addition to the other hemodynamic parameters, predictive of survival. And on the right, we see the prediction of a long-term response to calcium channel blockers by pulmonary arterial compliance. So something that we may, if we do the vasoreactivity testing, consider in addition to just pressure, look also at PA compliance. And now about phenotyping of the patients with pulmonary arterial hypertension in the diagnostic workup. So in the end, we want to know how can we help the patients, and then we have the treatment algorithms from the guidelines and from the World Symposium. I want to keep this very short here. I just want to make the point that the phenotyping in terms of comorbidities is integrated into the ESC-ERS treatment algorithm. It's not directly integrated in the treatment algorithm of the World Symposium. Nevertheless, it's stated there in the text that we have to precisely phenotype the patients and then come up with a diagnosis. And obviously, phenotyping in this context is then important. And if we think about the comorbidities in this context, we have learned that age is obviously an important factor, but then we may have comorbidities that could contribute to pulmonary hypertension, to an increase in pressure, and we have others that may be present independently from hemodynamics and from pulmonary hypertension, and then we also have environmental factors and habits, including smoking status. And these comorbidities have impact on the symptoms, some of them on hemodynamics, on the treatment patterns, also on the drug tolerability, and even on risk and survival. So it's important to consider them. And we have learned that there were two main phenotypes in the context of comorbidities that we should consider at the time of diagnosis. One is the pulmonary phenotype, and here data from two large registries, Compara and Aspire, have taught us that if the DLCO in the patients is low and or there's a smoking history, then such patients that were labeled in the registries as PAH were very similar in terms of treatment response and survival to the patients with group 3 PH. So when we do the diagnostic workup and the classification, DLCO, we have ignored that in the past, is something to consider, and also the smoking history of the patients. And then we have the cardiac phenotype that was initially introduced in the AMBITION trial. So here we look at factors or risk factors that could be suggestive of left heart disease and heart failure with preserved ejection fraction. In particular, also something that we should consider at the time of a diagnosis. The guidelines list this table of parameters, clinical parameters, imaging, hemodynamics, that may help us to properly distinguish between group 1 PAH in patients who have comorbidities and group 2 PH, and this obviously has direct implications on treatment decisions, so it's important to do our best in order to phenotype these patients and classify them then as group 1 or group 2, and that's not easy. And if we talk about PAH with specifically cardiometabolic comorbidities, what we usually talk about is we have elderly patients, and at the time of diagnosis, we look for the presence of such comorbidities. But what we also see is many of the patients who were diagnosed at younger age, now due to the treatments that are available, live longer, and as they age, they may develop comorbidities over time, something that also in the diagnostic repeat workup is important to capture. And finally, sometimes we have patients with genetic variants, so we have heritable PAH, and if we ask in this context about relatives, we usually ask about siblings and their and children of the patients, but not necessarily their patients. But we have seen, and that's not rare, that all of a sudden the patients would show up, and obviously they are then at advanced age, and you do the genetic testing and you find the same genetic mutations and pulmonary hypertension even in the patients. So that's an example of, so to speak, proof of heritable PAH also in elderly patients with comorbidities. And in the diagnostic workup, that may also highlight the importance of genetic testing in this context. When we interpret data from patients with comorbidities, it's probably important to discriminate between data obtained in randomized controlled trials, where we have inclusion and exclusion criteria, and data that were captured from registries, where the patients with comorbidities may even be more complex. So if we talk about phenotypes, it's important to consider clinical variables, imaging, and hemodynamics. And we may have pure PAH at one side, then things are clear. We may have clear group 2 pH, elevated wedge pressure, and also signs of left heart disease, and then we may have something in the middle, and that's when the diagnostic workup and the interpretation of the data may be difficult. So in this context, PAH with metabolic comorbidities, we may have cases that are more to the left, elderly patients, moderately impaired hemodynamics, clinical signs, echo signs suggestive of left heart failure. We probably wouldn't treat such patients, and we would be reluctant to call this PAH still. We may have something that's more in the middle, higher PVR, the clinical aspects that are more suggestive of a predominant right heart condition. And then we have something that may be more close to the green box here, elderly patient in the mid-50s with well-controlled comorbidities and severely impaired hemodynamics. So these are the phenotypes that we may see, and we have to interpret whatever we find during the diagnostic workup in this context. And this nice image I really like, because we have to consider not only the number and the presence of comorbidities, but also the severity or the control of these comorbidities, and relate that to the severity of pulmonary hypertension when we classify these patients. And that's obviously not trivial. So finally, last two slides here. In group 2 PH, the World Symposium has introduced this staging of patients with pulmonary hypertension and left heart disease, stages A to D, and that also requires specific information that is captured during the diagnostic workup of these patients, and I'm sure we'll hear more about that later today from Jean-Luc Vachery. One important thing, and that's a very new paper that was published by Jason Weatherald and colleagues, is what about the phenotyping characteristics in patients with pulmonary hypertension and left heart disease? And the point that is made here is we may have reversible pulmonary hypertension, that's shown on the left, and we may have persistent pulmonary hypertension. Persistent means it's expected to persist even if we get rid of the left heart condition. And there are several contributors, which may be genetic modifications, molecular and metabolic aspects, but importantly also the chronicity of the disease. There may be a point of no return when pulmonary hypertension becomes the predominant problem, and several things are listed here that we may use in our diagnostic workup, particularly LA size, but also left atrial function or left atrial stiffness that gives us additional information in this context. So finally, I just show here four chamber views of echocardiographic phenotypes. All of these patients may have pulmonary hypertension, but obviously if you look at specific features in the left heart and in the right heart, it looks totally different. And that is also part of our interpretation, not only focus on the wedge pressure when you make your diagnosis, but also particularly look at the echocardiographic phenotype when making a diagnosis. And with that was a little overview of diagnostic workup, guidelines, and World Symposium combined. And I thank you very much for your attention. Thank you very much.
Thank you, Stefan. Thank you. Will you join us on the podium here? The questions will be afterwards. Please join us here. My pleasure to introduce Professor Anton Noordegraaf from University of Amsterdam, who will take us into the fascinating journey of the right ventricular function. Anton?
Thank you very much, and also thanks for this invitation to see wonderful friends, and also to recognize that it can also rain in Riyadh. I didn't know that, but in the Netherlands it's always raining, so I felt very much at home. my conflicts of interest. So I want to make it simple. We have discussed the right ventricle on the World Symposium, but I will also give a little bit a brief overview from the past, and then we enter in the area. So what is the load on the right ventricle in pulmonary hypertension? So what makes the right ventricle to adapt? Then what about the failing right ventricle? We have already heard about the coupling and systolic function. What's next? And then reverse remodeling of the right ventricle. Now, first of all, we had to recognize the normal right ventricle. The vessels are thin-walled in the lungs, distensible, huge recruitment capacity, and that leads to the fact that we have a very low pulmonary vascular resistance, that the resistance even go down when we do exercise, and so we have a very low pressure system, and the right ventricle is only responding to volume change. So if you are walking, the volume changes, increases, leading to an increase in cardiac output, which is of course what we need, and stroke volume goes up by 30%. And immediately we are in trouble if the load increases, let's say in acute pulmonary embolism, because then the right ventricle is not developed to meet such a load, and our patient is in a critical situation. And the right ventricle is also built differently from the left ventricle. It's a bellow function, and you can see it here, and the apex is moving to the tricuspid plane, and the free wall is moving to the septum, and the septum remains in the middle line, and the right ventricle wall is very thin-walled. So this is the bellow function like we know, a triangular-shaped right ventricle with also the trabecula inside. When we are looking to pulmonary hypertension, we are looking to advanced pulmonary vascular diseases, and this is already known for a very long period of time. So you needed a loss of probably around 70% of the pulmonary vascular bed before the pulmonary artery pressure increases. But after that, increases steeply. So if a patient meets the clinic with a mean pulmonary artery pressure of 50, we already lost a huge amount of pulmonary vessels. And of course, exercise might, because the cardiac output goes up, might indicate a bit earlier diseases, and we do know that this might be important to diagnose, for instance, in genetic PMPR2 carriers, the disease. The load of the right ventricle is based on the pulmonary circulation, and it has two components, actually three, but there are two major components, and that is one is the compliance, or the reverse is the stiffness, and that's the ease of distension, and we already, Stefan, talked about that, and the other one is the pulmonary vascular resistance. Now, the pulmonary circulation is developed in such a way that the amount of load spent on the pulmonary vascular resistance and compliance always remains the same, and that is unique because it's different from the systemic circulation. Why it is? There are several reasons for that. I will not go into that, but it makes things very simple. So this is the point. We spend around 77% of the work, the energy in joules, to overcome the pulmonary vascular resistance, irrespective of what the pulmonary vascular resistance is, and 23% on the compliance, so to distend the vessels. Why is it so important? Because if we know the pulmonary vascular resistance, we have a very good measure of the load on the right ventricle and the amount of work it has to be spent on. So we don't need the compliance measurements to know exactly what is the load of the pulmonary vascular resistance. This even stands in chronic thromboembolic pulmonary hypertension. This even stands in very central pulmonary embolisms. So this makes things nice and easy to calculate. That is why the compliance is important, but in terms of physiology, pulmonary vascular resistance gives you the possibility to calculate everything. Now, of course, the failing right ventricle in pulmonary hypertension we should recognize is not a failing right ventricle in its own, but it's failing because the load is too much. And actually, if we would carry the same burden as our patients, we would be dead in a minute because our right ventricle are not adapted. So our patients do have better adapted right ventricles than we do have to the load. So the failing situation is a very particular situation if you compare it to the left. And this is old work, but still relevant to recognize that if you look, for instance, to the contractility of the right ventricle, which is the ES, it is fivefold increased in the patients
in comparison to the controls. So the patients, of course, have the hypertrophic state. They have a better control function of the right ventricle. And this, of course, is needed to the load. But what is causing the problem? That the load is too much. And as a consequence of that, if you make the load is the arterial elastance, EA, and if you make the coupling, EAS, EA, then you see that especially during exercise, there is uncoupling, and uncoupling means a lower ratio, meaning that stroke volume would not go up or even go down, cardiac output can't go up, and that leads, of course, to decreased exercise capacity. So that's in a nutshell what happens in a patient. Now, of course, if you simplify everything, then you have, of course, the adaptive state, and then the patient might be in near functional class 1 or 2, and then the right ventricle is hypertrophic, slightly dilated, then it continues and to the dilated septal movements, and then finally we have the uncoupling, and then we have to reduce the output. Now, if you look to the advanced PAH and you look and compare this to the controls, you can see many differences. First of all, you can see that the apical movements to the tricuspid annular plane is decreased. So there is less movement, and that, of course, is also catched in TAPSI movements. Second of all, if you look to the movement of the free wall to the septum, you see that at a certain moment in time, it's not moving to each other, but even going away from each other. And that's due to the septum. So the septum moves to the right, or to the left, and the free wall is moving inward, but the septum movement is more than the free wall septal movement. It's very mechanical inefficient, and this is due to the consequence that the right ventricular contraction time is prolonged, leading to the fact that the right ventricle is still contracting, whereas the left ventricle is already in its diastolic phase, so the pressure drops down, and that leads to the left ventricular movements. Then, of course, you also can see this bulging of the apex, and the thin-walled apex. This is not very effective, and it loses its annular plane, and also changes the orientation of the myocyte fibers. And there's a lot of work done at this moment on this aspect of the right ventricle. So the myocytes are not in a good direction as they should stand in the correct right ventricle. One way to visualize the physiology is the pressure volume loop. It is a very old way to do so, and that is you measure the end diastolic volumes, the end systolic volume by means of conductance catheters, the pressures in diastole and systole, and then the cardiac cycle circles around, and that leads to the pressure volume loop. The normal situation, you have low pressures. The green one is more the adapted state. The difference between the two vertical lines is the stroke volume, and then the pink one is the non-adaptive state. And what you can see here is that stroke volume is lowered, that the end diastolic pressure is increased, and as well as the pressure, and that the contractility, which is the dotted line, is more going down, so it loses its contractility. And of course, this makes to all the parameters we do know are predictive of the response. And I do know that Atenai Buckley will present also the risk stratification, but still we do know from the old times that stroke volume or cardiac output, right atrial pressure, and also information of the pericardial fluids, which indicate indirectly the right atrial pressure, are highly predictive parameters. And this was already known for a very long time and has been confirmed by many invasive measurements and risk classifications afterwards. And I'm sure that this will be covered tomorrow or this afternoon. So this is also what is now in the guidelines. We have the hemodynamics, the right atrial pressure, and we have also in the echocardiography the right atrial area, and then also the right so we have two parameters which seems to be quite independent to systolic function. So what about right atrial pressure, and why is this so important? Now, this is something which also was discussed in length at the World Symposium, because it is not linked to systolic function. Sometimes you have a patient with a huge right atrial pressure and quite preserved systolic function, and sometimes it's the opposite. And it was already in the Alonso data, but afterwards found again and again that right atrial pressure is an independent risk factor. Indeed, diastolic function also matters in the right ventricle. So the right ventricle not only loses his systolic function, and I showed the images, but also needed to become more stiff. Why does it need to become more stiff? Because it has to keep its shape, its triangular shape. So that is why we do have stiffness, but stiffness can be too much. And if stiffness becomes too much, then the atrium has difficulty to kick in the blood, and then the right atrium enlarges. It is predictive. This is diastolic stiffening. And then you can see indeed, and then I just took a couple of patients, that you can see all phenotypes in the patients. Some of them have no dilatation, other ones have right ventricle dilatations. These are all long axis views of the right ventricle. Then we have this huge right ventricle and right atrial dilatation. And sometimes you have this phenotype of almost a normal right ventricle but a huge dilatation. And of course, tricuspid regurgitation also plays a role in that, but it's not only that, it's more related to the size of to the stiffening of the right ventricle. So I like ice cream, so I make a picture of that, how that could be the case. So the right ventricle has to choose in the process of adaptation between stiffening and not too much stiffening. And if you stiffen too much, then your right atrium will increase, then you have more the diastolic function loss, and otherwise the systolic function loss. We do know that the pathways are important in that, and the wind signaling pathway is important in that. And this is work from the Sony Palacetti lab, which indeed indicate that. And then, of course, we don't know why in PAH probably different decisions are made, as for instance, scleroderma, where you get more stiff heart. These are things we had to learn from the development of pulmonary hypertension, so this is open field. Now, why might right atrial pressure in itself also be important? Because it's not related to stroke volume. One of the reasons might be that the stiffening of the right ventricle leads to the right atrial contraction, and the right atrium is contracting to a stiff ventricle, not sending only its blood inside the right ventricle, but also reverse it to the vena cava. And the vena cava is of course connected to the liver and to the kidney. And that in itself might have the impact on all the organs we do have. So I do think the right atrial pressure just indicates and mediates the venous backflow, which has impact on all the organs. And we do know, especially for the kidney, that the kidney function is mandatory to survive in PAH. Then final word on the reverse remodeling, and it was already covered last night, but of course we want to reverse remodel the right ventricle, and the good point is that we can do so. So we can reverse the right ventricle by, for instance, putting in a new lung, and this also will be discussed this day by John Crenton, but another way is to lower the load very much by means of reducing pulmonary vascular resistance. So this is from the lung transplantation program. Some patients get a little bit of abnormal right ventricle afterwards, but in general speaking, the right ventricle reverses to very much. And there's almost no state that the right ventricle can't reverse. This is data from the endarterectomy, more recently published, and it showed again that after a couple of weeks already, but after 6 months especially, everything reverses, even diastolic stiffening and also the impact of the fibrosis on the right ventricle. So the right ventricle is really unbelievable in repairing itself afterwards, and everybody who has the possibility to face patients after lung transplant or after pulmonary endarterectomy will know these images from one day to the other, and so how to see the right ventricle reverses. Though this is good, then this point was discussed yesterday. We needed a certain amount, by Professor Gaillet, we needed a certain amount of pulmonary vascular resistance reduction to guarantee that the right ventricle reverses. So 20% of reduction in pulmonary vascular resistance might lead to a better 6-minute walking distance, but might not guarantee that the right ventricle reverses. And that long-term survival, which is determined by the right ventricle, is okay. This comes from 2 studies. In my view, I do think you can explain it as follows. Patient A, pulmonary vascular resistance, reduction of about 20% in pulmonary vascular resistance by means of a drug. Cardiac output goes up, but pulmonary artery pressures remain the same. So the right ventricle work even increased, and stretch on the myocytes, which is determined by the mean pulmonary artery pressure, remains the same. Patient B, you are more successful, probably by means of more drugs, and then also cardiac output goes up to the normal situation, but also pulmonary vascular resistance drops down to 50%, and mean pulmonary artery pressure drops down to 35 millimeters of mercury. This is quite important to get a drop in mean pulmonary artery pressure. Then the right ventricle work decreases and the stretch on the myocytes lowered, and then the right ventricle has holiday and has time to reverse to its normal situation. So that is probably that in a small amount of reduction in pulmonary vascular resistance, we only get an increase in cardiac output, and if we have more reduction in pulmonary vascular resistance, we can get also the drop in pulmonary artery pressure. And we all know the data from the French group showing that if you upfront have triple agent treatment, that you are really able to reduce the pulmonary artery pressure. This is coming from the mono treatment studies. Then you see that most of the mono trials, and that was just looking to the pulmonary vascular, to the stroke volume and the pulmonary artery pressure. If you look to the blue lines, you can see that most of them are a green area, meaning that the right ventricle has no benefit of this reduction, only stroke volume goes up. So the patients can walk more, but it has little impact on survival. And of interest in this, and this was discussed yesterday and will be discussed tomorrow more length, the STELLAR trial showed that mean pulmonary artery pressure can go down without an increase in cardiac output. Now, of course, you can debate in length what is causing this improvement, but also exact minute walking distance was improved, so it means at least that cardiac output at exercise is increased. And then you can see this reverse remodeling, as has been highlighted by Sousa et al. In the recent ERG paper. So that is a favorable, at least, response for the right ventricle to have a mean pulmonary artery pressure without too much increase in cardiac output. So, in summary, the load of the right ventricle in the pulmonary circulation is very easy to understand, and it's just, you can describe it well by means of pulmonary vascular resistance, because then you can capture as well the compliance. Pulmonary hypertension not only impacts the systolic function, which is of course is very important, but also diastolic function, and we are just at the point of learning how and why the patients choose the different paths between adaptation and what is the impact. And we also can learn a lot of congenital heart disease in that respect. Stiffening has an impact on the right atrium. The right atrium has to fill the right ventricle. If it's too stiff, then we have the venous reverse flow. So that has impact on the organs. And the good news of everything is, if you can cure the disease, the right ventricle will be okay. So don't worry about the right ventricle. We had to treat the disease well. Thank you for your attention.
Thank you, Anton. Will you please come join us here?
This is the World Promotion Group.
Come sit with us. David is with us online. So we'll open the floor for some questions. And to kick the question part, I have a question for both Stefan and David, if he's with us, about the mandate of having symptoms in the definition and in the diagnosis of pulmonary hypertension. At one point, that was very much stressed upon, that the patient has to be symptomatic. I understand for initiating the workup, but now we do get patients who are completely asymptomatic with very mild elevation in the PA pressure. Incidentally, they had a right heart cath for whatever reason, or an echo for another reason, but they are asymptomatic. Any comments from the guidelines?
Well, first of all, we have to define what means really asymptomatic, because sometimes if we ask the questions, do you have dyspnea, do you have shortness of breath, they will say no. And then if you ask further, what happens if you walk upstairs or something, then it becomes obvious. So I guess, number one, we might need a more specific, structured approach to assess the symptoms in our patients. That's the first comment, because sometimes asymptomatic patients are not asymptomatic. The second point is, if a right heart catheter is performed, there should be a reason for it, and usually it's symptoms. And I would argue that most of our patients that we see, at least with PAH, but also CTEPH, they're not in the range of 21 to 24 pressure or 2 to 3 PVR, they're far above. The only patients in whom we perform screening, obviously, are the patients with
with connective tissue disease, and there we will see such patients, and they may be asymptomatic. If they're asymptomatic, we should probably not treat them, but follow them and treat them if they become symptomatic or if the hemodynamics worsen over time.
But you know, the comorbidities in these patients are rheumatological, and often they don't ambulate. In fact, when you subject them to a 6-minute walk, they often fail that. So I think that's the point. Our rheumatology colleagues, probably they're just screening based on the, the criteria, but when we see them, we delve into their symptoms a little bit more. And more often than not, there is something, but they just don't do it because of the joint symptoms or the scleroderma or whatever.
But that's, again, taking me back into the same point, and it will take us into the hemodynamics definition of the pH. Now, in the referring centers, the average PVR or the average mean pressure will be in the 40s, maybe higher, right? Now, what are the chances that you will capture a pulmonary arterial hypertension with a mean PA pressure of 21? So the link between the symptoms and this level of the PA pressure and PVR is not quite there. So we end up seeing these patients having some secondarily worked up for some whatever reason, and they were discovered. The other problem that I'm facing in the recent past is that none, like let's say referring centers, the importance of having a very well calibrated right heart cath tools, it's getting us into the abnormally elevated, mildly elevated PA pressure, but the measures were not very well calibrated, and it's wrong. So when we repeat it in the referring center, it goes into the normal range below 20. So again, this is the impact that I see on these low threshold of picking up the patient with symptoms and really identifying them if they have COPD, for example. We see a lot of these cases because of the secondary workup.
That point is well taken, obviously. But I want to remind all of us that the definition is not restricted to PAH. And in fact, this borderline zone, but we may still call it that, may be much more important for instance, heart failure patients with left heart disease. Because there specifically, we see that even a moderate increase in pressure or resistance has quite dramatic impact on the prognosis of these patients. And we have tools at hand to control pulmonary artery pressure in this context. So it's not only PAH, but it's a general definition of pH, and depending on the underlying condition, we may do different things. And a very good example are the studies with CardioMEMS or other implantable pulmonary artery pressure sensors, where we have seen if we optimize the conditions on the left heart, including the volume load and diuretic therapy of heart failure patients, we can substantially reduce the decompensation rates and hospitalization rates of heart failure patients. And this is a good example for the importance of hemodynamics beyond PAH. And here I would argue the updated definitions are even more important as opposed to PAH, where we usually at the time of diagnosis have values that are much higher.
I have a question, but maybe Dr. Fatma has a burning question.
Sorry, yeah. I have a comment and a question, because you talked about the scleroderma. Why do we screen scleroderma patients if they're asymptomatic if we're not going to treat them? So probably if you find PH in a scleroderma patient, you need to treat. This is number one. There is a very small study that was done that showed if patients were found to have PH on the first examination, they did worse and they had worse mortality than the ones that they found that they have PH on subsequent, because they caught the PH earlier, even when they were asymptomatic. So like not committing patient to PH-specific therapy just because they're asymptomatic, what's the point of screening them? That's number one. My question actually, for Anton, I love the RV. I'm a heart failure cardiologist, and I love the RV. And you talk like 77% of the RV is due to PVR and 23% is due to pulmonary artery compliance. Because I've read the lectures beforehand, the titles of the lectures, I've done an exercise where I did 2 patients with similar PVR of 10, pulmonary artery compliance of like 2.5, 2.6, and one of them has a BSA of body surface area of 1.5. They will have a cardiac index of 40 mil per beat per meter square, and the other one will have probably a body surface area of 2.2 and a cardiac and a stroke volume index of probably 25 mil per beat per meter square. So my question is, we're just putting those numbers and we're labeling severe PH as one who has a PVR of more than 5, where even in the guidelines, those are not the indicators for severe PH. In the guidelines, we look for RA pressure, we look for cardiac index, stroke volume index, and mixed venous saturation. My question, why are we getting those values involved more, while we don't have, like, they might be very variable, and we will need to depend on the other variables in assessing how poor the pH is. That's number one. And number two, what do you think about RV stroke work index? Because in heart failure, we do use it as a guide for LVAD therapy, for advanced therapy. And do you think, like, RV stroke work index can be a good surrogate in assessing the patient's risk in PH? Thank you.
Thank you very much for these very valuable comments and questions. A little bit on the connective tissue disease back. Indeed, the connective tissue disease is a very special one. So even in the mean pulmonary artery pressure we showed in 21 millimeter mercury, you can have advanced pulmonary vascular disease. So that is indeed something we are going to detect, and that's probably why the guidelines give now some room to think about the treatment decisions. About the right ventricle determines the fate of the patient, and that's what you clearly indicated. This is also what clearly comes from risk factors. So we should classify PAH on the right ventricle symptoms, not on the pulmonary vascular resistance. You are completely right. However, if we think about the right ventricle load and we want to decrease the load, then we can have enough on the pulmonary vascular resistance. So pulmonary vascular resistance is not prognostic, not important in assessing the severity. But in thinking about reversing the right ventricle, we do know if we have a 100% reduction in pulmonary vascular resistance, we get a right ventricle who is very happy. And if you have only a reduction which is small, then the right ventricle might not be happy, because the right ventricle is serving the body. And how is it serving the body? By increasing stroke volume. Now, your point is about stroke volume index. It's a very critical parameter. If I had to choose one, I would choose that one. Because it's a coupled measure, of course. It's the right ventricle in relation to its load. And it is, it's corrected for the heart rate, so it's even better than the cardiac index. It's also coming from the data from Weatherbolt, but also from the MRI data, where you can assess it non-invasively. And stroke volume index is very important, and I would add another one to that, which is related to that, is just measure the pulse of your patient. If the heart rate is low, I do think you can be happy. Yes. If the heart rate is increased or on the EKG, oh. So take the stroke volume index is excellent, so I agree.
Thank you.
Thank you, and thank you for all speakers. Now, my question to Dr. David is regarding the responders. You mentioned that the long-term responders. Is there any study comparing those kind of patients when they put them on other vasodilator, whether monotherapy or combination therapy? And regarding the long-term responders, do we need to wait for the whole 1 year before we have an effect? Yes or no? Thank you.
Thank you very much for the question. I hope you hear me well. Regarding long-term calcium channel blockers, we have some patients who failed during time, but in fact, most of the patients who are long-term responders at 1 year continue to respond to calcium channel blockers. Usually, if a patient fails to respond to calcium channel blockers, we continue calcium channel blockers and add specific pH therapy. There is no good literature on this and how the patient responds. I'm not sure that they respond as well as idiopathic PH to the over treatment. And after, regarding the question about the response to calcium channel blockers, if we look, there is at least one paper, and we have finished to analyze our data also. I think quite all is done at 4 months. But most of the patients who fail very quickly, and after we have long-term response. But usually the best time probably to determine is between 8 to 12 months to consider the patient for a long-term responder to calcium channel blockers.
Identify these patients. Khalid?
Yeah, hi, Khalid Saleh from Abu Dhabi. Question for Dr. Anton. Thank you for excellent lecture. So I have a question. I've seen in some patients when we treat with the triple therapy, the cardiac output actually becomes supranormal, too high. We get worried. What do we do with them? Sometimes we cut down the medications or because we are concerned, does the RV like bounce back to produce supranormal cardiac output? And what do you do in this situation?
It's a very tough question and a very important one, and that's the supernormal cardiac output. And I do think the what is guiding in our life, the physiology, is the baroreceptor. And the baroreceptor is about getting the pressure in our head, okay, so that's how we are made. And that is when we drop systemic pressure by means of our PAH medication, and that can be, of course, happening if you give triple agent and procyclin at a very high dose, that you are over-activating the right ventricle to produce cardiac output. And actually, this was already recognized a long time ago. That's the paradox between therapeutical success and not success in survival. So that was at the time of the calcium channel blockers, when they were provided to patients who were not responding. Those patients had a drop in systemic pressure. They had an increase in cardiac output, but were failing very quickly. So I do think if you see this and the cardiac output is too much, then you had to probably to titrate it a bit down. I do think the strength of the triple agent treatments is that you don't increase that procyclin to the high levels. So normally, that's my opinion about how to deal with that.
I guess that's a real important point. And I mean, if we extrapolate to the left ventricle, there are very good data on the hyperdynamic LV and mortality. And then the mortality curve with the EF rising is very steep. And we don't have good data, I guess, for the right heart, but at least for the left heart that has been shown very convincingly. Hyperdynamic conditions is obviously not good. Perhaps finally a question to you, Anton, from my side. Brilliant talk as always. My question is related to the pressure-loaded right heart versus the volume load. Your images have nicely shown the changes in the dilation that we all see in our patients. And in this context, we also frequently see tricuspid regurgitation. So I guess the concept that we always say it's a pressure-loaded RV is only partially correct, and we tend to ignore potentially the impact of the additional volume load on the right heart. And in the context of tricuspid interventions, we have learned that also the geometry, RV and RA, is really important. We have the atrial TR versus ventricular TR. What's your opinion on the additional impact on the volume load and also the impact of the tricuspid regurgitation in this context?
Yeah, thank you, Stefan, for this question. So the huge right ventricle, of course, has a huge tricuspid regurgitation because the valves don't fit to each other. This can go up to 20% volume pendling around. And we have measured this by means of MRI, so it can be significant, 20%. But then, of course, some inefficiency is needed for the right ventricle, because if the right ventricle has to push its work all down in the pulmonary circulation, and the right ventricle would be mechanical very efficient, then this could also cause a problem, because this would lead to high peak pressures. So we did some modeling, Japanese fellow with me, on this. What would happen if you close the right ventricle in the presence of the tricuspid, in the presence of significant pulmonary hypertension? This would lead to immense amounts of peak pressures, even although it's 20% of the volume. So that is why it is recommended also by the cardiologist, tricuspid regurgitation, valve replacement or whatever,
is only needed in case of, can only be done if the pulmonary artery pressure is really down. And I do think that can happen sometimes after endarterectomy or whatever, when there is something wrong with the right ventricle, for instance, after pacemaker leads or whatever is done with the right ventricle. But normally, closing of the right tricuspid regurgitation is helping mechanically, but is killing the patient, in my opinion, based on physiology and the peak pressure. Do you agree?
If I can also add, we know from the left ventricle, when you have severe mitral regurgitation, the pathophysiology is continuous ventricular dilatation. So by arresting the mitral regurgitation, the LV can remodel positively. So now we have two problems, right? You have the tricuspid regurgitation causing RV dysfunction, plus the elevated pulmonary vascular resistance causing the pressure volume overload. And I think we also didn't discuss the patients with ASD, right? Because then they also have another volume load as well. So these are very complex interactions, and I think the RV is just coming into its prominence because of the treatments we have. I do actually have a question, and it's a little controversial. It relates something to what you mentioned about the data from STELLAR. So STELLAR, there's a remarkable clinical response. We know that, it's clear. There's a very good PVR response. Is there any physiological explanation? I've read as much as I can about the lack of cardiac output increase, in the face of which there's a tremendous clinical response. How can we postulate that?
Well, I do think there is a cardiac output response, and if you look to the supplementary data, it was not really significant, but the patient with the lowered cardiac output had at least more increase in cardiac output than the patient with the almost normal, and this was also quite a prevalent population. I do think it is important also to study the possible impact of sotatercept on the right ventricle, and many studies are on the way. But so far as I can understand, if you have no impact on the stroke, on the systemic pressure, and sotatercept doesn't have any impact on that, and your cardiac output is normal, cardiac index is normal, you don't need an increase in cardiac index. So it's not necessarily wrong. I don't think it's wrong. And actually it's very helpful because that is not increasing this cardiac index to a supernatural state. So indeed we had to go in depth to study the impact on sotatercept, but I do think it's physiological explainable what happens. Yes. And probably it's the best way to happen to the right ventricle because that reverses the right ventricle.
Right, thank you.
I would like to thank all the speakers in this interesting session. This concludes the morning activity for SAPH for today. We will have a relatively long break up until 1:30 where people can catch up with the prayer, and there will be lunch served in the ground floor, below, one level below this one, where we had dinner last night, up until 1:30. Please come back at 1:30 sharp because that's when we will start, and we promise you a very interesting session coming up starting at 1:30. For the time being, thank you, our esteemed speakers. The next activity will be a pharmaceutical-sponsored activity by Johnson & Johnson. Our speaker, Hani Sabbour, is our friend, colleague, and a long-time SAPH member. He will talk about triple therapy in pulmonary hypertension. Hani, the floor is yours. He's coming from UAE, cardiologist.
Thank you very much, Hussam. It's always a privilege to be part of this incredible meeting. The level of discourse and discussion here is really superb. So I'm going to talk about something practical. This is the underutilization of combination therapy, a lethal inertia in a lethal disease. This is my QR code. If anybody wants the lecture, has a question, wants to discuss a patient, I'm always ready to do that. You scan the QR code, it comes up on WhatsApp. And I will send you the slides, I promise. So we'll talk about the underutilization of combination therapy in the modern era, which is a lethal inertia. We talk about PAH disease progression, the treatment of PAH, and classifying available evidence. So the underutilization of combination therapy is a common theme. Now, if you consider, has the survival of PAH patients over the decades improved? In fact, even in the setting of triple therapy, oral, sub-Q, IV, I'll note to you these percentages. Mortality rates of 8 in 1 year, 16% and 21% in the FAR registry, which combines recent updates, is very high.
If you leave these patients to progress, there's an increase in mortality of up to 30%, and you can see here in the third year, these patients using the Compara or Reveal Light score, 28 to 55%. So the intermediate risk patient is the patient we need to target for more therapy, because that's when you can start to shift the curve downwards. So the rates remain unacceptably high, even in the presence of advanced and utilizing combination therapy. And there's got to be a reason for that. So why is the survival still suboptimal? We've seen the randomized clinical studies, we've seen the registries. This is the reason. Therapy use at the time of enrollment and follow-up visits in large registries, triple therapy, only 6% at enrollment, 19% at last follow-up. How does that advance? Survival is poor. So you really need to think about how combination therapy can help your patient and why you need to continuously advance it. The reason is finally our problem. Most patients still do not receive therapies targeting all three core pathways. So PAH disease progression. We're very familiar with this. We've seen this the entire of all of the lectures, and you see this change of the structure of the right ventricle. So there's RV remodeling resulting from vascular remodeling as well. This is a progressive disease. So the guidelines and the World Symposium have all made the same context very clear. The goal of PAH treatment is to focus on long-term outcomes by managing disease progression. And just to summarize, achieving and maintaining low-risk mortality profile on optimized medical therapy has been the statement from the 2022 guidelines and beyond. So it's fairly clear this is what we need to do. Optimizing therapy, maintaining low risk. So the progressive worsening of the pathology of distal pulmonary arteries requires us to halt the disease progression. Why? By addressing the three components of endothelial dysfunction. And if you don't do these steps, the patients will progress, and these are the problems. The arterial wall, the endothelial cells, fibroblasts, and smooth muscles each have a role in PAH pathogenesis. So there are three core pathways that are important for balancing endothelial dysfunction. So endothelin-1 antagonist, nitric oxide pathway restoration, prostacyclin pathway in the context of all three. So when you look at the prostacyclin pathway, that's the pathway that is most often missed in clinical practice, right? It's the one we most fear because of side effects. There's an issue of access. This is where most patients fail. You find patients on monotherapy or dual therapy for a long time, even though they're progressing. So achieving and maintaining a low-risk profile is the key objective in treatment of pulmonary arterial hypertension. And how do you do that? Do you depend on symptoms? Absolutely not. You give these patients a little bit of sildenafil, a little of diuretics, they feel a lot better because they were struggling before. But you have to do the sequential evaluation at the first visit, which is key, and then every 3 to 6 months. This is to evaluate and assess prognosis, treatment success, and plan treatment escalation if it doesn't work. So what I say is early assessment means early intervention, regular monitoring. And we're supposed to go for green. Green in the guidelines is the lowest risk group, less than 5% over the 1 year. Now, can patients feel better and appear to be clinically stable with RV remodeling? Absolutely yes. Lack of symptoms while the RV remodeling progresses is a feature of many of these clinical studies. 6-minute walk here appears to be stabilizing above 400. Functional class appears to be 2. However, in the same patients, the RV end diastolic volume is increasing. RV ejection fraction is dropping. The same identical patient. So the symptoms do not tell you exactly what's happening to the RV. And this is one of the things that the 4-strata model is not giving us. It only depends on these criteria, and we may be missing a lot of RV dysfunction, and that may be too late. So structured assessment is key. Now, the 3 core pathways need to be targeted. In any patient who is not low risk on double therapy, not low risk means not low risk. Intermediate, intermediate plus, intermediate, high, and of course high. You need to target these patients in order, because of your risk assessment, to reduce the risk of death at 1 year. Remember, in our disease, mortality is more than 20% per year in the high-risk patient. and it's not much better if you're intermediate high. So I think we really need to do everything we can, literally throw the kitchen sink at these patients to prevent this progression. And the prostacyclin pathway, which is either prostacyclin analogs or IP receptor agonists, is very powerful because it looks at vasodilatation and antiproliferation as well. So targeting the prostacyclin pathway is essential. Why do we need that? We need that in order to go for green. By early utilization of triple pathway in the non-low-risk patient, we can impact progression and disease. So this is some data from the Selexipag trials. Targeting the prostacyclin pathway in the Griffin study resulted in a 37% reduction of the risk of morbidity and mortality compared to placebo. This is a long-term study with over 1,100 patients, and this is triple therapy. In addition, 33% reduction of PH-related hospitalization, which, as you know, is another feature of time to clinical worsening and can precede mortality. So it's clinically very important. So let's think of this concept of remodeling. So it's been talked about before, and I think in the context of sotatercept, this is coming into another revolution. But let's look at the old data from the Selexipag, the preclinical studies. What happens with the monocrotaline or the Sugen hypoxia model in the mouse? Normal, placebo, and Selexipag. You clearly see the remodeling and the pulmonary artery whorl hypertrophy, and therefore you reverse vascular remodeling as well as impacting the RV function. So this is disease modification. Now, in the initial study, the phase 2 study for Selexipag, the primary driver at 17 weeks was a very early reduction in the pulmonary vascular resistance. And we've talked about this in depth in the previous session. 33% reduction in PVR compared to placebo, but it occurs early. So does hemodynamic disease modification occur early? And I would say based on this data, the answer is yes. So now we need to talk about early targeting of the prostacyclin pathway. So in this portion of the patients, these are patients, mortality and morbidity reduction, who received this early, and these are the patients who received it late. You had a 64% reduction early, 26% reduction in functional class 2. So clearly, the more effective you are is when the stage is earlier, and this is also affecting the cellular remodeling in the myocardium. The second component of this to remember is that functional class 2 patients are not stable even on dual therapy, right? If you remember the AMBITION study, the results were pretty similar. So here, the placebo arm on dual therapy has a significant difference compared to placebo, compared to Selexipag given in the early stage. So don't be fooled by functional class. And this is, again, when you look at the 4-strata model, the functional class may be missing a significant change in the vascular remodeling. Now, what is the clinical value of this disease modification? I would say it's reduction of mortality and morbidity reduction. So if you look here in the post hoc analysis of Griffin study, 55% reduction of morbidity or mortality, and in these patients were more than 6 months and less than 6 months. So what's this telling you? The earlier you intervene, the greater benefit you have in terms of the vascular remodeling process. So this is true disease modification coming to outcomes and delivering better therapy. So the earlier triple therapy in the non-low risk patient on dual therapy prevents long-term progression and prevents remodeling. When you look at the Selexipag study, there's consistent effectiveness across all the subgroups. So the overall mortality morbidity outcome, 40%, which was obviously adjudicated in connective tissue disease patients, in patients on dual therapy, in patients with 3 comorbidities or more than 3 comorbidities, in less than 65, more than 65, functional class 2 and functional class 3. All of these are statistically significant. But the benefit is greater when you start early. And I think that's a key component of this treatment. So when we look at the open label extension, which is the real world data, this is now 10-year survival, looking at evidence of long-term disease modification. So you have the extended data from the open label extension. This gives you an additional 57% survival at 10 years in 46,000 patients treated worldwide with Selexipag. So this is a very important concept to remember. You have a lot of evidence that supports the triple therapy pathway activation. Now, when you look at the historical milestones in PH, and these are the randomized clinical studies that led to approval, and of course the changes in the guidelines that followed that, we have a lot of long-term experience with triple therapy that is both evidence-based and real-world, and literally looking at survivorship and outcome. So, many drugs have also been developed to look at alternative pathways, non-vasodilator pathways.
from etanercept to anastrozole, many different pathways. However, many of these biologically plausible mechanisms have often failed. So you can't just go with biological mechanism, you have to have randomized evidence. Now, something that's new, and I think this is very important, is the addition of sotatercept as the fourth pathway. And we believe that the activin signaling inhibition with sotatercept, which targets the BMPR2 mutation, should be very effective in patients with the BMPR2 mutation. And this is something I came up on just yesterday as I was preparing for this slide. This is actually a patient with a GDF2 BMPR2 mutation on triple therapy who actually did not improve with the addition of sotatercept. I mean, mechanism may not mean universal utilization or success in every single patient. So these are the positive study, or the studies that have been positive in phase 2 and 3 that are moving forward, and some of them are quite interesting. SGLT2 inhibitor, we know seralutinib and sotatercept are moving forward with very powerful data. And these are looking at established and novel pathways. So now we want to look at the available evidence and best management strategies in the future treatment of pulmonary hypertension. So evidence-based guidelines are based on these rules: the level of evidence, the number of clinical studies, and their outcomes, solid clinical studies. Now, we've all seen this, and this was a unique presentation from the World Symposium. And remember, the World Symposium is forward-thinking. They're not just including the pure data and the evidence-based medicine. There are a lot of recommendations that come based on the experts. And here, we've seen the addition of activin signaling, oral inhaled PPA at the intermediate low stage, and here, activin signaling in intermediate high and high risk, where it was pretty much all intravenous prostacyclin at that time. So just to remember the process here, the ERS guideline from 2022, evidence-based recommendation, reviewing the evidence in some detail and coming up with a concrete pathway. The World Symposium is fantastic because it gives you a little bit more leeway. There's more flexibility to consider ideas and future thinking discussions. It's expert opinion. It summarizes the needs and future needs in PH. And it's a very important process, but it's not binding. It's more of a consensus and a recommendation for experts. So let's look at the PULSAR study. The PULSAR study was the first study looking at patients with sotatercept. And the mean time from diagnosis was 7.5 years. Most were idiopathic, 56% were triple, and a third of those were prostacyclin infusion. So this is not really an intermediate risk population. So this is where I have a little question for the audience. Intermediate risk, does this look like intermediate risk? I think not. The STELLAR study is, of course, the approval study, and this is a very powerful piece of evidence. 77% were idiopathic, 8.8 long duration from initiation on treatment, and the background therapy double is 34%, triple 61%, including a very large number on parenteral prostanoids. So these are much higher risk patients. And of course, as you know, the endpoint was 6-minute walk as the primary and additional endpoints as well, and the data is capped at 6 months. Now, again, we'll question the idea of intermediate risk. Now, the comparator, of course, is the Griffin study, and this comparison has been made a lot. If you look at Griffin, these are connective tissue, more, and idiopathic. The number is larger, 2.5 years, so they're earlier. And monotherapy, 47%, double background therapy, 32.5%. So it's 1,100 patients, earlier diagnosis, time to morbidity and mortality was the first endpoint, which is a hard endpoint, and we see a 10-year survival of 57%. So when you put these together side by side, they are looking at a completely different therapy in a completely different population in terms of size, duration, background therapy, and etiology, particularly connective tissue disease. So I just wanted to dissect this and show it. How do you apply this to what patient, right? So in the STELLAR study, you had only 64 patients, connective tissue disease and congenital heart disease. 96% were on triple or dual therapy, 61 on triple, 40 on parenteral, and a long duration. So these are long-term prevalent on a lot of treatment. That's a good kind of patient to consider the treatment for. When you look at Griffin, there are 444 patients, 38% are connective tissue and congenital, so very well represented. 67%, 700 patients monotherapy or even treatment naive, and 33% on dual, and the time to diagnosis is 2.4. So you would say these are earlier stage patients on good standard of care who need additional therapy to do what? To reduce mortality and morbidity, not just 6-minute walk. So the evolution of risk stratification is also an important point because you know now risk stratification ties you into the diagnosis and guides you as to what's your next step. How can we use it to overcome lethal inertia? So 2016 guideline, very clear. You have a lot of points to look at considering your CPET, your BNP, your imaging and effusion, as well as hemodynamics, right? A lot more points to do. But what's the point here? Go for green. Keep using triple therapy until every element is in the green. The updated guideline had two very important points. First of all, you have a mortality risking of more than 20% in the high-risk patient and up to 19% in the intermediate risk patient. So again, the concept of go for green is there. The addition, of course, of cardiac MRI specifically, the TAPSE SPAP ratio were innovations, and they also added the 4 risk strata. So I just want to stop on this for a minute. Is the 4 risk strata a granular test to tell you don't treat? That was never the intention of the 4 risk strata. The idea is many physicians, up to 70%, were not even doing risk stratification and follow-up. So let's give them a clue, a simple tool based on clinical criteria and a BNP. Is there something wrong? Should I do more? It's not saying don't do an echo. It's not saying don't escalate. It's saying, warning, your patient isn't right. Do something else. Check them out. And unfortunately, I've seen situations where people do the calculation here and inhibit themselves from doing another echo, doing another right heart cath, or escalating. That was never the intention of the guideline. So again, go for green using more detailed risk stratification. Use the risk stratification tool at every visit to keep an eye on the patient, and then low threshold to retest or add more information. We spend hundreds of thousands on the medication. We shouldn't be worried about spending a little bit more on doing an echo or a right heart cath in a patient who is not progressing properly. So the World Symposium had a very nice update to this point, and that is the value of adding right heart cath. So this is the analysis done by Professor Buckley, and I'm just going to unhide this so you can see it. What is the characteristic that you need? A right heart cath. So you do the non-invasive 4 strata, and it should drive you into a question. Does the patient need an additional investigation? Yes. Stroke volume index and SVO2, you're good to go. Here's your survival. You're in green. Stroke volume index is high. Stroke SVO2, one of those falls into the intermediate high. Your risk is not green, and you need to do something about it. So it implies that the 4 strata should lead you to a more thoughtful risk stratification and an earlier right heart cath. And I think these are the important points that we need to consider. So when we go back to the 22 guidelines, there's a lot of evidence guiding the utilization and positioning of selexipag in the 2022 ERS guidelines. So when we look at this, this is where you're going to do your follow-up. The first follow-up is critical. This has been pointed out. I don't do anything less than, I do 3 months. It's automatic, but I also see the patients 1, 2, and 3 as well. And you can't wait for 6 months, I believe. The evidence for selexipag covers from the initial therapy to the intermediate therapies, and therefore, this is where those group of patients have to be systematically reassessed, and if they're not low risk, consider the addition of the 3rd pathway. Of course, assuming the patients are on dual pathway, well, sometimes we don't even see that. So, this is clearly the area that you have to think about. When you do your risk stratification, you do it early and you continuously do it, you will find the patients in the middle who are going to benefit from that early disease modification process. Now, what's the evidence for it? And this is also referenced. So selexipag on double therapy within 6 months from Griffin and Triton pooled analysis, time sensitivity of doing it early. Within 6 months, you have a much bigger bang for your buck. If you also look at long-term outcomes, so Griffin open label, Griffin and Triton, plus exposure and sphere, these are real-world studies, this reduces disease progression and risk of hospitalization, and it works very well early. And most of the patients are in the intermediate risk after the combination therapy. And adding patients with selexipag not fully at low risk, i.e. here, which you can call intermediate low, Griffin, consistent results regardless of the functional class or risk at baseline, and real-world evidence, again, predicts patients at all risk levels can be initiated in clinical practice safely. And this is the group of patients we want to avoid going to high risk, and you do it by doing so early. So this is just to remind you of the overlapping area where the evidence supports the use of selexipag after initial combination therapy in the intermediate low patient to prevent progression to disease. So the long-term overall survival is what we've been talking about through my lecture. Overall survival outcomes with Select-IPAG in the Griffin open label extension also support what we saw in Griffin. So remember the top line data, 40% relative risk reduction on mortality and morbidity events, 30% relative risk reduction for PH-related death and hospitalization, hard endpoints. So consider what is the best early add-on therapy for your intermediate risk patients to maintain long-term outcomes. You decide, this is the data. So the 1-year survival, 92%, 3 years, 79%, 5 years, 71%, 63% at year 7. And over 46,000 patients worldwide up to 2023 have been treated with Select-IPAG. And the objective again is your decision, how to maintain long-term outcome in the patients who are not low risk at the first visit. So with that, I thank you very much. And here again is the QR code if you want this lecture. So we have some couple minutes for questions or concerns, hopefully no complaints.
So thank you so much, Dr. Hani. Brilliant as usual. How about, like, I understand that we need to add, and probably add is better than switch because add is a 2A recommendation, which is a 2B recommendation. How about those patients with like very severe side effects when you add?
Sorry?
So like very severe side effects like severe headache that make them just cut off the medication or just stop their phosphodiesterase inhibitor. Do you recommend switching and adding?
Okay. Fantastic question. So the practical management of prostanoids is always difficult. And I think as experts, we know where the prostanoids have an advantage and where the patients are going to have a problem. So first of all, it's about a discussion. Whenever I start a prostanoid, whether it's oral or sub-Q or an IV, I tell the patient the following. You have something that's in the realm of cancer in terms of mortality. You're going to have to live with it the rest of your life. The medication is probably initially as bad in terms of side effects as the disease itself, but we have ways to beat it. So I've learned from cardio-oncology colleagues that you manage side effects. When you have a cancer, you don't tell the patient, I'm not going to give you your chemotherapy because of nausea, vomiting, headache. And these side effects can be very intense, so we give the patients counseling and treatment to specifically attack these side effects. We also know that with any agonist in general pharmacology, but specifically with these prostanoids, there is some sort of a threshold. Once you reach a certain dose and you saturate the GI receptors and the jaw receptors, you don't have that side effect anymore and you can titrate. So here this requires individualization. So I'm telling the patient, we're going to go up slowly, see where you go. If you have headache, I'm giving you paracetamol with codeine. If you have nausea, I'm going to give you a powerful antiemetic. If you have diarrhea, we're going to treat that. Now, if we reach a certain threshold, we stop, we wait for several weeks even, and then we keep going up. The value of these therapies is in the fact that it has an impact on the vascular resistance, and as we saw, remodeling, the cellular vascular remodeling. So we have to hold the patient's hand. This is a really serious problem, and I know that 100% of the patients have side effects. The other thing that I found useful is to connect patients to each other. A patient who's successfully been through the pathway connects with a patient who's starting, and that's very reassuring. They tell them, not me, that this will get better and you will now get an improvement in your overall functional capacity. And I have one of my elderly patients who's very intelligent, and she said, how does this medicine increase my oxygen, doctor? Because she noticed on her 6-minute walk that her oxygen is improving. And I told her it's not an oxygen pill, but it actually improves your heart function. So I think these motivated patients help the patients who are starting. Your point is excellent because I think we can't really say come to a dead stop. Now in terms of switching, that's a more complex decision making. Most of the symptoms that you get from PDE5 are headache, right? So switching to another pathway is still going to cause headache. So I'm a little cautious doing that because I know I'm replacing one side effect for the other. There may be some advantage of, and this is just clinical observation, of riociguat over high-dose tadalafil in terms of headache. But again, all of these patients, I tell them, here's a pack of medications for side effects, here's the nurse, here's the phone number. Don't stop the medicine. That's the key point, because I think once they stop, they become very demotivated and it's very difficult to catch them on something else.
One more question. There is some practice where they say PAH-specific therapy are big guns and we should use them wisely and we shouldn't use all our guns at the time. Although there's a very good evidence that early starting and early escalation of therapy is associated with better with less mortality. How do you answer that? And do we really need to do a GCC patient support group? Because this is where we need to start something, because patient support group just help the patient in assessing which physician who's saying the right things and the wrong things.
So the second question is very easy. I agree, 100% yes. Patient support groups are essential. You know, the PH Foundation in the US, the PVRI, they have a very powerful patient lobbying organization. And not only helps the patients overcome these obstacles, but also overcome some social and access problems, which we have plenty in the region. So I think that's really essential, and I think a big group with some motivated patients is very important. In terms of your first question, see, why don't we use triple therapy right from the beginning? So we have the Triton study. Some patients, as you saw from the survival curves, will do fine on dual combination therapy. You just don't know that until the first visit, right? And until the first visit with very specific risk assessment tools. That's how you tease out the patients who will respond and the patients who don't respond. I think that's really the key point of that question. Why is triple combination therapy essential in high-risk patients? So from Professor Sidbon's study from 2013, using IV triple from the beginning, 2013, in high-risk patients with very severe PVR, very poor hemodynamics, gave them 100% survival. So those are two different patients, right? This is the ICU guy who gets referred to you at 3 in the morning. This is the patient who comes to you in the clinic, right? And I think those are different patients because of the progression of their disease untreated versus the patients we should be getting early, and we can use that. So my argument is for early initiation, not upfront, but early means 1 to 3 months.
Yes. One more question. The patient is like on a lower risk group. We aimed for green, but this patient is on like furosemide of 160.
I'm sorry, say again?
The patient is on furosemide of 160. He's on high doses of diuresis. High doses of diuresis, or the patient is having desatting, or the patient is at extreme of age. He's just 20, but he's just walking for like 450 or just 500 meters where he should walk more. Do we have now certain triggers that trigger escalation of treatment in patients who are in green zone? And if they are in green zone, how do I reclassify them if these things happen? Like, do I reclassify them to intermediate low, or do I reclassify them to intermediate high? If someone who's in green but he's taking high doses of diuresis, where do I place him now?
So I think this concept of how do you get to the green is very important. If you still require large amounts of diuretics, that means, as Professor Anton mentioned, there is right atrial remodeling and there's diastolic dysfunction, right? There you still got a volume load even though your pulmonary vascular resistance is improving. So those patients are a little bit suspicious. I think that patient, if I'm not happy with these clinical characteristics, a cardiac MRI is going to be very helpful. Alternatively, doing a repeat right heart cath, I think, is reasonable too, because you need to get the hemodynamics. I mean, I think the complete intention of the guideline, putting in the 4 strata, was to trigger people who don't do risk assessment to do risk assessment, right? And the idea here is not just to depend on it and say yes or no, but to move forward. So this is unlike, if you would permit this analogy, of the CHADS-VAS score. The CHADS-VAS score is quite concrete, give or don't give, right? Here it's not. It has a lot of gray zone and nuances, but when it's looked at in most PH clinics that they don't do risk assessment for whatever reason, you need at least a clinical score to trigger a test and trigger that mindset. The lower risk right green patient, I'm not really sure, but I don't have a lot of those. I'll be honest with you, I don't have a lot of those. Most of the patients we have are such late referrals that we're struggling to keep them in the yellow zone to begin with. So, but there again, the role of cardiac MRI and right heart cath is very clear, because that may be, as we saw, you can get worsening of the pulmonary artery, the SVR. You can get worsening of the pulmonary artery systolic index. The pulmonary artery compliance can be measured more accurately than the clinical criteria. I mean, the right heart cath is still the king. Right?
And do you have a suggestion on where to classify them? Like, do we take them just one notch up to intermediate?
Yeah, so I would use then the typical 3-component criteria. Yeah. So once I have a right heart cath, I have a more granular piece of information to see where they stand, right? The right heart cath, again, is the king. We don't do it every month. We can't do it frequently. You have to use surrogates, but if I have a right heart cath that falls me into the intermediate risk somehow, And you know that increase of diuretics can have a false impact on the BNP. That's the problem. You know, on
Exactly.
diuretics or SGLT2 inhibitors, the BNP can come down falsely. And that may not reflect the pulmonary vascular resistance, pulmonary artery compliance, or the more important factors like the RV stroke work index. All right, thank you. Any other questions? I don't want to keep everybody from prayers and lunch. You don't want to go to hell or something else like that. Okay, thank you very much.
check, check, low, low, test, test, check, 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 low, check, check, low, test, test, check, low, low, test 1, 2, low, check, 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 1, 2, low, test, 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 low, check, check, low, check, 1, 2, low, check.
السلام عليكم ورحمة
Hypertension is a specialized group of elite physicians in Saudi Arabia. Welcome to
On behalf of my colleagues and SAPH members, I welcome you to the 13th annual meeting.
Welcome to our 11th SAPH meeting. SAPH is the Saudi Association for Pulmonary Hypertension. It's a specialized group of elite physicians in Saudi Arabia and including some of the Gulf region.
Saudi Association for Pulmonary Hypertension meeting, SAPH 2018, that was just held here in Dubai in April 2018. It's It's been actually a great meeting. I've had a chance to interact with some of the leaders in the field.
Every year this Congress is providing the best possible knowledge and evidence for the different treatments in any area, including drugs, including interventional procedures, and including also surgical procedures.
Prevalence of DAH and connective tissue disease, diagnosis, prognostic, and risk factor differences between idiopathic and connective tissue disease rating.
As I'm part of the SAPH family for many years now, I like to come to this conference year by year. The organization, as usual, is perfect, and the quality of the scientific program is outstanding.
We have more than 500 patients included in this different trial, meaning a lot of small trials, and we have the AIR study that has nicely we showed in the previous meeting.
We're grouping many people from different countries, and from Europe, from the US, and from Saudi Arabia, and Dubai, and many other countries.
Many colleagues that really worked very well. From the international speaker, I really need to say that the PBRI had a great role in SAPH success, so I'd like to have a basically special thanks.
Where you can meet all the experts in the field, get all the updates, and get to know your colleagues and mingle with them. SAPH is an amazing team, which we are really proud of, and I hope it will continue for years and years to come.
ارتفاع ضغط الدم داخل شرايين الرئة وليس الضغط العام. وبما أنه يرتفع ضغط الدم داخل شرايين الرئة والقلب تدفق الدم من الجهة اليمنى من القلب يدخل الرئة. إذا كان هناك ضغط عالي في شرايين الرئة يأثر على عضلات القلب بالذات الجهة اليمنى مع الوقت.
نعم ممتاز. طيب هل هو يعتبر مثل ما ذكرت في البداية أنه هل هو مرض نادر وهل تشخيصه سهل؟ وبرضه يا دكتور يعني كيف تشوف طريقة علاجه؟
هو طبعا نادر لكن له أسباب كثيرة، بعضها شائع وبعضها نادر، وهو يصنف إلى عدة مجاميع.
نعم.
هناك ما تسمى المجموعة الأولى وهذه هي اللي نادرة، وعادة يعني بالطروحات والنشرات العلمية
نعم.
تقريبا بين 50 إلى 100 كل مليون يصابون في هذا المرض. وهو طبعا بحكم أنه نادر وبالتالي يكون تشخيصه في العادة صعب شوية. وعادة التشخيص يعني يتولى عملية فرز أسباب المرض، لأنه هو صحيح المحصلة هو ارتفاع ضغط الشريان الرئوي، المحصلة النهائية، ولكن لها عدة أسباب. فدقة التشخيص تكمن في فرز هذه الأسباب، وبالتالي وصول التشخيص الدقيق، وبالتالي أيضا العلاج الدقيق، لأن لكل مرض لكل سبب من هالأسباب علاج.
الاستوديو رئيس الجمعية السعودية لمرض ارتفاع الضغط الشريان الرئوي الدكتور عبد الله الضلعان. اهلا بك دكتور عبد الله، مساء الخير.
اهلا وسهلا.
دعني ابدا معك في البدايه عن تعريف يعني بهذا المرض.
الريبورتاج المقدم لخص المرض لكن ببساطه تعريفه هو ارتفاع الضغط داخل الشرايين الرئويه وليس الضغط العام للجسم. وكما هو معروف ضغط الشرايين الرئوية دائما يكون قليل جدا ليستوعب تدفق الدم من القلب. إذا بدأ ارتفاع الضغط بدأ يأثر على القلب، وبالتالي تتعب الجهة اليمنى من القلب عادة. طبعا له أسباب كثيرة، يعني ما أظن هذا مجالها، إنما هو يعني بشكل أو بآخر يعتبر مرض نادر. تشخيصه يحتاج دقة في التشخيص، وعلاجه أيضا يحتاج دقة لأنه هو أكثر من مرض، أكثر من مسبب، والنتيجة واحدة اللي هو ارتفاع ضغط الشريان الرئوي. ولذلك احنا في الجمعية يعني تبنينا الحملة هذه الجديدة عن المرض لزيادة التوعية. وأنا أظن عندك سؤال آخر عن الجمعية.
نعم، أنواع المرض دكتور.
هو طبعا هو كما قلت النتيجة النهائية هي ارتفاع ضغط الشريان الرئوي، لكن المسببات عديدة. أشهرها وأكثرها عنفا ما نسميه احنا في الطب الغير معروف السبب، ضغط الشريان الرئوي الغير معروف السبب. وانت تسالني عن السبب واقول لك الغير معروف عن السبب.
لان هناك امراض
نعم هذا
او انواع غير معروف.
اي نعم، هو في الواقع الرئيسي.
نعم.
واللي هو شويه عنيف اللي هو الغير معروف السبب. وهناك انواع اخرى طبعا من الاسباب، منها القلبيه، منها امراض الروماتيزم، منها امراض البلهارسيا، منها التجلطات المتكرره على سبيل المثال، وهذه صرنا نشوفها كثير، وهي عادة من الأنواع اللي ممكن علاجها ضغط الشريان الرئوي لأنها تعالج جراحيا، والجراحة هذه بفضل الله متوفرة في المملكة. نعم. فأسبابها كثيرة، إنما النتيجة هي في الأخير هي ارتفاع الضغط في شريان الرئة. المفروض يكون ضغطه واطي عشان القلب لما يتدفق الدم
نعم.
داخل الرئة، تستطيع الرئة أن تستوعب كل تدفق الدم من القلب.
جميل.
نعم.
أبرز المراكز لدينا في المملكة لمعالجة هذا المرض؟
بفضل الله المملكة متقدمة جدا في تشخيص وعلاج المرض. هناك 4 أو 5 مراكز كبيرة. مستشفى الملك فيصل التخصصي على سبيل المثال ربما هو الأكبر والأكمل من ناحية التدخلات الجراحية والعلاجية والتشخيصية، لكن مركز الأمير سلطان لأمراض القلب في المستشفى العسكري، المدينة الطبية، منظومة الحرس الوطني عندهم مركزين، في الغربية عندنا تقريبا مركزين أيضا، في الحرس الوطني في وزارة الصحة وفي تخصصي مدينة جدة. فهناك بفضل الله مراكز في المملكة ويديرها مجموعة من الزملاء متخصصين في هذا المرض وقديرين، والعلاجات بفضل الله. Assalamu alaikum wa rahmatullahi wa barakatuh. On behalf of my colleagues and SAPH members, I welcome you to the 13th annual meeting. Thank you all and enjoy.
لا لا check.
End of a very extensive 3 days. Thank you all and have a safe trip back home. بفضل الله تم اكتشاف عدة أدوية لعلاج هذا المرض، وبفضل الله أيضا هذه الأدوية متوفرة الآن في المراكز الكبيرة في المملكة مثلا وفي مصر. لكن المهم في الموضوع هو الوصول للتشخيص الدقيق في ضغط الشريان الرئوي، لأن هو المحصلة ضغط الشريان الرئوي ارتفاع ضغط الشريان الرئوي في شرايين الرئة، ولكن له عدة. ورحمة الله وبركاته، أهلا وسهلا بكم في المؤتمر الأول لارتفاع ضغط الشريان الرئوي، وهو ثمرة التعاون بين الجمعية السعودية لأمراض ارتفاع ضغط الشريان الرئوي والجمعية المصرية لأمراض الرئة.
يسعدنا ويشرفنا استضافة جمعية الساف، الجمعية السعودية لدراسة أمراض ارتفاع ضغط الشريان الرئوي في القاهرة يومي 20 و21 يناير القادمين.
الرحمن الرحيم. الحقيقة أنا في منتهى السعادة إن إحنا في الجمعية المصرية لأمراض الصدر والتضرن يعني يسعدنا كثيرا إن إحنا يكون معنا هذا اللقاء العلمي والمؤتمر الخاص بارتفاع ضغط الشريان الرئوي.
ارتفاع ضغط الدم داخل شرايين الرئة وليس الضغط العام، وبما أن يرتفع ضغط الدم داخل شرايين الرئة مرضى ضغط الشريان.
Arabia. It's a pleasure to chair this session with my colleague, Dr. Nasser Bousaid from Oman. So to resume our program, since we started from the basic concept of pulmonary hypertension and then moved to the definition, classification, in addition of the approach or diagnosis, now this is the time to have a session for risk stratification and management. And it's a pleasure to my colleague to introduce the first speaker.
Thank you very much, Badr. And it's my great pleasure to introduce the first speaker, Professor Athénée Bockley. She's from France. She's going to talk about risk stratification evidence-based update.
Good morning, everyone. Sorry, but I don't have anything to change the slide. Can I have the pointer? Thanks. So, first of all, I would like to thank the scientific committee for inviting me today. It's a great pleasure for me to be here and to talk about risk stratification. Here are my disclosures. So, I was lucky enough to be part of the task force on risk stratification at the last World Symposium on pulmonary hypertension. So what I'm going to explain now is our thought and our conclusion of the task force. You probably all know that for almost 10 years now, risk assessment has played a central role in risk stratification in the treatment algorithm of pulmonary arterial hypertension. Before 2015, the treatment algorithm was mainly based on the functional class. The severity was assessed by the functional class. And in 2015, the concept of risk assessment and multiparametric risk assessment emerged. And since then, we all use the multiparametric risk assessment to evaluate the severity of PAH. So in 2015, you can see on the left side of the slide, in the guidelines published in 2015, that both risk status and functional class coexisted in the treatment algorithm. But in 2022, you can see the new guidelines, and you can see that the treatment algorithm is based on a risk assessment at both baseline and follow-up to guide treatment strategy. If we take a closer look at the treatment algorithm proposed in the last guidelines published in 2022, you can see that risk stratification is recommended only in PAH patients without cardiopulmonary comorbidities, which are defined as either an obesity, a diabetes, systemic hypertension, coronary heart disease, or mild parenchyma disease. And in those patients without cardiopulmonary comorbidities, we have to assess the risk of mortality at time of diagnosis using a three-strata model. So this model is based on this famous risk stratification table, and you can see in the first column there is a list of variables that may provide prognostic information. And for each variable, we will classify them according to thresholds established by PAH experts. It's very important to understand that the minimal set of variables that we have to assess at baseline includes functional class, wall test, BNP or NT-proBNP, but also hemodynamics using right-at-cat. Right-at-cat is mandatory at baseline. And to assess the risk status of a patient, we will assign a score of 1 point for each variable classified in the low-risk zone, a score of 2 for each variable classified in the intermediate risk zone, and a score of 3 for each variable classified at high risk. And then we will calculate an average score rounded to the nearest integer to define the risk status. Using this methodology, we can classify the patient into 3 strata, and it has been shown in the Compera registry, but also in the Swedish registry, that there is a very good granularity between the 3 survival curves. Low-risk patient has very good survival compared to intermediate or high-risk status. But if we remember the treatment algorithm, you can see that we will classify the patient into 3 strata, but only 2 are really necessary at baseline to guide the treatment strategy. High-risk patient versus non-high risk. During follow-up, we will use a simpler tool, which is based on 3 non-invasive variables, which are functional class, wall test, and BNP or NT-proBNP. And using a similar methodology, we will classify the patient into 4 risk status: low risk, intermediate low risk, intermediate high risk, or high risk of death. And using this methodology, we can clearly assess the risk of death. It has been shown in the Compera registry and in the French registry as well. So all these risk tools work to predict the mortality, but the question we had during our task force was, are these risk tools suitable to guide treatment decisions in patients with PAH? We therefore listed the limitations of the current risk stratification tools, and we consider that for functional class, this is a subjective and non-standardized evaluation. Regarding 6-minute walk test, it's not always correlated with PH severity, especially in younger patients in whom there is a risk of underestimation of PH severity. And for the older patients, we know that a short walk distance can be associated or related with other conditions such as comorbidities. Normal BNP values do not rule out the presence of severe PH, so finally for these 3 variables, we know that they may be influenced by other factors such as comorbidities or age. In the US, they mainly use another risk tool, which is the Reveal Light 2 score, which includes 6 variables, the same 3 variables, functional class, qual test, and BNP, but also 3 other variables, which are systolic blood pressure, heart rate, and eGFR. We also listed the limitation of these variables, and we consider that tachycardia may reflect PAH, but may be also influenced by other reasons. This is also true for the systolic blood pressure, and we all know that a younger patient may have a low systolic blood pressure with a very well-controlled pH. And finally, regarding eGFR, it may indicate renal insufficiency due to pH, but also due to another reason. So we consider that all those non-invasive variables correlate with the severity of PAH, but they were not specific with the disease. And we consider that these variables alone are not sufficient to guide the treatment in PAH. So we wanted to add other variables, which of course are prognosis, hopefully, directly reflect the severity of PAH, and also amenable to modifications through therapeutic interventions. So we considered 3 potential tools, which are right-heart cath, echocardiography, and MRI. First, for right-heart cath, we have published recently this study. So this is a study that we performed in the French PH registry. We analyzed more than 1,000 PH patients who have been reassessed within a year of diagnosis with the 3 non-invasive variables, functional class, qual test, and BNP, plus right-heart cath. And we wanted to show the added value of hemodynamics variables on top of the 3 non-invasive variables used in our clinical practice. And we showed that in patients classified at either low risk or high risk of death using the 3 non-invasive variables, in those patients there were no added value of hemodynamics variables to improve risk stratification. But in between, in patients classified at either intermediate low or intermediate high risk of death, we identified 2 important prognosis variables, which were stroke volume index and mixed venous oxygen saturation, both measured during right-heart cath. And using these 2 variables, we are able to better stratify the patient at intermediate low or intermediate high risk of death. Regarding echo, this is also an important question, of course, and a lot of papers have been published showing that echo is a good method for risk stratification. This paper, published by Stéphane Guillot a couple of years ago, classified the patient into 3 risk status. The low risk status was defined as a preserved TAPC and non-significant tricuspid regurgitation. High-risk status was defined by impaired TAPC and dilated inferior vena cava, and in between, the patients were classified in intermediate risk. Using this method, you can classify the patient into the 3 risk status, and you can see on the Kaplan-Meier survival curves that there is a very good granularity between the 3 curves corresponding to the 3 risk status. But the question we had is, what is the added value of echo on top of the 3 non-invasive variables, which are functional class, qual test, and BNP? This is a question that Charles Fauvel from France tried to answer, and in this study, he assessed the outcomes of patients according to the number of low-risk variables using 3 variables: functional class, NT-proBNP, and the ratio TAPC on SPAP. And he showed that this method is a possible alternative to the model based on functional class, qual test, and BNP. So this is very convenient, especially in older patients who are not able to walk, but you can see on the survival curves that there is a lack of discrimination between the 2 first curves, which are superimposed. And finally, he performed a sub-analysis of patients who had the 3 low-risk criteria, the 3 usual low-risk criteria, I mean functional class 1, 2, normal BNP, and normal wall distance. And in this subset of patients, there was no added value of the type C on SPAP ratio to discriminate the patient. Other studies showed that ECHO improved risk stratification of intermediate risk patients at follow-up, but again, it has never been studied on top of the 4-strata risk score. In both studies, they use the older definition of risk status using a 3-strata model. And recently, last year, the Reveal ECHO score has been published. This score is based on 4 variables, which are the tricuspid regurgitation severity, the RV dilation, the RV function, and the pericardial effusion. You can see an excellent granularity between the 4 survival curves, but again, we still don't know what is the added value on top of the functional class qual test and BNP to stratify the patient. Regarding MRI, we all know that this is an interesting tool, especially to assess the outcomes of the patient. And we can use a right ventricular and systolic volume index, which is very useful. The higher this index, the higher the mortality. And we know that MRI improves Reveal 2.0 and the non-invasive French score. But again, it has not been studied on top of the 4-strata risk status. So finally, in our task force, we considered that the ultimate goal for a patient with PH would be to achieve the cure with a complete remission without the necessary of drugs. But this is probably a bit optimistic. So a more attainable treatment objective will be to achieve the partial or complete reversal of pulmonary vascular remodeling, which can be reflected by either right heart cath or right heart function. So finally, regarding the treatment goals in PH, we consider that we always have to assess the big 3 variables, non-invasive, functional class, wall test, and BNP plus NT-proBNP. And right heart cath is also very important at follow-up, especially in patients neither at low risk nor at high risk of death. Regarding ECHO and MRI, we considered in our task force that there was uncertain added value of these 2 tools, so we need for research prioritization to have a new result and a new conclusion regarding this. So to conclude this presentation, it's very important to assess the severity and the risk of death of patients using a multi-parametric risk assessment. The treatment goal is to achieve the low risk status, irrespective of the risk tool used. We can use either the model used in Europe or in the US. But we always have to consider the 3 variables, functional class, wall test, BNP, plus right heart cath in non-low risk patients. And so far, there is a less well-defined role of ECHO and MRI as a treatment goal. So thank you for your attention.
Thank you so much, Athenas. You can join us here. Before I introduce the second speaker, just would like to announce that we have a parallel session for the 6-minute walk test. So anyone, RT or technician or others would like to join, he's most welcome. So the next talk is about the new and old treatment pathways, a sum up of current treatment evidence, and going to be presented by Oliver Sipon. Yeah, Oliver.
Yes. Thank you very much for your kind introduction, and now we will discuss the different pathways that are utilized to treat pulmonary arterial hypertension, and I would like to summarize this in the forthcoming 20 minutes. So it's my disclosures. So let's start with this. You all know this very famous picture of the tree
dysfunctional pathway of endothelial dysfunction, the endothelin pathway, the nitric oxide pathway, and the procyclin pathway. You know that in PAH, there is an imbalance between the vasodilators and the substance that inhibit the vasodilation, that are vasoconstrictors, and also the anti-proliferative and pro-proliferative mediators. The typical example is in these three pathways, so we have an excess of endothelin, we don't have enough nitric oxide and procyclin that contribute to this imbalance. So we can target these different pathways by giving procyclin, by manipulating the nitric oxide pathway, or by blocking the endothelin pathway, or by blocking the endothelin receptors. You know that. And we know that for a long, long time. In the first treatment algorithm, the first guidelines, it was the guideline from ESC. ERS was not in at that time. And as you can see here, we have the endothelin receptor antagonists, the procyclin analogs, and the PDE5 inhibitors. We already had these three pathways as early as in 2004 during these first guidelines. And over the last 3 decades, a lot of drugs have been developed. And as you can see, we have now 3 endothelin receptor antagonists. We have two PD5 inhibitors plus riociguat, a soluble guanylate cyclase stimulator. And we have some procyclin analogs, and more recently, the selexipag, which is a procyclin receptor agonist using by oral administration. And if you look at the algorithm, the last algorithm from the last guidelines in 2022, we found exactly the same class of drugs. We found the endothelin receptor antagonist, we have the PDE5 inhibitors, the soluble guanylate cyclase stimulator, and the procyclin and procyclin receptor agonist. So between 2004 and 2022, the drugs remained about the same. And let's go back now by the beginning of the story. So why these 3 pathways were and are still very, very important? They came from at least 2 Nobel Prizes, the Nobel Prize in Physiology or Medicine in 1982, received by the 3 scientists, including John Vane, and for the discovery of the prostaglandins and all the work they did on the prostaglandin. Then we had the Nobel Prize in Physiology or Medicine in 1998, and 3 scientists for the discoveries on the nitric oxide and signaling molecules. And also, during the same time, we had the discovery of the endothelin. It was not a Nobel Prize, but it was a very, very, very important discovery during the 1980s. And those drugs have been tested in randomized controlled trials in treatment-naive patients, because at that time we didn't have background therapy in those patients. It was true for epoprostenol, the first trial published to date, then with bosentan, which was the first endothelin receptor antagonist, and sildenafil in 2005, which has been published by Nazareno-Gallet in the New England Journal of Medicine. And after that, the main pivotal studies using endothelin receptor antagonists, PDE5 inhibitors, and soluble guanylate cyclase stimulator used the six-minute walk distance as a primary endpoint until 2013. You have here the result of BRIS-1 with bosentan, with SUPER-1 with sildenafil, first with tadalafil, ARIS with ambrisentan, and more recently the data from the patent study with riociguat. As you can see, it works. It works short-term, and it improves symptoms. They improve symptoms, and they improve exercise capacity as measured by the 6-minute walk distance assessed after 12 to 16 weeks. Then, all the trials conducted since 2013, they change the primary endpoint. Why we changed the primary endpoint? Because we wanted to see what happened on the long-term outcome, not only on what happened at the short term, just an increase in walk test, but also what happened on the long term. And that Seraphine, published by Thomas here, and Seraphine was the first study using time to clinical worsening or morbid mortality event as a primary endpoint. And Seraphine clearly showed that that macitentan, was better than a placebo to delay clinical worsening in patients with pulmonary arterial hypertension. Of course, all those studies need much more patients to demonstrate something as compared to short-term trial using 6-minute walk distance. It was true for Griffin with selexipag. It was also true for Freedom EV for oral treprostinil. Compass 2 was not a positive study, and also it was used for an initial therapy, and in the Ambition trial, we were able to demonstrate that initial combination therapy was superior to monotherapy using either an endothelin receptor antagonist or a PDE5 inhibitor. So we can demonstrate that we improve outcome with this, but there is still room for improvement. What are the new developments to target what I called also the big three, the different big three that Athena discussed previously? The big three for me that the three pathway of endothelial dysfunction, endothelin, nitric oxide, and prostacyclin pathway. Now we have high dose of macitentan. The study is ongoing. We also have the combination of macitentan and tadalafil. We have also the drugs administered with inhaled route. It's true for PDE5 inhibitors, also for soluble guanylate cyclase stimulator, and for some others, prostacyclin analog given by inhaled route. And we also have some development on ralinepag, which is another non-prostanoid IP receptor agonist. Just some example. This is the data published last year. And the ADU trial, it was a multi-center randomized controlled double-blind trial. It was a phase 3 trial comparing initial monotherapy with some of those patients were initially treatment naive, and some other were already treated, but some received macitentan compared to the tadalafil monotherapy as compared to double combination therapy. Very, very similar group that was done in the Ambition trial. But here the primary endpoint was the change in pulmonary vascular resistance. It was not a morbid mortality trial. And as you can see, we observe a much more hemodynamic improvement, much better hemodynamic improvement with the combination of macitentan and tadalafil as compared to monotherapy with either macitentan or tadalafil. Another trial, it has been also shown yesterday, the Insignia PH. Insignia was the phase 2 study of MK5475, which is an inhaled soluble guanylate cyclase stimulator given by inhalation. And the study was a phase 2 study comparing 3 doses of this component with a placebo. And interestingly, this drug can be used in combination with PDE5 inhibitor, because you know that it's not possible to combine riociguat, oral riociguat, with oral PDE5 inhibitors because there was too much side effects and when you use these two drugs together. And in that study, it has been shown that we observed a nice improvement in PVR and also nice improvement in PVR, in particular with the two higher dose of this component, and it was well tolerated. And interestingly, the vast majority of those patients were on PDE5 inhibitors. Unfortunately, the company decided not to continue the development, not of the drug, but the development of the drug in PAH, and they wanted to focus just on PAH associated with COPD. A third drug I would like to discuss is ralinepag. The ralinepag, we just have the result of the phase 2 study published more than 5 years ago, showing that an improvement in PVR and marked decrease in NT-proBNP using ralinepag in patients with PAH. We have 2 ongoing phase 3 studies, but there is no news of the, in particular, the advanced outcome. It is 900 patients to enroll, and to my knowledge, the study is still ongoing. Let's move now to the other pathways. We know that beyond endothelial dysfunction, there are a lot of drivers of PAH pathophysiology. It's true for vasoactive mediators, so for genetics, some growth factors like PDGF, inflammation, metabolism, neurohumoral mechanism, oxidative stress. So we have a lot of drivers of and a lot of mechanism involved in the pathophysiology of pulmonary arterial hypertension. And there is no shortage of drug targets. So we have a long list here of mechanism, of potential targets, and a long list of novel or repurposed drugs. And unfortunately, many trials were negative. We have a lot of many disappointing trials using a lot of those drugs. This is a long list of what has been published or not, but as you can see, only 1/2 of the trials have been published. Some other, we don't have any data or just a press release, and that's it. So as you can see here, we have a lot of drugs with either negative results or neutral results, no effect, no difference as compared to placebo, and at least one trial, rodatrista, which has a negative effect on pulmonary vascular resistance. Just to illustrate this, 3 examples: ASK1 inhibitor to target oxidative stress in PAH. We know that PAH patients, they have elevated levels of circulating stress, oxidative stress biomarkers, and we know that ASK1 it's activated by the oxidative stress and may play an active role in pulmonary arterial hypertension. So selonsertib is a first-in-class small molecule. It is an ASK1 inhibitor, and it has been tested in a large trial, in a large phase 2 trial, ARROW, and as you can see, we did not observe any change in pulmonary vascular resistance, irrespective of the dose of selonsertib used, 2 milligrams, 6 milligrams, or 18 milligrams. We do not have any difference compared to placebo. Another example, recent trial published by Steve Cowett in the Blue Journal last year. It is using the anastrozole, and the anastrozole is an inhibit the aromatase, and is an anti-estrogen drug. We know that if you block the estrogen production, you can improve the lesions observed in animal models of pulmonary hypertension. So those drugs has been tested in a phase 2 trial, and as you can see here, the primary endpoint was not PVR, it was the 6-minute walk distance. We did not observe any difference as compared to placebo. It was true for the 6-minute walk. It was true for the change in NT-proBNP. And this, despite a decrease in estradiol or in estrone, which are the estrogen metabolites. And as you can see also, we don't have any change in the outcome. And last but not least, serotonin. We know that serotonin plays a major role in the pathophysiology of PAH, and we have a lot of experimental models showing that serotonin is very, very important in this pathophysiology. And that's why rodatristat, which is a TPH, tryptophan hydroxylase inhibitors, has been tested in a trial, in a large phase 2 trial named Elevate-2. So if you inhibit the TPH, if you inhibit the tryptophan hydroxylase, you can decrease the peripheral synthesis of serotonin. And as you can see in that trial, of course, the rodatristat decreased the peripheral serotonin concentration, but the price to pay for that was an increase in PVR, which was totally not awaited. So this study is the first, to my knowledge, who have a negative result, not a neutral result. It's a negative. It is significant. It is significant. Placebo did much better than the active drug. So, be more optimistic now. At least there are two promising therapeutic pathways. First, genetics, and second, growth factors. Genetics, BMPR2 activin, that's very important. We will discuss in the forthcoming minutes, and the growth factors, the PDGF. And these two pathways has been shown to play a pivotal role in the pathogenesis of PAH. Let's start with the BMPR2 and the TGF-beta receptor superfamily signaling. We know that in normal subject there is a vascular homeostasis with a balance between the anti-proliferative pathway and the pro-proliferative pathway. The anti-proliferative pathway is mediated by BMPs and the BMPR2, and the anti-proliferative pathway is mediated by the activin, the GDF, and the receptors. To be very simple, this is the normal vascular homeostasis. In PAH, there is an imbalance in favor of activin and its receptors as compared to the BMPR2 and the BMP and BMPR2 pathway. The typical example is the patient having BMPR2 mutation. We have this kind of imbalance, but not only in patients with BMPR2 mutation, we also know that patients with PAH without a mutation, they also have this kind of imbalance. And sotatercept was developed as an activin signaling inhibitor that is a ligand trap. They trap the ligand of the activin receptor, mainly the activin and the GDF, in order to try to rebalance between between the pro-proliferative and anti-proliferative balance. And you know that sotatercept has been tested first in a phase 2 trial, the PULSAR trial, which was a 24-week phase 2 study showing a nice improvement in pulmonary vascular resistance by decreased by about 3 Wood units in average. In patients heavily treated, as you can see, the majority of those patients, they were on triple combination therapy, including, for some of them, parenteral prostacyclin. Then the results have been confirmed in the phase 3 trial with STELLAR, including 323 PAH patients, also on heavy background therapy, mainly on triple combination therapy. And despite this heavy background therapy, the patients were able to improve their 6-minute walk distance, and the change to 6-minute walk distance as compared to placebo was about 40 meters. This was, in terms of hemodynamics, this was mainly due to a change in mean pulmonary artery pressure, increase in PA compliance, which is calculated using the PAP, also improvement in RV function as measured by the TAPSI, and also the change in NT-proBNP. But interestingly, there was no change in cardiac output or cardiac index in that study. And regarding the adverse event, we know that the drug is able to increase hemoglobin, to induce thrombocytopenia, but also the drug may induce some bleedings, in particular epistaxis and gingival bleeding, and also some telangiectasia in about 14% of the patients. We observed that in the phase 2 trial. Now I would like to move to the last pathway. I would like to discuss that the PDGF and PDGF receptor, we know that there is an aberrant PDGF signaling in PAH, and this drives pulmonary vascular remodeling and could be reversed by tyrosine kinase inhibitors. And we know that in experimental models, imatinib, for example, is able to reverse PA remodeling. Imatinib was first tested in pulmonary arterial hypertension, first in the phase 2 trial, showing a nice improvement in pulmonary vascular resistance. And the phase 3 trial, using 6-minute walk distance as an endpoint, showed a positive result in terms of improvement in 6-minute walk distance, but 1/3 of those patients discontinued during the time of the study, 24 weeks, due to serious adverse events. So the drug, the development of the drug was not continued in PAH. Of course, the drug is used in other indications. And that's why there is some attempt to use imatinib at a much lower dose, but using an inhalation. And this is the data from the IMPACT study. And the IMPACT study, which is a phase 2 trial, showed disappointing results with inhaled imatinib because we did not observe any significant change in PVR, which was the primary endpoint of the trial. And let's move now to a more optimistic approach. PDGF inhibition with seralutinib, which seralutinib is another tyrosine kinase inhibitor acting on PDGF receptors, CSF1 receptor, c-KIT, and also having interaction with BMPR2. So the drug was tested by inhalation in a phase 2 trial, the TORREY trial, and the primary endpoint was met with the primary endpoint with a change in pulmonary vascular resistance. We observed a significant reduction in PVR as compared to placebo, and this was more pronounced in patients, the more severe patient, the patient in functional class 3 or with a high REVEAL score. I will discuss this more in detail tomorrow, so I don't want to spend too much time on seralutinib. So this is the picture. This is a rapidly evolving pipeline paving the way of precision medicine, and we hope that in the future we will be able to know which patient may respond to this drug or to another drug using some multi-omics approach. And this is my conclusion about the new and old treatment pathway in PAH. What is for me the future of PAH therapy? First of all, using something old, the big 3 pathway, using something new, the TGF-beta and PGF-beta pathway, using something borrowed, like the aggressive strategy, we borrow that to the oncology or cardiology field, and finally something blue. Blue is the new green. I think the American guys and the people from UK, Ireland, Canada, they will understand this. So thank you very much.
Thank you for joining us. It's my great pleasure now to introduce next speaker. Sean Gain is going to talk about a walkthrough update treatment algorithm.
everyone. Thanks. Lovely to be here, and thank you very much, particularly for this. Professor Adlan and Hassan for getting us here. It's always a great pleasure and an honor. Well, you've heard already today a bit about risk and old and new treatments and something borrowed and something blue. And what I'm going to do is give you a little walk, if I might, down through the treatment algorithm and a little bit about how we got there and how we might use these drugs that we now have available. But before I do, I thought I might bring you back a little bit. And I don't know if there are many of you in the audience who were at the first World Symposium of the Modern Era back in 1998. And I can tell you that I was, as a young investigator in pulmonary hypertension. And it was a remarkable event in Evian in France. And I thought what was particularly fascinating about it was the amount of innovation that was going on, the amount of discussion, the collaboration, and a bit like what we've got going on here, where you meet people from around the world, you talk about pH, you share experiences, and then you get new ideas about how you might go about treating patients. There's a lot of courage involved in these meetings as well, I think. As people come along with ideas, they bring it to their peers. And I remember particularly well in 1998, Professor Gérald Simonot from Paris, and he was challenging the primary and secondary classification that was there at the time. You can see it was the World Symposium in Primary Pulmonary Hypertension. And it was a bit of a shocker to hear that maybe we shouldn't have primary and secondary anymore. Maybe we should change it to something that was a little bit more inclusive and something that might be more treatment-based. So that was then back, and I also think the World Symposiums and indeed meetings like this, they're full of hope too, because you hear about what's coming. You also have an idea about what might be happening in 5 years' time. What will it be like in 5 years' time for the next World Symposium, or indeed the next SAPH? What new things will happen in the meantime? So it's 26 years since that first meeting. And so I was delighted to be on the task force for the treatment algorithm for PAH in 2024. And I'll introduce you, know many of these people already, but we had Kelly Chin was really the driver behind this from Southwestern in Dallas in the USA. Very quietly ran things, got people to do the work that needed to be done and reviewing the literature and coming up with ideas and writing sections. Christian Gerges was from Austria, and he was in as a young investigator, but he had the head of a man a lot older than his age. We have Professor Jing, who you see, who's here at the meeting and will be speaking again tomorrow. Siva Matei from Johns Hopkins, who actually took over from me at Hopkins after I left the US a number of years ago. Yuichi Tamura from Tokyo in Japan. And then we had the two chair people who were really keeping us on target, Valerie McLaughlin for Michigan, and of course, who you just heard speak from there, Olivier Sipon from Paris. And they were very much keeping us on track to make sure that we were answering the questions that needed to be answered in a timely fashion for the World Symposium. And you have to put it a little bit in context at the time. So coming up to it, where were we is the first question we had to ask. And what we knew is we had a lot of medications for pulmonary hypertension, and they had an impact on the disease. We know that the medications we have can improve functional class, exercise capacity, hemodynamics, and right heart function. We also knew that combination therapy worked better than monotherapy in most circumstances. And we also knew that we were having an impact on all-cause mortality, but a fairly modest improvement when it came to median survival times. We're still under 10 years. So we were very much aware that we still had a lot to do. The pulmonary hypertension had made great advances. We had a lot of ideas, but we still had a lot to do. And then, of course, the World Symposium this time around, and you can even see it from the presentations you've heard already, and indeed Olivier Sitban's talk about drugs that we've just heard about, that the fourth pathway was the exciting new thing that had come along, and we had to see where would you put that with the old medications that were already there. Well, in fact, we came up at the time with a figure to try and explain what was going on. Here's our three pathways that we know well that Olivier has already alluded to, and then we were able to, with some pomp and circumstance, add in the fourth pathway. And again, as Olivier mentioned, this pathway, the activin pathway, was an effort to rebalance this pro-proliferative picture that we see in pulmonary hypertension, to move away a little bit from this vasodilation story towards something that was anti-proliferative. And that was really the setting that the World Symposium took place in. So we had really sort of three tasks, and I'm going to walk you through what we did in those three tasks. The first was to look at the data that had emerged since the previous World Symposium. First of all, that was our first, since the 2018 World Symposium. The second thing was to kind of create an algorithm that was workable, and finally to have a look at where the knowledge gaps were and look at a few specific groups, and we'll come to that at the end. So let's have a look first at what was available to us, and in fact, since the previous World Symposium in 2018, there have been 10 clinical trials of value that had results that really needed to be looked at. And those clinical trials that I've listed here, which is to say there were trials that looked at initial therapy. So for example, Triton looked at initial upfront therapy. There were trials that looked at sequential combination. So for example, Stellar, looking at sotatercept, was sequentially added to patients already on background therapy. There were new endpoints. One of the studies there was the TRACE study looking at selexipag and looking at an endpoint of actigraphy movement, how it impacted on patients' activity levels. There were new pathways, which we heard about with sotatercept, and the Stellar and Pulsar studies, specific subgroups. We actually had studies, the Portico study looking at portopulmonary hypertension, and the Maestro looking at patients with Eisenmenger syndrome. And then finally, there was a mortality non-inferiority trial looking at sildenafil at 5 milligrams and at 80 milligrams, which was a study that reported before. So we had to look through all of these, and as you can see, some of them, in the green there, they met the primary endpoint, and there were others that did not meet the primary endpoint, were really not of as much value to us as we decided, where would you put these things into that algorithm? Just to remind ourselves of kind of the big studies that we had to look at and see what they told us about how the algorithm should be rebuilt. One was Triton, and as you know, Triton looking at macitentan and tadalafil versus macitentan, tadalafil, and selexipag up front. It was a negative study because the primary endpoint was pulmonary vascular resistance, and there was no difference between those two approaches. Also, we looked at secondary endpoints of 6-minute walk and NT-proBNP. There was no difference between the two approaches. There was a robust clinical improvement, but selexipag didn't add to that. There was a look at longer-term clinical worsening, and there was a hint there might be something, but it was an exploratory endpoint and didn't reach statistical significance. And in many ways, you might think it was kind of a lost opportunity to do that study, just looking at pulmonary vascular resistance. And perhaps if we'd looked at time to clinical worsening, or we'd looked at a more morbidity, mortality, we might have seen something different, but that wasn't the question that was asked. The second study that we looked at that had come out, and again, a very valuable question, was what's the role of oral treprostinil in pulmonary arterial hypertension? And it was a positive study, oral treprostinil on background monotherapy. As you can see, 100% were background monotherapy. The majority, 71%, were on a PD5 or a soluble guanylate cyclase agonist. About 28% were on an ERA, but they were all on monotherapy. It was a positive study, but we don't use monotherapy up front in patients anymore. So the actual value of this study and the results was diminished by that. And so we had to take that into account, and we couldn't grade this or give it the same weight as you would in something that was on background with double combination therapy. Another study that had come out, and we were all kind of familiar with this now, but it had been published since the previous World Symposium, was Replace, looking at the switching of a PD5 inhibitor with soluble guanylate cyclase agonist. The problem with this study, as we all know, is that it was an open label study, and as a result, again, the results were positive, but because it wasn't structured as a study in the way that we would have liked, the results have to be taken with a caveat, and when you're putting in the algorithm, there needs to be a caveat about how you interpret this data. And as you just heard from Olivier, the other study that we had to look at was this new fourth pathway, very positive study, 323 patients in the study, 61% of them being on background triple combination therapy. You probably wouldn't have imagined that a drug could have an impact on 6-minute walk when they had the disease for quite a long time, were already on triple combination therapy, many on parenteral prostacyclins, and yet there was an improvement. So very impressive, and the secondary endpoints across the board, more or less, had improved PVR, NT-proBNP, function class, and the French risk score. And then the final one, and you've heard about this already today, the final study that we also put into the mix, it was a consortium between the Germans, Austria, and Switzerland to come up with evidence on the vasodilator responders. And it's interesting that this was something that often has got pushed to the side with new drugs and pulmonary hypertension, but calcium channel blocker responsive patients are really important. And we actually, you'll see from the way questions emerged even in the earlier sessions, people are still intrigued by what's the right thing to do in these patients? What we learned from that study was about 10% of patients with idiopathic pulmonary arterial hypertension, heritable or drug and toxin associated, had a positive response rate, and that was great. But actually, their overall survival also was quite good, 86%. But when you looked at them, they divided into long-term responders and ones that were not long-term responders. The 5-year survival in the long-term responders was a remarkable 98.5%, a cure, as you would reckon it. Those who were not long-term responders had a 73% 5-year survival. There were a few markers as well that indicated who might become a long-term responder in terms of CAT predictors in pulmonary vascular resistance improvements and pulmonary artery compliance, and also in some of the more rapid improvement in functional class and 6-minute walk. But a very valuable information that would underpin a change or a new algorithm for calcium channel blocker approach. So that brings us to the new algorithm, or new algorithms, in fact. In fact, the first thing we were able to do was to propose a calcium channel blocker treatment algorithm and modify it a bit, bearing in mind what we had learned from the studies that occurred since the last World Symposium. Now, I just wanted to say one important thing about the algorithms from the World Symposium that we're really keen to get out when we were doing it. It's that there's an algorithm and there's key points, but the key points aren't footnotes. They're actually really important, and because to try and make the algorithm as simple as possible, we kind of stacked a lot of information into the key points, and we were keen that they would always be shown together rather than discard the key points and end up by just the algorithm on its own, and maybe it could be misinterpreted in that way. I'll explain that to you again in a moment, but if you watch here, you can see therapy for group 1 PAH responders. You put them on calcium channel blockers. You follow up risk assessment 3 to 4 months and repeat it frequently. I would say in real life clinical practice, you watch these guys even more carefully than that in that early stage. And then we come down to low risk. You continue the calcium channel blocker alone, but if you ask what is low risk, you go across to E, and it's low risk status. But what's important also in that is that it refers to near normal hemodynamics. It's not just about doing your non-invasive approach. You really have to recap patients, make sure you've got somebody who you now have on a pathway for a 5-year survival of 98%. For the others who are not low risk, they've lost that opportunity to go down the more simplified pathway, and then they join the normal algorithm that we use. So this is the normal algorithm then that we've proposed from that, and you'll be all familiar with the image. Again, you'll see there's quite a comprehensive list of key points that will need to be taken into account. So initial risk assessment, and we've heard from Athenas all the various different approaches that we have to take in that, really important, and the importance of continuing to look at risk stratification. One of the things that comes up if you look at the key points is this algorithm is for patients who have a mean pulmonary artery pressure greater than 20 or equal to 25 and a PVR that is greater than 3, because that's where the data on all of the drugs that we're going to have in the algorithm were studied under. So that's important, even though the definition, as we've heard this morning from David Matani, that it was that the definition has changed. This is what the algorithm speaks to for the time being. When it comes to the sickness of patients, and again we've heard about this just recently, initial risk is very much a binary question. Are you high risk or are you non-high risk? And in that situation, that initial risk assessment, again, close follow-up on patients. But what's most important about the high-risk patients, I would say, is there's no talk here about the fourth pathway sotatercept. It goes straight to what we know and what we have proof of, and that is the patients who are high risk be considered by initial triple combination therapy, including a parenteral prostacyclin, and importantly, they should also be referred to the transplant group so that they are aware of the patient. This is somebody that's really sick. We're putting them on up front, triple combination therapy, but you need to be aware of this patient because if they don't respond, they're heading your direction. The reason why that approach was taken was data that had emerged in 2021, again from Athenas and the group in Paris, looking from their registry. And you can see that if you look at patients with high or intermediate risk and low risk, but look at the first two here, high risk and intermediate risk. The ones in red here with the really good survival are those patients who are triple upfront combination therapy, including a parenteral prostacyclin. So it's not just a story really about high. You really have to look at those patients who are intermediate, they're on the very edge of it, that those are patients that do benefit from it. So the response to a triple approach with parenteral prostacyclin for the 6th of the patients is really robust. And it really goes back to the older drugs that we know best for that stage and the algorithm. Well, what about the next step? After that initial assessment, start them on treatment and send them off. One of the things I like particularly from the new algorithm is there's a new little box here, a little measure, talking about the first follow-up visit. The first follow-up visit, and I've often said this in the past, it's actually the most important visit of a patient who has pulmonary hypertension because it gives you a chance to decide what way that your treatment strategy, when you first saw them, and your prognostication and everything else, were you right? Did your treatment work? And looking at the first follow-up. And indeed, if we have a look for a moment, this is again from the French registry, you can see 2,879 patients, first follow-up using the 4 risk strata. You can see the remarkably good discrimination that you can get from doing that risk stratification at the first follow-up visit. And you have a really good idea about how sick your patient is and what now you need to do for the next step in the algorithm. So going back to the algorithm, what do you do with low risk? Most low-risk patients at follow-up should continue their initial combination therapy. I would say, and we've often talked about this, those low-risk patients you need to look at very, very carefully because you're saying that those are patients that you're not going to do anything with for the time being or maybe for the longer haul. You need to be sure that that's what you have based on the low-risk, the non-invasive criteria, and also using your clinical gestalt, looking at your echo reports, having a look at MR if you use MR. Make sure you're happy that you're in the right place in those patients. For the intermediate low risk, you can see that it has the little E up above, and E brings you over to point out a little bit of detail. So intermediate low risk, add an activin signaling inhibitor, oral or inhaled prostacyclin pathway agonist, and can consider switch. In there, there's a little story, and that really is that you have to be able to use your own clinical gestalt again and how you would approach that. Many people there would say, well, will I use an oral prostacyclin pathway agent? You may. That might be a simpler path in the first circumstance. And again, it'll depend. You may choose to do the risk stratification that we heard from Atinas, get a right heart catheterization, just see where they are when you more thoroughly risk stratify them to decide. But there are options there. And the or, whenever you see this, is usually where we don't have enough data to point you strongly in one direction or another. But over on the side it says clinical trials with oral and inhaled treprostinil only included patients on monotherapy. That's to inform you about what evidence, a level of evidence that you're going to be using to make your decision. And then while studies selected packages that have included patients on background combination therapy. If we look at intermediate high and the high, and they both have a little F beside them, and that's immediately to prompt you to consider transplant referral. Once patients are not doing well in response to initial therapies, that should be considered. The treatment options for intermediate high would include intravenous prostacyclin, or you can consider activin signaling inhibitor if you have it available to you. But really when you're high risk, the guidelines really, or the recommendations are still pushing strongly to consider parenteral prostacyclin as the first choice if they're not already on it. If they're already on it, you can consider a 4th drug. And so for persistent intermediate high or high, 4 drug therapies are available. So that's really, I suppose, how that algorithm has developed over time. And the effort was not to push prostacyclin aside. It was to very much hold on to what we've already got in the drugs we have and consider where will sotatercept sit into that algorithm without losing from what we've already learnt over the years in the past. So a couple of little comments then about specific subgroups and knowledge gaps. And the biggest one, and you've heard about this already today, was where did the comorbidities go in the algorithm? It seems to have disappeared from the ESC/ERS guidelines. And it was really just an effort to simplify them. In the text, there's plenty of discussion about how to approach comorbidities. And again, going back to what Stefan Rosenkrantz had mentioned earlier, when you're at the bedside, you're going to mix the severity of the pH with the severity of the comorbidities. If it's all comorbidities and a small bit of pH, treat the comorbidities. If it's all very severe pH and a small bit of comorbidities, you most likely have a patient with group 1 and you're going to treat their underlying pH. So there's room in the algorithm for people to take clinical gestalt at the bedside and decide how to proceed, and it's laid out in the text. One other area that we discussed in some detail was transitioning, because this is probably going to be fairly important in the future. The earliest information we have is when bosentan was first approved, people tried to get people off intravenous prostacyclin, and that was the idea that maybe oral therapies would be all we needed for PH, and of course that didn't transpire. There really isn't very good long-term data, although sometimes you will consider weaning people off parenteral prostacyclins. For example, you may consider where they're really low risk, and have near normal RV function, or the PVR less than 5. You need frequent monitoring if you choose to go that pathway, because both early and late failures have been well described. And I'd say in general the advice is you don't do it. It's a very difficult thing to do. It very often fails. So it's just to say we did discuss, because in the future with sotatercept it may become something that'll be discussed a lot more frequently. And then just a few words on the knowledge gaps and future directions. It was widely accepted that we needed more real-world data. What's going to happen when sotatercept is out there it's already FDA approved in the US, for example, and it's being widely used? What kind of information are they getting back from real-life studies there? Paucity of clinical trial data is available for the mean pulmonary artery pressure 21 to 24 and the PVR 2 to 3. What do we do with these patients? We need more information on that between now and the next World Symposium. A little comment to some subgroups not being well represented in data that we currently have: portopulmonary, unrepaired congenital, what about schistosomiasis associated with PH, which many of you here would be very familiar with, and PVOD. And then finally, something which is, I think, becoming increasingly important is that optimal treatment in resource-poor parts of the world is really important. These drugs are incredibly expensive, and they're going to get more expensive, and we're going to have a two-tier to the treatment of PH. And we did put a bit of text into the publication on how we might address this in the future. One issue that came up quite a bit was septostomy, for example. People ask questions afterwards about septostomy, because in places where you're resource limited or maybe don't have access to intravenous epoprostenol, septostomy can be a very good option. It can be a good bridging option. It can be an option on its own. And a lot of people thought that should be in there. But we needed more data to be able to put it robustly back into the treatment algorithm. And we can't talk about a treatment algorithm unless we talk about patients. And we did spend some time also talking about just the things that impact upon the decisions that we make with our patients. The patients are all going to be different. Some are going to have comorbidities which impact upon how they can handle the algorithm. Other patient factors, their supports at home. Routes of delivery are obviously a really important aspect of how patients will respond to being prompted to start new therapies. RV function and imaging is an important thing too. Someone can walk 450 meters but be a young woman, and you think, is that okay? And you look at the RV on echo and it just doesn't look right. And you make decisions that are part of your gestalt with each individual patient. So let me round up, if I might. I'll just say so that I talked about the first World Symposium that I had the pleasure of being involved in 26 years ago. And then the more recent World Symposium. It's 26 years, I think, fairly steady progress. And I think it's interesting to imagine what it'll be like in 26 years' time, in the year 2050. What will the World Symposium look like? What will PH look like? What will the world look like? But it's an interesting thought. I think there has been great progress there. It's great that we have an extra pathway to think about and an extra data. But over the next 5 years, hopefully we'll have even more and more to consider. Some conclusions just to say. So optimal treatment therapy, despite everything that's been said, is combination therapy for the majority. Upfront ERA and a PDE5 for newly diagnosed patients who are not high risk. Parenteral prostacyclin remains an important component of the treatment. It hasn't changed. It's still there for the sickest of patients. And the maximum therapy now for many places in the world is slowly emerging, that we have 4 pathways to go after. Reassessment of early escalation is very much recommended. Earlier is better because we realize that we can do more for the right heart. We stop it from failing in the first place. And then the final comment, I suppose, is transplant remains a treatment option for now. And the goal of the future will be that we put transplant to historical use. But for now, we still need it. Thank you.
Thank you so much, Sean, for this nice talk. So let's move to the last but not the least talk in this session by Luke Howard from UK. He will talk about the trials design in PH, current and potential futures endpoints.
Thank you very much. Olivier, I have to admit you lost me on the blue-green thing. I thought it might have been a rugby reference, but then since we beat you last weekend, on that. So I'll have to speak to you afterwards about it. Fill me in. Well, look, thank you very much to the organizers for inviting me back. It's a great pleasure to be here. So these are my disclosures. I also have a second disclosure side, which is that there are a lot of endpoints that I could talk about. But I got this wrong. It's not the last session, it's the penultimate session. I didn't turn the page over. And it's on a Friday. So I'm not going to talk about them all. I'm going to talk about primary endpoints. And I'm sorry for any offense that you may have perceived from me by not mentioning your favorite endpoint. So, spoiler alert, I'm not about to change what I think the recommendation should be for a phase 2 and a phase 3 trial, particularly not with Nazareno Gallier sitting in the front row. So there's going to be no great revelations, I'm afraid. This is what has worked for the last few decades in getting approval for drugs which have significantly impacted the lives of PH patients. So phase 2 trials, typically, PVR and phase 3, we've shuttled now between 6-minute walk and over to morbidity, mortality, and back to 6-minute walk again. So here's just a nice schema of the different phases of clinical trials. And so you can see moving from phase 1 through to phase 2. In phase 2, you're focusing more on efficacy. It's much more of an important factor at that stage because you're trying to work out whether or not you can take your drug through to a phase 3. So the question is, at the bottom, does the drug do what it's supposed to do? And typically, in the drugs we are looking at, does it impact the pulmonary hemodynamics? Does it drop the PVR? So for phase 3, this is your regulatory approval trial. And at this point, you're trying to generate the data to get approval. Is this drug actually making your patients feel better, function better, survive longer? So currently in use have been the 6-minute walk and time to clinical worsening. And as yet, there's no validated surrogate endpoint in PAH, and I'll discuss that in a little bit more detail. I remember at a John Vane symposium about 10 years ago plus, I remember Lou Rubin saying this about a drug in a phase 2. If you cannot show your drug drops PVR, you're done right there. And that's stuck with me ever since. It requires a catheter at baseline and at week 24 typically, and then sometimes a follow-up catheter after that. Typically, phase 2 is going to be about 80 to 100 patients. It's going to have to be multi-center, it's going to have to be regional or probably global. And at the last count, I think it's probably going to cost you something like $50 million US. So I suppose one of the questions when I was thinking about this, and I'm going to spend a little bit of time on phase 2, but mainly phase 3, was, is there a step before going to a full phase 2 that you could potentially employ? Perhaps you could do PVR a little bit better, a little bit more accurately. Could that be done with repeated measures, for example, in the same patient to reduce the noise? Could you use the same patient as their own control with crossover? So you could potentially reduce the sample size and go through to a phase 3, but you're going to be missing out on some quite important secondary endpoint data and some safety data. So imagining that you can do a 20-patient phase 2, then go into a phase 3, I'm not sure I can envisage that. But what you could do is maybe do a quicker phase 2A or a 1B, looking at whether or not you can make a quick go, no-go decision into a formal phase 2. So, for those of you who know me, this slide is now not going to be a surprise, but you could, for example, explore the use of a CardioMEMS implantable pulmonary artery pressure device. So this is licensed in heart failure. It's a small device that sits in the left lower lobe pulmonary artery, and it measures PA pressure. There's no battery in it, so it can sit there forever. And it's powered by firing RF energy at it from a mat that you lie on, which gives it enough power to sense the pressure and send it back to the mat and then up to the cloud. And so you can get a 30-second reading with a high-fidelity pressure trace. They've also developed a cardiac output algorithm as well, so you can get a measure of transpulmonary or total pulmonary resistance. Pleased to say, last week we had this paper accepted from the UK into the Blue Journal, and this is a study of 17 patients re-exploring imatinib. This was sort of two aspects to this study. One was looking at can we, in a group of 13 patients, find the maximum tolerated dose, which we believe to be 200 milligrams, but also can we capture some hemodynamic data. And you can see how if you're measuring pulmonary artery pressure on a daily basis, and you can pick up total pulmonary resistance, you can track the drop in total pulmonary resistance over time. And you can also get interesting data on drug withdrawal, and so you can start to explore things like remodeling effects. So it's quite a powerful tool in a very small number of patients that might give you a go, no go to a bigger study. And here's just an example of an individual patient who was in intermediate high risk, who went on to 300 milligrams in part of the up-titration process, and whose TPR dropped, and then they withdrew the drug after the fixed pre-specified time period. They worsened, and then they reintroduced it. So you can see how you can start to get these interesting sort of granular data on small numbers of patients. And we're using this study in the UK in two trials. One is the Phoenix trial, which has come out of Sheffield, which is looking at this question of what to do at the stage when you are in intermediate low risk at 3-month follow-up. Do you switch or do you add? And so patients are implanted with a CardioMEMS device, and they will either switch or add, then cross over, and we're already getting some really interesting data from that. We're about to start a 12-patient study, a phase 2A study called Vital pH with a ZIP12 monoclonal antibody. And so this will give us a very rapid idea after 12 patients as to whether or not the drug should proceed or whether you should just call it quits after having hopefully spent only a few $1 million, not 50 million. What if you could image PVR? So this is something called a fluid technology. This uses standard non-contrast CT scans and looks at the volume of blood in the 10 millimeter and 5 millimeter blood vessels. And in the remodeling process, or it could be vasoconstriction and remodeling, you see a peripheral pruning and a shift of the blood from the small vessels to the large vessels. And so this was used in the TORREY trial, which is the phase 2 seralutinib study, which Olivier's already alluded to. And you can see an example of a patient who was on seralutinib after 24 weeks. If you look at the upper lobe, you can see there's a greater distribution of blood into the peripheral vessels. And these were data presented by Roham Zamanian at the ERS a couple of years ago. And at that stage, we saw a relationship between the changes on CT and pulmonary hemodynamics. So, what if this gave you an idea of a change in the pulmonary hemodynamics that was akin to PVR, for example, and you could use this in place. Obviously, it's early days. We don't know if you'd be able to shrink the numbers or not, but it's certainly a non-invasive way of dealing with the problem, and it might provide more precision. It's just a thought. So I've already discussed this a little bit. If you reduce the size, you don't collect enough data for secondary endpoints. So there are definitely downsides to this. But say, for example, with a drug like imatinib or another drug you're repurposing where you've got a lot of safety data, you may only need to do a small trial before deciding to go to a bigger phase 3. And so there are pros and cons with this approach. There are other ways, of course, you can measure PVR. You can estimate it from echo or cardiac MR, but I think that's only going to increase your sample size. So it might be non-invasive. So I don't think that is an appropriate measure for does your drug work? Does it do what you think it is supposed to do? Of course, important information, but it's not telling you if your drug is hitting the target. So phase 3. So this is taken from the World Symposium paper that, from one of the other task forces. So an efficacy endpoint in phase 3 must be consistently and readily measurable. So let's think about 6-minute walk and time to clinical worsening when we're going through this list. Sensitive to the effects of interventions, it must be well-defined, reliable, and they must be direct measures of how patients feel, function, or survive, or there must be a properly validated surrogate. And I've already said we don't really have one. And so this was an article published pre-Sotatercept, and so this was looking at the phases of going through. We had from 2001 to 2013, all the trials being proved there on 6-minute walk, and then flipping over in the last decade into outcome trials of morbidity, mortality. But of course, Sotatercept came along and changed things. But this is just another way of looking at the patient numbers on the y-axis and the trial duration on the x-axis. And so you can really see how these larger, longer-term studies take in a lot of patients. And so they're more expensive to run. Patients get trial fatigue, but they do give you some of those more measures that actually patients might find relevant to them in terms of hospitalization and so on. I'll come on to that. So what about change in 6-minute walk as a primary endpoint? Well, on the plus side, it's easy, it's cheap, it's repeatable, it's part of your everyday clinical practice, and patients are used to it, you're used to it. It's currently accepted by the FDA and EMA. It is a measure of function. It's a measure of what you can do. And it also has a relationship with how patients feel, and we think with a roughly a minimally important clinical difference of around 30 meters. And that generates studies typically around 200 to 300. On the downside, it's got a high standard deviation, it's noisy. There might be a ceiling effect. So you might have younger patients who may still benefit from drug in terms of longer-term outcomes, but you're not going to pick that up. And certainly a walking speed may not be relevant to them. It can be affected by other factors, musculoskeletal, mood, so on. And it certainly does not have a strong relationship with survival and morbidity in terms of the delta. So coming on to M&M, or time to clinical worsening, it's a composite endpoint, typically including death, pH-related hospitalization, some marker of clinical worsening, usually a 15% drop in 6-minute walk and something else, such as change in functional class, or rolled into that can be treatment escalation, and finally transplant or septostomy. Now these endpoints, I think you'll agree, if you look down at these things, they're all relevant to patients. If you say to a patient, do you want to deteriorate? No thanks. Do you want to be hospitalized? No thanks. Do you want us to have to add in more drug? No thanks. So I think these are meaningful to the patient. The problem is that some of the really important things happen very infrequently. So if you look at the sort of the weighted proportion of outcomes in terms of what goes into the final endpoint, you can see that little line down there at the bottom in terms of mortality. Look at the line rather than the dots. You can see mortality contributes very little to the combined endpoint. Hospitalization and disease progression seem to be the things that drive the endpoints mainly. So still important things, but perhaps not the most important thing. Then the other thing is that actually does the treatment effect of the drug does that effect that it has on the clinical worsening also translate into mortality? And the answer in this analysis, which is way beyond my understanding, I can't even understand the blue-green reference from Olivier, let alone this paper. So, but the point of these very clever statisticians say is that the treatment effect that it has on the worsening doesn't correlate and translate into the treatment effect on mortality. So in other words, it's not a very good surrogate. So could you do a mortality study? Well, it turns out you can. This is a study published by Marius Hooper. I think it was many, many years in the making, and this came out of an FDA concern of an increased mortality with sildenafil. So they said, go back and look at mortality. And you can see over many, many years, you can actually run a study, and in this case, it showed that that the mortality from higher doses of sildenafil was not inferior, and in fact seemed to be superior to lower doses of sildenafil. So it can be done, but you're getting the feeling that, okay, it's being done, but with a drug that we've had around for many, many years. So it's quite hard to see how you might do that type of study in a completely brand new drug. So you could do mortality studies. You can easily see how you might do it with something like dapagliflozin or spironolactone or anything, anticoagulation. You could put it into a platform trial, just as we did in COVID. When we took large volumes of patients and we gave this drug, that drug, and we started to pull people out of certain arms when arms looked to be ineffective. So you can see how you might be able to do it in some of these other drugs, but doing it in a brand new drug in this particular way is going to require quite a leap of faith, I think, and quite some serious organization, although I think there are a few youngsters who have got it in their blood to try and push that agenda forward. So what about surrogates? There's that famous quote that we always come back to, which is, a correlate does not a surrogate make. And so wouldn't it be nice if, rather than just looking at deterioration, you could look at improvement? Can you capture improvement for a patient? And also, is this a surrogate survival? So this was came out of the PULSAR study, which then rolled into the STELLA trial with sotatercept, of looking at this multi-component improvement index. So functional class, exercise capacity, and RV function as assessed by NT-proBNP. So if you hit a 30-meter improvement or in a 6-minute walk, and your NT-proBNP dropped by more than 30% or stayed below 300, and you improved to functional class 2. If you got all of those 3, then you got a big tick in the box. So that was met in STELLA. And then Marius Hooper looked into the comparer cohort and said, okay, fine, if we take incident patients and we look at those people who met that multi-component improvement score, and compared to those who didn't, did they have better survival? And the answer was yes. So could that be a surrogate? The same thing as if you apply the ERS-ESC for strata risk score. If they met if they improved, did they have better survival? And yes. And so you look at all of these different scores, and they all appear to be surrogates for survival. But then you come back to the clever statisticians and say, okay, but is it the case that the effect that you're seeing in that risk score going to mortality is mediated by the risk score, and how much of it is mediated by the treatment? So in other words, just look at the top line here. So in the perfect case where the risk score is telling you exactly what's going on, your treatment changes the risk score, which changes your long-term outcome. But in the bottom one, your treatment changes your risk score a bit, but has a bigger effect on outcome, and the risk score is only telling you a bit about outcome. And you can probably tell where this is going. In fact, I'll skip that slide. Where this is going is it's basically saying that, look, I'm sorry, but what the risk score is telling you in terms of survival in a clinical trial setting is only a small part of the picture. So it's not a good surrogate, unfortunately, to use risk scores as yet in a clinical trial as an endpoint. Then finally, I thought I'd put this in, what about actigraphy? Could that be a primary endpoint in clinical trials? So this was attempted using an actigraph monitor and collecting quality of life information in the TRACE study, which Olivier mentioned, or somebody mentioned, might have been Sean. So this was a phase 4 study looking at different types of activity using an actigraph. So you've got sedentary, then you've got non-sedentary activity, and then you've got moderately vigorous physical activity. So you've got these different categories. And the intervention of choice was selective bag in this example. So this was a randomized, double-blinded, placebo-controlled phase 4 study looking at whether or not actigraphy improved following selective bag. And it's the first time we did this study, and you can go back and you can say, oh, maybe we should have left it longer, or we should have, intervened with something to encourage exercise. And what this shows, along with the Bellerophon study with inhaled nitric, is that, yes, while you might be able to improve other outcomes, patients don't necessarily do more. So I think we've got a bit of work to do, actually, on actigraphy, and there's probably a lot more we can look into inside the metrics we get back in terms of the granularity, how fast do people move when they choose to move, and so on. But it was just a start, but it's certainly not ready for prime time. So, very simple conclusion. In early phase studies, I think there is scope to look at new technologies, but I can't see phase 2s changing a great deal. In phase 3, 6-minute walk has made a comeback. And it does lead to more rapid approval of drugs with less study fatigue, and we haven't yet found a good surrogate. So, thank you for your attention.
Thank you so much, Lok, for your nice talk.
Thank you. The session open for discussion. Fatma, go ahead.
Hello. Sorry. I just want to ask about the CardioMEMS, since there's an option to get a cardiac output, and probably maybe even pulmonary artery compliance could be checked, because heart rate could be checked and stroke volume could be checked. And I know that probably CPR or pulmonary artery compliance may be an endpoint in clinical trial, but until date we don't have it as an endpoint in real clinical practice. So whether it might be helpful in clinical practice or not, this is a question whether this is going to be helpful in clinical practice or not. And number 2, did you have any difficulties in implanting CardioMEMS in patients with pulmonary hypertension, because those patients, they have dilated PA, and it's very hard to get the proper size, that proximal and distal loop, you need a proper sizing to implant the CardioMEM. Were there any difficulties? And if there were any difficulties, was it related to the severity of PH, like a patient with a lower risk had easier access to implant the device as opposed to patients with high risk?
So, it can be a bit hairy putting these devices in. I'm fortunately not the person who has to put them in, but we have a very good proctor who comes down from Sheffield, Alex Rothman, and he's been training us up at Hammersmith to put these devices in. And we have an interventional cardiologist with us, as well as the standard person doing the right heart cath. So it can be a bit hairy when it springs back and you've got no way of dealing with it. But we're building up experience. We had no complications, but it's not something to do on your own. But I'm not really aware that there's a particular difference or difficulty between, for example, a PAH patient, whether they're high risk, low risk, and say a heart failure patient. Once you can navigate around the right atrium, right ventricle, I think it's fairly similar. And then in terms of, we're still learning. We're still learning about these devices and the information they can give us. I think having the cardiac output and therefore the stroke volume is going to give you that instantaneous measurement of pulmonary artery compliance, which, as you say, is important, and I think is increasing in importance. So it's still very much early days, and we're trying to see what we can use it for in trials. I've used it in a couple of patients when I've had to withdraw, for example, ibuprostanol for particular reasons, and it's given me that kind of certainty. So in certain scenarios, I find it useful, but it's certainly not something that we should be going out and doing on everyone.
Majdi, go ahead.
Thank you to all. This is really wonderful. I'm just having a question probably to Shane, but I'm really troubled with this statement. We don't know if we need to treat early or mild, basically disease, when the real mean pressure is between 21 to 24. I think this statement is really unheard in any field of medicine. I mean, you don't see oncologists saying that I'm not going to treat mild cancer. I'm going to wait until cancer is spread, and so I don't treat mild heart failure or diabetes. Now, I do understand that really the studies were done on the mean pressure of really 25, but isn't the whole idea of picking the early disease is to improve the outcome. I mean, there is really good data by Aurelie Sidbo and others saying that those who are treated at function class 1 or 2 getting better outcome. Really Nazarino-Galay data, really in the early study showing that if you don't treat patients early within 6 months, the really PVR deteriorated. So why we are still hearing this really statement? We don't know whether we should treat patients with mild disease. I'm gonna kind of telling my patient, let me wait until you have more aggressive disease to treat you. So what's your take on that?
Yeah, thank you. Well, I'd say, first of all, the reason for changing, as we heard, the guidelines, was that having any amount of pulmonary hypertension and rise in your PVR is associated with worse outcomes. So it is a risk. That's the first thing. So it's important to identify those patients because something bad is going on. The studies don't tell us whether or not treating that will make a difference. They may do worse, but whether treating will make a difference. So that's the second part. The third, I suppose, is in general, when you've got a patient with IPAH, they don't show up at 22 and then a month later, you sit on them and you find out that they're at 50. That rarely happens. It might happen in scleroderma and in a few other areas, but in PAH, proper, heritable, or a young lady arriving with PAH, when they arrive in, even with those who have genetic or heritable, you see that you might be following them for a couple of years, but then they're right up here. So I don't know that there is this slow trend and we're in danger if we don't treat this, that we're missing a really key opportunity. I think when we get somebody in that area, we've got to look at their risks and say, is it the comorbidities? Is it their sleep apnea? Is it their stiff left ventricle? Is it their mild COPD? Look at all those things. And if you're left in the end with somebody, we'll say, scleroderma who's in a screening program, you pick up on them and they're mild there. Those are ones then that I would agree with you that you say, well, look, I have no other explanation for this except they got scleroderma. I went looking for it in a screening program. They were asymptomatic. I've got to do something here. And in that situation, I think as a clinician, I often will treat the patient in that situation, monotherapy, say, I'm going to do this. But that's after doing a lot of other things before I get to that one particular patient. So it is associated with bad outcome. The studies weren't done to tell us. And it's not really the natural history of the majority of Group 1 patients. They're just an important group for us to now study between now and maybe the next World Symposium.
Thank you.
Thank you.
Yes. Faiz. Thank you for the speakers. Really great session. And my question again to Prof. Shongen. So it's a kind of practical question. So we kind of seeing in the clinical practice a female young lady on dual combination therapy. She's low risk, but if you look into her hemodynamics, her mean of 70 and PVR more than 10, and she's still low risk on dual combination therapy. So what's your advice actually? Shall we continue just on dual therapy and ignoring mean of 70 and PVR more than 10?
I think we might direct the questions to Athenis because she's the one.
Thanks. I do think if the three non-invasive variables are in the green zone, we do not need to perform right at CAT, and it's unusual to have very severe hemodynamics. I mean, not only mean PAP and PVR, but also right atrial pressure and very low stroke volume index. It's unusual if the three non-invasive low risk are in the green zone in the low risk status. So for this kind of patient, I do think we should continue the same therapy and to reassess her. If we observe worsening in the future, of course, we have to consider treatment escalation.
Yeah. You want to add anything? Okay.
No.
Okay. So I think in the same thing of risk stratification, I think you show us the limitation of our risk stratification, the need of invasive, in addition of cardiac imaging. But I think we are not considering two important points when it comes to the risk stratification. Number one, the underlying disease. We have some specific characteristic, for instance, systemic sclerosis. We have some biomarker to tell me that this patient having good or poor prognosis in addition of the risk. In addition, the emerging therapy, now we have potential biomarker which might be incorporating the risk stratification. What's your intake from this?
Okay. Regarding the underlying diseases, I think it's of course very important to consider this and to consider the use of immunosuppressive therapy if needed, and maybe if the patient has a lupus, for example. But we have to consider both the severity of PH and the underlying disease. What was the second question regarding biomarkers? There are several teams who try to identify new biomarkers which can be added in the current risk stratification tools, and some interesting biomarkers have been identified, but we need further studies to validate them in real life, because so far they have been validated in 2 or 3 cohorts, not more. But this is where we want to go in the future, and especially also to identify biomarkers which will be able to predict response to therapy and not only the survival, because what we want is to go to precision medicine.
Okay, thank you. Yeah, go ahead.
Sorry, that was just touching on what I was going to ask. So I just wanted to ask Anthony and Olivier if there was any update on the biomarkers since we've met in Sheffield about 9 months ago.
Sorry?
Asking about the new biomarker for the emerging therapy, if anything to be considered for the future biomarker.
Biomarkers?
Yeah.
No, I think biomarkers, that's very, very important, and I think you partially replied about biomarkers. I think we need perhaps more prognostic biomarkers, but BNP or NT-proBNP works very well. And perhaps now we need theranostic biomarkers to predict the response to therapy. And with the different new drugs we have today, it seems that perhaps some patients may respond to certain drugs and some other to others. So today we don't know how to detect those patients who are more able to respond to, I would say, sotatercept or PDGF receptor inhibitors. Today we don't know. So today our attitude is to combine to be sure that the patient will respond. But perhaps in the future we will have more precise.
May I add something, Olivier? Regarding prognostic biomarkers, I don't think we need new prognostic biomarkers because we are not that bad so far using the 3 non-invasive variable plus hemodynamics. I'm not sure we will do better. We are able to identify the patient with a poor prognosis, and this is sufficient so far to say we need a treatment escalation, but I think the future is to identify the theranostic biomarkers. This is a very important thing, much better than the prognosis biomarkers.
Khalid, and then
I think
Naz.
I would give it to Galia. He's way more important than me.
Go ahead, Naz.
Thank you. I congratulate Athenaeus for her presentations, very informative. And I would like to ask her help for a doubt I have. For example, I have 2 patients in the intermediate risk group, but one patient has 1 drug as background, another patient has 3 drugs. But the current risk stratification is not capturing the information of the background therapy. How can solve this issue?
Wait.
This is an important question indeed. What do you want to say, Olivier? The way. Now, we are not capturing the use of the drugs that we have in the current risk stratification. I don't know how to integrate this in our risk tool.
So this is work for the 8 World Symposium.
Exactly.
Okay, Khalid, last question.
Thank you. I think this is for Olivier, since he has probably the biggest registry of patients in this hall. Do you think eventually we will have these registries publicly available for people to try to work with them and try to come up with some ideas. Like we have for sepsis, we have 35,000 patient database publicly available where researchers can work on them. Or maybe the data where PVR between 20 and 25. If you have all this data out for people to capture and play with, especially now with AI, there are some computer scientists very interested in looking at this and using AI and machine learning.
No, I agree that registry data are very, very important to try to answer unanswered questions we have today, like the 21-24, as it was discussed previously. The problem is, it's good to have data in registry, but we have to get good data, not just to collect the data like that. We have to collect good data with a good monitoring. I think it's very, very important. And if you want to, we have a list of unanswered questions, of course, and I'm pretty sure that using collaboration between people having a quite big database, probably we will be able to respond to answer some of the questions we all are in our minds.
I think there should be a move from the World Symposium to really push these researchers to bring out all the knowledge and all the information.
There are two examples. Either you have very good data, very good proper registry, or you have just a mixture of all the registry worldwide, because it is an initiative of PVRI, for example, the GoDeep registry. For me, GoDeep registry is not the right example to follow, because that's non-monitored data. Perhaps some have a different opinion about that, but you put all the data all together and you can try to pick up the data you are interested in, and you try to reply to a question. So for me, it's much better to have good registries like they have like we have in France, like they have in Bologna, like Compera, like others, and to try to collaborate, to try to answer some unresolved questions.
Thank you. We need to close. If you have more questions, please approach the speakers during the break. I would like to thank all the speakers and my co-chair. Thank you very much. The break for 15 minutes, so we'll be back at 3:30. Thank you.
السلام عليكم ورحمة الله وبركاته. Good afternoon, everyone. So I would like first to thank Dr. Abdullah Dal'an and the organizing committee and the sponsoring company for this great opportunity to be here this year with you and SAPH 2025. So we'll be opening and introducing the last session for today, so session number 5, and it's going to be about pH management. I know it's a Friday afternoon, but we hope that will keep you entertained until the end of this session. I will be sharing this session with my colleague, Dr. Shaya Shai, who will be introducing the first speaker today.
Hi, good evening, everybody. So I would like to introduce first speaker. He's Professor of Cardiology, Dr. Jean-Luc Fachiri, from Brussels and Belgium. He's going to talk about group 2 pulmonary hypertension versus pulmonary arterial hypertension with cardiac comorbidities. And we're going to talk about some updates. Go ahead.
Salam alaikum. Thank you very much. I would also like to thank Abdullah and Mr. Hassan for their kind invitation. It's always an honor and a pleasure to come and visit Saudi and attend the SAPH meeting. So the task I received is twofold, looking at pH with comorbidities, for which you already had a couple of insights from my previous speakers and colleagues, and also going a little bit deeper in the group 2 pulmonary hypertension in terms of what are the treatment updates. I just want to start with sharing with you a couple of financial disclosures, including companies that you see highlighted in red with whom I've worked on the field of pulmonary hypertension in left heart disease. So I thought to start by giving you a little bit of an overview of pulmonary hypertension associated with left heart disease since the beginning of the World Symposium. It's 64 years of history, but it's only in 2013 that we had a separate task force looking at pulmonary hypertension due to left heart disease, that it was named at that time. And then we had the 2018 and 2024 new task force proceedings being published. And I must say that despite the apparent stagnation in the field, there were a lot of progress that were made over the years. Of course, you know that the problem we're having in making the difference between what is PAH with comorbidities and group 2 pulmonary hypertension is a little bit a matter of differential diagnosis, but a lot of problems when it comes to management, because of course the management of both conditions are extremely different from one another. And also one thing we should underscore is that when our patients are growing older, even if they have PAH, they may happen to have cardiovascular comorbidities. They may start having systemic hypertension, obesity, and other common risk factors in the elderly population. Not only it has direct impact on treatment strategies, but also in clinical trials. And this is something we have to recognize, specifically when we talk about clinical trials for PAH. And I would like to show you one of the slides. I think I left SAPH last year in February with these following 2 slides looking at what has changed in the field of PAH with respect to comorbidities. I think the first step was made at the AMBITION trial when this new emerging population started to arise and start to be identified in clinical trials, i.e. those patients who had PAH, precapillary pulmonary hypertension, but also traits and risk factors, mostly for heart failure and preserved ejection fraction. And this is why the amendment was proposed during the AMBITION trial, which is a change, of course, in the inclusion of patients, especially for those who had wood units, PVR between 300 and 500, which is typically the range in which we would find patients with group 2 PH or more likely to have HFpEF as a cause of pulmonary hypertension. And hence, the exclusion criteria came up with 3 of the following: BMI, hypertension, diabetes, and history of significant coronary artery disease. So that was really the first step forward to try to tackle or at least identify the problem of comorbidities in PH. If you look further down, and in more recent years, especially those phase 3 trials or phase 2-3 trials that are currently ongoing or were stopped, you see that there's a kind of an heterogeneity between what is qualified as a cardiovascular comorbidities, left heart disease traits, and hemodynamic criteria. There's no homogeneity across those three important trials, PROSERA, IMPACT, and INSIGNA PAH. And this is something we need to work on because one of the difficulties for us to get patients in trial is to be sure that we recognize those traits before embarking in specific new treatments. Now, the consideration I would like to make, and to close that question of PH with comorbidities, is that surely Ambition has identified this population that we know will have a blunted response to specific therapy, especially the components of any of the risk scores, and this is important. Even if in Ambition there was a benefit in time to clinical worsening, the effect on NT-proBNP walking distance and functional class was not as spectacular as for the other patients. In contrast, what we see in registries are patients with PH with a burden of comorbidity that is so high and also ages that is so different from the clinical trials that you may sometimes wonder whether those patients who are referred to be atypical PH may be in between PAH and pulmonary hypertension associated with left heart disease. And in fact, also their hemodynamic presentation appears the same. This is something that was seen surely in the Compera registry. So we have options to have a better representation of those patients with PH and comorbidities to get them closer to a real life situation in clinical trials. The first option is to expand the inclusion criteria by allowing patients with cardiovascular comorbidities in randomized controlled trials. However, this is increasing the risk of confounding the benefit of new therapies and probably might raise issues with safety and tolerability. So maybe it's not the right way to go. Surely the right way to go would be to start thinking of pragmatic trials, and this is something I will come back on a little bit later, with approved therapies, Ambition-like trials, and this is something that is currently done by our colleagues in France with the commodity trials led by Laurent Saval and Olivier Sidbon. The idea is very simple. It's similar to Ambition. It's using drugs that are on the market and including patients who have those comorbidities and see whether there's a difference in outcome. I think this is something we might do further down the road when more drugs will be on the market. Now, one of the things I've always wondered, after all, is why do we care that much about the response of patients in the real life? And as a heart failure specialist, I like to bring up that the way we do see things in the heart failure world, this is a very interesting analysis that was done by colleagues mostly in the Netherlands, looking at the response to the approved therapies in heart failure with reduced ejection fraction in registries. So what they did is that they took the average inclusion-exclusion criteria of 5 leading randomized controlled trials, the ones that we all know in heart failure. This is quite recent, it's been published 2 or 3 years ago. And they compared those 16,000 patients, that's quite significant, to 46,000 patients coming from 2 heart failure registries. So you have here the application of inclusion-exclusion criteria of clinical trials to registries and the clinical trials. There was a big difference between RCTs and registries, higher left ventricular ejection fraction in RCTs, but in registries, whether you look at eligible patients or non-eligible patients, they were older, more female predominance, lower ejection fraction, and more non-cardiac comorbidities. And the result of their analysis was that in a registry, if patients were meeting the inclusion-exclusion criteria, they had still a worse outcome as compared to what was learned in clinical trials. But it was even worse for patients who would have been excluded in clinical trials. Despite this, we use those treatments for patients with cardiovascular comorbidities who do not fit with the inclusion-exclusion criteria in clinical practice. So why would we in PAH, a rare disease, do differently from what is done in heart failure? And the main driver for difference were age and gender. So clearly, what we see in heart failure may be exactly the same than what we see in pulmonary arterial hypertension. You've seen that a few times already today. And not only today, so weighing comorbidities to make treatment decisions is fine. I've heard that from all of my colleagues and friends. But the question is, how are we going to do this in clinical practice? And what does that mean, weighing comorbidities versus pH severities? Well, this is something that we've worked on in the task force at the World Symposium with colleagues who are present, Stefan Rosenkrantz and Ian Lange, and other colleagues from the US. We thought that it would be important to raise attention to the fact that there's a difference between a comorbidity such as obesity, diabetes, valvular heart disease, for example, as opposed to risk factors. These are two different things. A comorbidity is something, it is a disease that is present. A risk factor is increasing the risk to develop a disease. When you get that, and when you look at this list, you can immediately see that not all those comorbidities or risk factors weigh the same in the balance. For example, if you have valvular heart disease, arrhythmia such as atrial fibrillation, the weight is much higher as opposed to systemic hypertension, for example. And surely this is true for differential diagnosis, but also for patients in PH. So we propose an absolutely arbitrary scoring system, so it hasn't been validated by anybody, but we think it would be worth testing the hypothesis that the burden of comorbidity can be weighed according to mild, moderate, or severe risk factors, then low, intermediate, and high probability. I'm not going to detail this, but again, if you have obesity with a BMI over 40 kilograms per square meter, if you have atrial fibrillation at baseline, the severity of the comorbidity is much higher as opposed as a patient who had a single episode of atrial fibrillation. So this probably is one of the things that can be tested, at least surely, in well-kept registries. We also made a proposal to match a little bit what the ACC and the HA are doing, colleagues are doing in terms of staging left heart disease and staging heart failure. And we put forward the idea that there's probably a difference in the various stages of pulmonary hypertension associated with left heart disease. And we would like to emphasize the fact that it starts with a stage A, which are the patients who are at risk. So the accumulation of risk factors, the accumulation of comorbidities is increasing the risk to have pulmonary hypertension. And then progressively, patients would transition to over-structural heart disease, symptomatic heart disease, and advanced right ventricular, predominant right heart phenotype, so tilting from a predominant left heart to a right heart phenotype. This is important for potential therapeutic targets because this will have an importance, especially when we think about potentially preventing pulmonary hypertension in some left heart disease, and future investigations and future endeavor at each stage of development of pulmonary hypertension in the context of left heart disease. Now, treatment update, I could in fact stop here and say there's nothing new, which is not completely true. We have novel information that I would like to share with you, but I couldn't emphasize more the fact that for any cardiac condition that is a cause of pulmonary hypertension, there's a treatment. And this is showing how the association of angiotensin receptor and neprilysin inhibitor therapy may reduce PVR in patients with end-stage heart failure with reduced ejection fraction who are candidate to receive a left ventricular assist device. So not only those treatments are able to lower PVR, but if you also unload the heart with an LVAD, you will have a normalization of pulmonary vascular resistance. So this is important for my friends cardiologists to know that, but also my friends and colleagues pulmonologists who may not be aware of this specific situation. So treatment of pulmonary hypertension in group 2 starts with the treatment of the underlying disease, that is the cardiac function. Now, one of the things that we need to keep in mind if we want to establish a treatment algorithm for group 2 PH, which by the way does not exist, is that we need to rely on the best evidence that we have, which is supporting practice with something that I believe is extremely important for us as physicians, is that our patients must receive therapies combining efficacy and safety. And the evidence is coming from clinical trials, meta-analysis, registry, retrospective analysis, and case series with a lower level of evidence for registries and retrospective analysis, the highest for meta-analysis and randomized control trial. So I know you're probably sick of seeing those slides. It seems that I'm like a broken record, but I'm trying to change the design of the slides each time I'm presenting. So it's a little bit more entertaining rather than putting the same tables again. So this is showing you the number of trials that were done in all the settings of the current classification for group 2 pulmonary hypertension. Valvular heart disease, we have one study, COVAC, using sildenafil. In heart failure, this is clearly where you're going to find the highest level of evidence. I'm not saying whether it's positive or negative at this stage. You see HFrEF, mildly reduced ejection fraction, and preserved ejection fraction. So this is a list of the acronyms of the randomized controlled trials that you can find. I think it's important to highlight that in the recent years, in red, some trials were associated with safety concerns. In bold, you will see the trials that are stopped before completion. Whatever the reason that the study didn't complete, in most cases it was because the sponsor was pulling out his support to the completion of a clinical trial. So this to me starts to be worrisome because we have now more harm than good in most of the situation in the context of well-kept randomized controlled trials. And unfortunately, the last one that was published by Max Hupper and Stefan Rosenkrantz, it's the PASSION trial looking at oral tadalafil in a group of patients with combined post- and precapillary pulmonary hypertension with a primary endpoint as a time to first event. The study was stopped early because of pulling out funding for the study medication. In the population that was analyzed, that was almost half of the one that was supposed to be recruited, there was no benefit, no sign of benefit, but there was a signal towards a higher increase, to an increase in all-cause mortality. So my plea here, and that's of course personal opinion, should be D5 be given to patients with pulmonary hypertension associated with heart failure. The answer is no. And this has been reinforced at the last World Symposium, where we do keep the negative recommendation based on those analyses. I would like to draw, I'm not going to read this in details, but this table of recommendation, gaps in evidence and unmet need, is really pointing out to the direction that we want to make, and especially one thing that is very new is prevention of pulmonary hypertension in some left heart disease. Just to close on the last part, which is one that is commonly discussed, it's how do we build randomized controlled trials in group 2 pulmonary hypertension? One of the things we did is looking at the potential population and the endpoint and the strategy that can be used, and we made the difference between the conventional randomized controlled trial, observational studies, and pragmatic trials, and you will see that this might be important. One of the things we believe, at least at the early stage, is that we need right-side catheterization for proper phenotyping of pulmonary hypertension, otherwise it wouldn't make a lot of sense. Then we have different populations that are surely populations of interest. The one is probably the population that we all believe is the most important, which in fact turns out not to be the most important. So the patient who have HFpEF as a cause of pulmonary hypertension, very high PVR above 5 Wood units and wedge pressure above 15. There are other populations, those who have wedge pressures in the gray zone between 12 and 15 to 18, but an abnormal response to exercise, high NT-proBNP, and abnormal or signs of right ventricular dysfunction. And finally, those patients who do present in the cath lab with heart failure with preserved ejection fraction, but a wedge pressure below 15, who may also have an abnormal response to exercise. Here, you see the value of all these strategies. Clearly, randomized controlled trials and pragmatic trials are on top of the list of potential answers to those questions. Then we thought about design and endpoints. I like what Luke said on PVR as a primary endpoint in the phase 2 trial for PH. It is probably the same in pulmonary hypertension associated with left heart disease. At least we want to see PVR going down. It's going to be the preferred endpoint in phase 2 trials. It's feasible in RCTs, but also in pragmatic trials because it doesn't require much in terms of observation. Now, of course, you have two very important endpoints, not only for PH, but also for heart failure, exercise testing, as much as we love CPET, 6-minute walking distance is easier to perform. Probably one of the endpoints to look at, the secondary endpoint, surely in the phase 2 trials. And finally, we have NT-proBNP, echo-derived or CMR-derived variables that are at this stage probably exploratory. I mean, there's at least one trial in heart failure that look at left atrial strain as a primary endpoint with a drug called relaxin that unfortunately didn't demonstrate any benefit as opposed to placebo. So picking up the right endpoint is something that is very important. Finally, whether we like it or not, if you want to move from phase 2 to phase 3, you cannot escape the combined endpoint of mortality and morbidity. It can be either all-cause or it can be cardiovascular morbidity and mortality. And in this case, surely for all-cause mortality, morbidity, randomized controlled trials, but also pragmatic trials may inform the community on the performance of some of some interventions. And I would like to point out the fact that this has been done in the field of heart failure. Those two trials, STRONG-HS and ADVO, which by the way was a trial done in Belgium, are purely pragmatic trials using drugs that are already on the market, and those trials being positive supported changes in guidelines. So this is something we should keep as a potential food for thoughts. Last two slides. This one is depicting the selected ongoing trial in pulmonary hypertension, especially associated with heart failure. A cadence with sotatercept, change in PVR as a primary endpoint is close to completion. We hope to see the results by the end of this year. The inclusion criteria were expanded to allow patients with valvular heart disease. Levosimendan, that has been developed orally, is also looking at change in 6-minute walking distance in a very different population than the one we would have in a pure pulmonary hypertension trial, although right heart cath has to be performed with a PVR above 2 Wood units according to the new definition. And then two trials, very interesting, REFIRE and APEX, looking at a similar pathway, which is the recombinant relaxin that can be given subcutaneously in patients. Those two trials are looking also at changes in pulmonary vascular resistance after 24 weeks of injection. None of these drugs are pulmonary vasodilators, but they have an effect on the left heart and potentially the right heart. And that's probably one of the targets we need to have in mind when speaking about group 2 pulmonary hypertension trials. So a couple of take-home messages. That's again a personal opinion. It's very unlikely that we would include more patients with PH and comorbidities in future RCTs. Randomized controlled trials can be substituted with pragmatic trials, as one that was done that is currently done by our French colleagues. And after all, Ambition is a pragmatic trial in a way because it was using drugs that are already on the market. There's now, I think, a change in the paradigm in group 2 pulmonary hypertension where we move from applying PH science to group 2 PH, two different diseases, to think more about how to treat the left heart, how to prevent pulmonary hypertension, rather than trying to chase a ghost that is pulmonary hypertension, a severe pulmonary hypertension in group 2. And I believe this change in paradigm is going to help to develop more specific targets, perhaps not to the pulmonary circulation, but more closely affecting the cardiac function itself. Thank you very much.
Thank you so much, Professor. Please join us and join the panel for further questions at the end of the session. Now it's my pleasure to introduce our second speaker. So our second speaker is Professor Hossein Ghofrani. Professor Ghofrani is the head of the Pulmonary Hypertension Clinic and Center in Germany. He's also the head of the Pulmonary Vascular Research Institute. Dr. Ghofrani will be talking to us about group 3 pulmonary hypertension and PAH with pulmonary comorbidities and how do we treat and update and management. He's joining us virtually.
Assalamu alaikum.
Alaikum assalam.
I hope everyone can hear me well. Thank you, first of all, to the organizers for including me. And I'm really sorry I cannot be there in person, but I'm happy to share some thoughts on the subject of Group 3 PH. I'm also grateful to my good friend Jean-Luc for opening the discussion on non-PAH PH groups and as he said, I don't want to sound like a broken record, so I try to spice up my talk a little bit with some original data, which I hope you will also enjoy. So when talking about group 3 pulmonary hypertension, and the analogy was also mentioned already by Jean-Luc, we have to consider that there is a clear distinction from group 1 PAH with lung comorbidities, and there is a straightforward path to making the diagnosis. However, we also appreciate that there's a gray zone in which sometimes it's difficult to distinguish whether the underlying parenchymal lung disease is a leading cause of the morbidity that patients experience, or whether it is predominantly a pulmonary vascular disease, which is basically enriched by problems caused by some components of underlying lung disease. So in the phenotypization, I believe that for all group 3 patients, an imaging, sophisticated imaging is paramount to really quantify the underlying lung disease. In COPD, we have a very strict classification, as we also have for the various forms of interstitial lung diseases, including those that are idiopathic and those that are associated with underlying disorders such as rheumatoid disorders. What both groups basically have in common when it comes to pulmonary hypertension is that there is a hemodynamic definition, which we borrowed from the classification for PAH, which demands the existence of chronic pulmonary hypertension according to the new classification criteria, with the involvement of lung parenchyma. Currently, there is no recommended therapy for those patients with COPD-PH amongst the patients with group 3 PH. However, for ILD-PH, according to the INCREASE study, there's a recommendation for using inhaled treprostinil, and I'm coming back to that particular study towards the end of my talk. In the guidelines, there is a note on the probable use of PDE5 inhibitors, which may be considered in severe forms of pulmonary hypertension in conjunction with interstitial lung disease. But at the time the guideline was assembled and the papers were written, there was very little documented evidence to support it, rather than some anecdotal and small center reports which promoted this idea. So what we basically need, and I'm coming back to the idea of looking deeper into registries and large-scale registries, is an in-depth phenotyping of these patients, and also learning from big data on how to distinguish between the leading causes that are relevant for the outcome of these patients in larger data sets. Regarding the management of these patients, it is said that obviously, particularly for patients with ILD-PH, but that is also true for patients with COPD-PH, that the treatment of the underlying disease and the hypoxemia is the basis for treating these patients. Patients, of course, who are eligible for lung transplantation should be referred accordingly to high-volume transplant centers. And then issues like pulmonary rehabilitation, supportive care, and symptom management are also recommended. Apart from that, and apart from the very few patients who have an indicated treatment with inhaled treprostinil, the inclusion into a clinical trial, such as also Jean-Luc advocated for the group 2 PH, and also individualized management is recommended. The domains upon which we can focus on working up these patients are both clinical domains, hemodynamic domains, functional domains, biological ones, morphological, and other considerations, which help us to categorize these patients. And based on the severity of the respective subdomains, there is either a higher likelihood of a predominance of vasculopathy versus a predominance of the underlying parenchymal disease. One other thing we should keep in mind is that this disease and the combination of both parenchymal and vascular disorder is a continuum and a matter of time in the course of the disease of our patients. So doing a right heart catheterization in a patient with ILD and proving that there is no pulmonary hypertension present today does not mean that the same patient may present with severe pulmonary hypertension in a time span as short as few months. And this is an observation we borrow from large-scale transplant programs where regularly right heart catheterization prior to transplantation is performed, and the percentage of those patients displaying chronic pulmonary hypertension is excessively high. So we undertook an attempt to gather large-scale data, which were hitherto lacking in the field of pulmonary hypertension. And for this, the PVRI Go Deep consortium, which is very well known to all of you, was formed. This is a meta database which subsummarizes in a central database, subsummarizes the data available from different high volume and mid volume sites across the world. So it's a truly international meta database, which this is even an older slide, meanwhile encompasses more than 33,000 patients in this meta database from centers such as Gießen, but also in the US, in the UK, and many other countries across the world. Basically on a daily basis, new sites are initiated and happily include their data into this meta database. So from this very large patient population, investigations were undertaken with certain selection criteria to come up with patients from group 3 pulmonary hypertension. And at the end of the day, in this particular paper, we were able to find more than 3,500 patients with non-imputed data from group 3 pulmonary hypertension. And the first thing that was of interest for us was how the hemodynamic severity across the different groups of pulmonary hypertension were in these patients. I draw your attention to the first column, which is PAH, with a typical gender distribution and functional class distribution with a severely impaired functional capacity of these patients and severe hemodynamics. And in group 3 PH, we found that there is also a substantial reduction in exercise capacity. However, hemodynamics, although being clearly impaired, were more moderately expressed than in group 1 PH. Another issue that was of interest for us was whether or not the established risk strata are applicable also to patients with group 3 PH. And for that, we compared the different strata we are currently using and their ability to predict outcome in patients with group 3 PH. And we're quite pleased to see that both the ESC ERS 3 strata model, the Compara 4 strata model, as well as the Reveal 2.0 risk score worked perfectly well when looking at the entire group of group 3 PH patients as compared also to group 1. Diving a little bit deeper into subcategories of group 3, the same was also undertaken for patients with COPD pulmonary hypertension in a sizable patient population that also had invasive hemodynamic data available. And it could be shown that as compared to IPAH in light blue, those patients with PAH of the non-IPAH group had a comparable outcome as opposed to patients with COPD PH who had a much worse 5-year survival rate than the PAH groups. The COPD PH groups could then be subdivided with regards to the impact of hemodynamic severity on outcome. and it could be shown that a PVR above and below 5 Wood units was a strong denominator of outcome in these patients, and that patients who had more mild vascular disease had a much better outcome, comparable actually to patients with PAH, as opposed to those that had hemodynamic severity with a PVR of higher than 5 Wood units. So this is very strong data. And then it was, again, an analysis was undertaken to not only distinguish by hemodynamic severity, but also the real-world use of PDE5 inhibitors. And I'd like to draw your attention to the green line and also the orange line, which shows that both patients with less severe hemodynamics as well as with more severe hemodynamics as compared to untreated patients had a survival benefit. This is not morbidity mortality, but this is a survival benefit shown in this large-scale registry when treated with off-label therapy of PDE5 inhibitors based on clinical grounds, so to say. So how about PH-ILD? I believe that this is a more a better characterized subpopulation, as also trials were conducted in using established PAH therapies. In this group, however, the majority of those trials so far have been negative, with the exception of the INCREASE study with inhaled treprostinil, which for the first time showed in a randomized controlled trial a beneficial impact of prostanoids when inhaled in these patients. Looking at real-life data, there is an earlier report from the UK experience with using PDE5 inhibitors in PH-ILD, and they could show that here in blue, those patients that were receiving PDE5 inhibitors had a better outcome as opposed to those patients who were untreated. Again, coming back to the PVRI Go Deep registry, this analysis was also undertaken in this large-scale registry, showing that when we remind ourselves of the impact of hemodynamic severity, the coexistence of pulmonary vascular disease with ILD is already of a prognostic relevance, showing that the outcome of these patients is significantly reduced as opposed to PAH patients. And when looking at PH-ILD and making a distinction between more severely impaired hemodynamics and less severely impaired hemodynamics, those with a PVR of lower than 5 Wood units had a much better outcome than patients with a PVR higher than 5 Wood units. So PVR at this threshold is a strong determinant of outcome. In these patients, again, comparison was made between those patients that were receiving PDE5 inhibitors in an off-label therapy, and in both subgroups, those with less severely impaired hemodynamics, as well as those with more severely impaired hemodynamics. PDE5 inhibitors had an impact on survival in these patients. But what about the so far only prospective randomized controlled trial, placebo-controlled, the INCREASE study? In fact, in this study, the primary endpoint of an improvement in 6-minute walking distance for the first time was met and led to the approval of inhaled treprostinil, at least in the US and in very few other countries, which again reminds us that it is worthwhile investigating into this subgroup of pulmonary hypertension patients. The unmet medical need is clearly higher than in PAH currently, and encouraged by the data from the large-scale registries, we can take those data, learn from them, and also design upcoming trials with the help of registry data to narrow down eligible patient populations with a higher likelihood of clinical response and potentially opening new avenues for novel therapies. So allow me to summarize that pulmonary hypertension associated with parenchymal lung disease is relatively common when compared to PAH, but sometimes it is difficult to distinguish from PAH with comorbidities, and it needs an in-depth phenotyping, including high-quality imaging and the discussion of every single case in multidisciplinary teams, as it was also suggested by Jean-Luc for patients with group 2 PH. Survival in both COPD-PH and ILD-PH is considerably worse than in patients with PAH. However, the risk scores that have been established for PAH, now based on large-scale registry data, are applicable to group PH as well. And the hemodynamic parameter of pulmonary vascular resistance greater than 5 Wood unit was associated with a dismal prognosis. Therefore, we should particularly focus on these patients and investigate the utility of medical therapies in this subgroup specifically. Off-label PDE5 inhibitors were associated with better outcome. This does not mean that it is generally recommendable. However, amongst those patients who, recommended by guidelines, should be considered for individualized therapies, I think we can feel a little bit more encouraged to at least try PDE5 inhibitors, particularly when the hemodynamic impairment is significant and right heart failure is imminent. The only approved therapy for PH-ILD currently is inhaled treprostinil. Good news is there are multiple clinical trials underway with different signaling pathways that are addressed. Apart from prostanoids, which are currently investigated, there are also tyrosine kinase inhibitors and other classes of drugs which are entering the field of clinical trials for group 3 PH. With that, I'd like to close my talk and thank you for your attention.
The floor is yours.
Thank you so much, Shaya. Assalamu alaikum. As-salatu wassalamu ala Sayyidi Rasulillah. So I'll be talking in the coming 20 minutes about PH in a special condition. But before I start, I would like to have two important points. Now, the first one, that today I forgot to bring my belt. So my pants is a little bit loose, so I'm a little bit anxious. But I'm glad that now I'm covered here. So this is the first thing. Now the second really point, I shaded a little bit the sickle cell disease. I know Dr. Abdullah always asks me to talk about sickle cell. Thank you so much. But I give this talk in the last 3 SAPH. So please go back and listen to the previous talks. I'm not going to talk about sickle cells. So sorry, Abdullah, for that. I'm going to touch base on it, though. So with that, we'll go to the now basically the case. So I'm going to discuss a case that I saw a few years ago in the private practice. Very fascinating case, sad story. 31 years old Saudi lady, known case of limited systemic sclerosis for really few years, 9-week pregnant, primary gravida, referred to us for worsening dyspnea, function class 3. Now this is in the first trimester. So that was really alarming. We know that dyspnea, lower limb edema can happen in the 3rd trimester, but in the 1st trimester, that's worth really noticing. Her baseline evaluation by her doctor 6 months before a pregnancy was kind of unremarkable from the echo point of view. It looks okay. TR jet velocity was only 2.3, good ejection fraction. HRCT showed mild reticulation, which was attributed by treating team to mild NSIP because of the systemic sclerosis. Lung function, only spirometry was done showing mild restrictive pattern. Saturation was okay. When we saw her, she was quite sick. She was tachycardic, tachypneic. Her saturation was low. She required a couple of liters of oxygen to maintain her saturation. She had loud P2 and some leg edema. So her chest X-ray was kind of unremarkable. You can say that probably she had some basically really engorgement in her pulmonary vessels. Her echo, though, was very different than the echo that was done before pregnancy. So you can see that really her TRG velocity now with really high dilated right cardiac chamber, impaired RV function, TAPC scoring is 1.5. So now we are talking about real business here in this young pregnant lady with scleroderma. So despite her pregnancy, we decided to go ahead and do CTPA to rule out acute pulmonary embolism. I think everybody will agree on that. And there was no pulmonary embolism, but what we noticed, dilated really pulmonary trunk and some RV strain. We proceeded to write heart cath, and this young lady had significantly severe precapillary pulmonary hypertension. Her PVR was 14, low cardiac index despite pregnancies, and high mean pressure. So the patient was admitted to ICU. I'm not going to tell you what happened in the ICU because she received some fluid there. Okay, I did not say that, but the intensivist gave her some fluid. Anyhow, we did talk about pregnancy termination. She refused, and her husband refused that, both of them. Parenteral prostacyclin was not available in that center. We tried to really get it, but she refused because someone told her that that drug, you will need it for the rest of your life. So we started her on sildenafil, low-dose sildenafil 25 TID, and we put her on inhaled iloprost every 3 hours in the ICU with Lasix. In a few days, she became sicker, hypoxic, worse. Chest X-ray showed frank pulmonary edema. So the decision was to really stop inhaled iloprost, diurese her aggressively. She needed 2 vasopressors to maintain her basically blood pressure. ECMO team was consulted, and really pregnancy termination was discussed with her obstetrician. After the termination of pregnancy, 3 days later, she died. Unfortunately, with basically multiple reasons. She gets septic, she had DIC picture, severe RV failure. So that was a sad story. So we start to talk about what happened in pregnancy, in normal pregnancy, and in pregnancy with pulmonary hypertension. Now, in normal pregnancy, we know that those patients, they maintain their blood pressure because of drop in the PVR and increase in the cardiac output. However, once you have pregnancy and pulmonary hypertension, you start to have mixed mechanism of that will affect the outcome of those patients, including the increased cardiac output in those patients, basically increase the blood volume, increase the likelihood or the basically likelihood of getting VTE, pulmonary embolism, etc. With all of that together, puts a significant strain on the right ventricle, and the patient can basically have worse outcome and death. So what did the guideline tell us? Now, if you look at the previous guideline, 1215, it was very, very straightforward. Simple recommendation, avoid pregnancy. Avoid pregnancy in pulmonary hypertension, yes. So that was a simple, straightforward case. Now, in the more recent guidelines, now you can see that the language became softer. And now we have a recommendation that, yes, those patients should be told that the pregnancy will have some uncertainty, it might have some risk. But now we are not talking about absolute contraindication for a pregnancy. And also the guidelines take us a step forward to say that in case of that the patient wanted to go ahead and become pregnant, then we need to talk about what we need to do and what to expect in those ladies. And of course, we need, of course, to really adjust the medication to avoid some of those medications that can harm the fetus. But you can see that now the recommendation is very different than the previous guidelines. Now we are giving a chance for some women to get really pregnant. And that based on some registry data. We have data coming from the developing country, mainly from China, talking about what's happening to those ladies. And we have more than 3,500 patients basically reported. We need to keep in mind that most diagnoses came from echocardiography, and in those patients who have basically rheumatic heart disease. And some of them, they have really congenital heart disease. But you can see from the different registry that now the mortality varies from different places. For example, the first basically case series of only 5 patients coming from Malaysia, 2 of them died. So that was 40% maternal death and 15% fetal death. If you look at the data coming from China and South Africa, very, very better result, only 2 to 3% of maternal death and very low 2 to 4% of fetal death. So this is much better than what we used to talk about when we talk about severe bad outcome in all pregnant women. Now, more data came from US, Europe, and South Korea, basically 7 really publication about 150 patients. And you can see now in really 6 of those 7 really case series or report showing 0 mortality for the mother, 0 mortality for the fetus. Only one study that showed 16% mortality in the mother, but most of those patients, they were not group 1, they were group 3, group 2, and group 4. So that could be, again, a different case scenario. But the message that's now coming that we do need to take this as really serious, but we need to discuss the outcome with the patient. It's not an absolute contraindication in those patients. Further on, the 7th World Conference now talk about basically a recommendation what to do in those really women who decided to become pregnant or they found themselves basically pregnant by going to the initial basically multidisciplinary team between pulmonologists, cardiologists, intensivists, and really obstetrician, start to talk about the medication, adjust medication, have a plan for really follow-up, and then plan the delivery and anesthesia in those people. To really summon the pregnancy part, I think it's very important when we look at the pregnancy and pulmonary hypertension, we need to address 4 important stages. We need to talk about the pH assessment, basically in those ladies, and of course symptoms are very important, but again we need to be careful that in the 3rd trimester, most women will get some symptoms, so that need to be addressed. Echo is very good, looking for TAPSE over systolic pressure is probably a good marker to check for RVPA coupling and coupling. Pro-BNP is one way to go, especially if it's really trending up. Right heart cath is not usually mandatory unless there is significant need for that. When you look at the pH medication, we know that many of our medication cannot be used in pregnancy because of their toxic effect on the really fetus. But sildenafil, tadalafil are category C, inhaled ileoprost and really prostacyclin, really analog C. IV epiprostinol is category B, it's very safe. The other drugs are really category X, although there is some case series of restarting endothelin receptor antagonist and selexipag in the 3rd trimester in those ladies who have basically difficult to control pulmonary hypertension. Now, sotatercept is quite important here because we know that sotatercept, in the studies of sotatercept, pregnancy patients were actually excluded. So we don't have data, but we know that the drug does reach the fetus. Also, we need to know that the lady on sotatercept, they are unlikely to conceive because we know that sotatercept is very important for the proper development of the oocyte, and it's also needed to maintain the healthy, really endometrium during pregnancy. So having ladies on sotatercept, it will be difficult sometimes to get pregnancy. What about labor? Recommendation by guidelines that C-section should be basically planned on 36 to 38 weeks. And really our center, we go to 34 to 36. That's our really cases. We believe that C-section, most people believe that C-section is the way to go, although vaginal delivery was also really attempted successfully. Anesthesia, again, all kind of anesthesia can be used, but more expert center will really prefer spinal epidural, very, very tiny dose spinal, mainly really epidural, and that will basically more probably safer for the patient. Postpartum is really very, very important phase to really follow those ladies. Continuous monitoring, some of those people, they might need blood if they became really anemic during the surgeries. And we need to re-intensify really the treatment immediately after delivery, unless the lady is breastfeeding, where again, really ERAs and selexipag are contraindicated. Few words about PVOD. Now really PVOD is an important disease, and it's been described for decades now that we need to be careful when we talk about PVOD patients. Very similar to PAH. It could be idiopathic, it could be heritable, and it could be drug and toxin. It can complicate other diseases, but for the sake of this talk, sickle cell disease is one of those really conditions that can predispose patients to veno-occlusive disease. I was talking with Luke and Sean about one of my cases recently that she is on riociguat macitentan, advanced, really sickler, very severe pre-capillary pulmonary hypertension. We started her on IV epiprostinol and she gets florid pulmonary edema. So she had PVOD and we don't know what to do with her. So when we look at PVOD is different from PAH, although both of them are under really group 1. But we know that they have different genetic, they do have different CT and lung function imaging, where DLCO is much lower in really PVOD, and the treatment are very, very different. In general, the medication that can be used for group 1 PAH can be sometimes hazardous if they are used really in PVOD. The hemodynamics, nevertheless, are exactly the same. So when you look at diagnosis for those patients, clinical diagnosis is really important. Most of those patients are diagnosed in retrospect after they are given vasodilator therapy and they get florid pulmonary edema. HRCT is now probably the gold standard really in those patients. VQ scan, sometimes it's helpful. It gives you very similar pictures to really CTEPH, but it could be normal in some of those patients. Bronchoscopy can be helpful to detect alveolar hemorrhage. Biopsy is not usually needed, and it can be dangerous in those patients. Of course, the gene, the EIF2AK4, is supposed to be really diagnostic. It's present 100% in the familial cases and about 25% in the sporadic cases. Finally, treatment, and very similar to, very easy to say, there is no treatment for PVOD as we speak. Lung transplant is the only approved, if you like, therapy for those patients. However, sometimes we try carefully the vasodilator therapy in those patients. Prostacyclin is usually the drug because you can adjust the dose. You can go on a very, very tiny dose and show efficacy. Other drugs as well have been shown some efficacy, but only the danger of having pulmonary edema is basically there. Some case report about specific treatment. We have one case reporting. We reported one case with really imatinib that she did very well over really few years period. She really moved from function class 4 to 2. Unfortunately, she died 4 years later with a ruptured appendix. So that was really, really problem. There is also some data in the literature about really another TKI, including basically sorafenib and others, but that showed some inconsistent efficacy in those patients. Experimental drugs, using drug that work on really mTOR inhibitor, which is a basically pathway that work on regulation angiogenesis. So now there is some study on that field, but still no conclusive evidence. And my last point to you, what about sotatercept? Would it work in those patients? I think the idea that sotatercept is really antiproliferative and does not increase the cardiac output make it very sexy kind of really basically drug that can be used maybe in those patients. The patient that I was discussing with Sheen and Luke, I said, should I put her on sotatercept? We don't know, there is no data in that, but it is really interesting to think about whether sotatercept will work in those patients. Take home message, ladies and gentlemen, I'm gonna just say 2 words. Pregnancy is still a problem, but it's probably not as much as we used to believe. And now with the modern therapy, more expertise, I think more and more women can go safely over pregnancy without a major problem. When it comes to the PVOD, I think it is a very interesting disease. It's luckily not that common, and we need to use a lot of HRCT, especially in those patients who have a higher risk, including systemic renal sclerosis, sarcoidosis, maybe sickle cell disease, to rule out really PVOD in those patients. There is no standard treatment as we speak, except really lung transplantation. And with that, I would move my pants on a little bit, and then thank you very much for your attention.
Thank you so much, Prof. Majdi, for this elegant presentation. As usual, please join us in the panel. So it is my great pleasure now to introduce Professor John Granton. I have been waiting for this since they announced the scientific information of this meeting. So Professor John Granton is coming from Toronto, Canada. He's the Professor of Faculty of Medicine in the University of Toronto, and he's the Pulmonary and Critical Care Medicine Consultant. He will be talking to us about lung transplantation, when to refer, when is the right timing, and what's the outcomes.
Thank you very much. Mahaba. Thank you very much, Professor Al-Dallin, for the invitation and the organizing committee for allowing me to meet with you today. As per usual, transplant is last to talk, but I'm hopefully going to convince you, and I think a lot of my predecessors on the podium today, I think, have already made a strong case that you should start to consider transplant much earlier in the patient's journey. So a few disclosures. What I hope to do is highlight the unique aspects of transplant as it relates to pulmonary hypertension, the diagnosis as it relates to what modality should we choose, the timing, some details around the allocation of organs, and a little bit around outcomes and how transplant for pulmonary hypertension differs for other indications. And finally, I'll discuss a little bit on how we've incorporated strategies to increase the donor pool, to increase the opportunities for patients who require transplant. I should emphasize that transplant is the only proven disease-modifying therapy for pulmonary hypertension. So first of all, the type of transplant. I think most, and this is a Delphi consensus, which was run by Nicholas Kalatis, which he allowed me to present. This is submitted for consideration. Most of the centers do not advocate for single lung transplant, given the technical challenges associated with that. Equally, most centers are moving away from heart-lung transplant. As was discussed earlier, after transplantation, the right ventricle essentially goes back to near normal very quickly. And as a result, there's generally no RV that is too sick to transplant. In addition, a heart-lung transplant utilizes a heart, which takes it away from another potential recipient. And as a result, represents a potential waste of an organ. And in general, heart transplant, heart-lung transplant, is reserved for patients with complex structural heart disease that can't be repaired at the time of lung transplant, or patients who have intrinsic left-sided heart disease, valvular heart disease, or cardiomyopathy. For the most part, bilateral lung transplant is applied for patients with pulmonary hypertension and pulmonary arterial hypertension. So what I hope to do for the next little bit is divide the discussion around transplantation into three phases. The first is a discussion around transplantation, and that really lies with us as pulmonary hypertension clinicians, and speaks to the importance to educate ourselves and our trainees about the nuances of transplantation. And as Sean showed, this discussion should be probably moved much earlier to the time of diagnosis, particularly in individuals who have a high-risk condition or who have high-risk factors. And then moving to referral and listing, and the cadence of moving through that really depends on several factors. And those are patient-related, disease-related, transplant or pulmonary hypertension center-related, which I call the pulmonary hypertension transplant ecosystem in your own area or jurisdiction. First of all, patient-related, we look at modifiable and non-modifiable risk factors. An earlier discussion for a patient who's obese, a smoker, may not have the social or financial resources to undergo transplant, gives them an opportunity to adjust their lifestyle and potentially, if needed, be successful in being listed for transplant. And then there are those non-modifiable factors, which include blood type or highly sensitized individuals, which may require a longer time on the transplant list in order to be properly matched. And then those, of course, which are disease-related, and many of my colleagues on the podium today have made a very strong case for earlier referral for transplantation for several patients. In particular, Dr. Gofrani emphasized, and as well Dr. Hoper and colleagues emphasized the importance of understanding the differences between patients who have classic pulmonary arterial hypertension phenotype and those who have a mixed phenotype, which is characterized by an older age, more comorbidities, smoking, low diffusing capacity, and perhaps more severe hypoxemia. And they, for the most part, behave like group 3 pulmonary hypertension patients. In particular, patients who have an elevated PVR above 5. Those patients have a relentless progression in their disease. And even though you can try different types of therapies, including approved therapies like inhaled iloprost or try a PDE5 inhibitor, I would argue that you do not sufficiently migrate their outcome, which would be better than you would obtain by transplanting that individual. We also recognize that people who have scleroderma, for example, have intrinsically more right ventricular dysfunction. They are less responsive to therapies and have other comorbid conditions, particularly esophageal, gastric, and peripheral vascular disease, which require more consideration during the workup phase and may require modification or treatments prior to being accepted for transplantation. The really tough one that I have difficulty with are patients with Eisenmenger physiology, who may or may not require heart-lung transplant. The timing is very difficult. Those patients have a much better prognosis and usually have a systemic right ventricle. And as a result, they don't typically go into right heart failure. They more commonly suffer from effects related to chronic hypoxemia, polycythemia, and other end organ disease. In addition, the timing is further complicated by the fact that the wait time for heart-lung transplant block is much greater. And so the crystal ball that you must have requires a lot more detail and complexity and a lot of guessing as it relates to the timing of transplant. And in that group, often the decision rests around improvement in quality of life. Dr. Bouclet showed us this as well, and this for me was very sobering. And this is real-world data. This is from the French registry. And basically looks at their experience over a period of time, which I think was from 2009 to 2017. And so it doesn't perhaps fully represent the pharmacological armamentarium we have right now, but nobody can do this better than the French. So this is the best you can get. And essentially what it demonstrates is that many patients who are high risk or intermediate high risk do not migrate out of that risk category. So the importance of evaluating patients at 3 months and being very realistic as it relates to a discussion and potential referral for transplant, I think is paramount. Sotatercept and other evolving therapies may alter this equation, but for the time being, the default should be to at least have that patient assessed for transplant as a safety net to ensure if they do not respond to those therapies, that they are ready for transplant. And I would challenge you that my survival curve, as it relates to transplant, rivals the survival curve without transplant for patients in particular who are intermediate high risk to high risk. You're looking at a median survival of 2 years, at most 3 years, for patients at intermediate to high risk. And that is very similar to what you would see in group 3 conditions. Whereas the median survival after lung transplant extends 7 to 8 years. So I would argue that if your patients are on the trajectory of an intermediate to high risk or high risk, those are the patients who should be referred for transplant and potentially listed. So as a result, again, stemming from the Delphi, we recommend discussions early on, early referral for those patients who have high-risk features, whatever risk score you use, and particular patients who have an underlying condition such as veno-occlusive disease or pulmonary capillary hemangiomatosis or group 3 disease, the cadence of moving through discussion to refer to listing should be accelerated. And particular patients who have other complications such as alveolar hemorrhage should be assessed for transplantation because they have an unpredictable clinical course. Now, what about transplant and pH center related issues and transplant ecosystem? And this varies from jurisdiction to jurisdiction. For me, this was a very useful paper which compared the experience in France to the experience in the UK. Similar population. France has essentially a single center of excellence as it relates to pulmonary hypertension and one hospital that does the lung transplantation. In the UK, they have 5 transplant centers and I believe 7 pH centers. And what they were able to demonstrate was the potential influence of that amalgamation of expertise on outcomes as one option. And what they demonstrated over a period of observation is the following. First, that if you look at on the A is at 6 months after registration and listing, and B is 12 months after registration and listing, comparing the UK and France. Now, France had the advantage of having an emergency listing program early on, whereas the UK did not have that in place until about 2018. So that may account for some of the differences, but not all the differences we see. First of all, if you look at the numbers actually transplanted, it was much lower in the UK compared to France at both 6 months and 1 year. In addition, the number of people who were delisted or died waiting for lung transplant, not so much heart-lung transplant, but lung transplant, was greater in the UK system. The other important thing, if you look at the characteristics of these patients, they're a bit different. If you look at the age of transplant, the French tended to transplant younger individuals. They also tended to transplant less sick individuals. And those were probably patients who had underlying high-risk conditions like pulmonary veno-occlusive disease. In addition, if you look at the rapidity at which prioritized patients received a transplant, the urgent waiting time days in the UK was 17 compared to France, which was 2, which is truly remarkable. The regular waiting time days, 165 days versus 45 days in France, which speaks to essentially access to the donor pool and the number of organs being utilized in France compared to the UK. The other interesting thing for me was if you look at the last line, the post-transplant hemodialysis hemofiltration was lower in the French population, suggesting that they were healthier coming in to transplant and had less morbid events after transplantation. And the really important one is that the outcomes in the French group was superior to that in the UK group. I don't mean to point fingers, but this is a worldwide phenomenon. The nuances of transplant are important to consider as well. First of all, survival in the first 3 months is the worst for pulmonary arterial hypertension compared to other indications for transplant, COPD, interstitial lung disease, and also cystic fibrosis. And that relates to the technical aspects and the RV failure and LV dysfunction that can occur after transplantation for pulmonary hypertension. If you look at conditional survival after 3 months, in fact, patients with pulmonary hypertension do better than their other cohort. The trick is getting them through those 3 months, and that can be done through bridging or ECMO after transplant, as well as other nuances related to care. And it speaks to the expertise of the centers doing this treatment. And we know from publication by Yang and colleagues that if you look at outcomes related to volumes, there's an uptick. If you're transplanting less than 33 patients in your center per year, the hazard ratio for adverse outcomes and mortality is increased. That is compounded by the fact that pulmonary hypertension, pulmonary arterial hypertension, is an uncommon indication for transplantation. And you can see in the dark purple, which is IPH, it represents the thin slice of the wedge as it relates to all transplantation. So even in high volume programs like ours that do over 200 transplants per year, we only transplant about 5 to 8 patients with pulmonary hypertension each year. So the expertise and the familiarity with transplanting pulmonary arterial hypertension cannot be emphasized enough. So small volume centers would uncommonly transplant a patient with pulmonary hypertension, as a result, have very poor outcomes. So I'm going to switch a little bit in the last few minutes. The other opportunity for our pulmonary hypertension patients is decrease the time on their waiting list, and that is access to organs. And we recognize that donations relative to the number of people who are dying are lowest for lung transplantation, about 18%, using the OPTN registry, which is the US registry. The one thing that has benefited our program, and in turn other programs, is the EVLP, which is the ex vivo lung perfusion prior to transplantation. And Dr. Seipel and colleagues demonstrated this in the New England Journal several years ago. The advantage here, if you look at the radiographs of an EVLP preparation, you can see the one on the left is a sick organ. It's got a very poor PF ratio, it's perhaps under-recruited, and the beauty of this is you can actually resuscitate this organ outside the person so that you get an improvement in PF ratio, an improvement in their radiology, an improvement in lung water, and an improvement in that lung's physiology, making it more suitable for transplant and giving the teams more confidence to transplant what otherwise would have been a very risky adventure. And as a result, if you look at Toronto's volumes, you can see that the number of EVLP cases that we're doing per year has increased dramatically and has allowed us to maintain a transplant volume, barring COVID, over about 200 patients per year. If you look at the outcomes from EVLP are as good as other non-EVLP transplants over time. The other beauty of EVLP is it allows teams to be rested. So this now becomes a semi-elective procedure. You can keep that lung on EVLP 6-8 hours, particularly with normothermic stabilization of the graft now, perhaps even 10 or 12 hours. And so you can have a rested team coming in the morning to do the transplant, which has been shown to improve outcomes. In addition, the logistics, and this is our OR board with about 8 transplants going on, is much better, where you're able to adjust the cadence of what's being transplanted and when. So it's been a huge boon for our transplant teams. In addition, it's allowed us to import organs from the United States. Hopefully Donald Trump does not recognize this happens. But basically, we are in the business of importing lungs from the United States. And when we do, a median of 9.5 US centers have refused those lungs that we've accepted. And if you look at the outcomes of those lungs compared to the lungs that we use within Canada, they're exactly the same as it relates to death and chronic lung allograft dysfunction. Again, because of the benefits of EVLP and the benefits of being able to resuscitate those lungs. The other thing that we've done in the final couple of minutes is that we've expanded our donor pool, and sadly, because of the fentanyl epidemic, many of our organ donors are brain deceased because of an event related to fentanyl. And many of those patients, unfortunately, also have hepatitis C. What we've been able to do is actually use UV light to kill the hep C virus in the perfusate, transplant those lungs, and then treat the recipient with antivirals with an excellent outcome. So this has further expanded our donor pool. And finally, much like in kidneys, we're now looking at creating a universal donor, essentially taking the lungs of A, B, and A donors, stripping off the A antibodies, and then transplanting that neutralized graft into a recipient. And so this is early days yet, but hopefully will further allow the ability, particularly of O recipients who tend to wait a long time on the list, in receiving a graft in a timely fashion. So hopefully I've encouraged you to move up the discussions about transplant earlier on, recognize that transplant is not the death knell, that it is an opportunity for your patients to live longer and live better. And I'd particularly like to thank my colleagues that I've worked through over the years, our transplant colleagues on the left, the Latner lab, which is the thoracic surgery medical research lab, and my colleagues at the World Symposium. Thank you very much for your time.
Thank you, Professor Grunton, for your excellent talk. Can you sit with us, please, in the podium? So we'll start, take your questions, if someone would like to ask. Okay, Professor Irene, go ahead.
Well, thank you for the great session. I start at the end because I have a very short memory. What is, Professor Granton, what is your expectation of how will sotatercept change your behavior? I think it's a very obvious question.
I think that's a very fair question. I have no idea, but there's clearly going to be people who respond and people who don't respond. And I think hopefully you'll understand early enough as to whether they're having a favorable response. And I think the general sense is after maybe 3 or 4 injections, you start to see a reasonable effect. I think if you start to see somebody who's deteriorating the face of those treatments for certain, but patients who certainly don't migrate out of that high risk or intermediate high risk group to a lower risk group, those patients should be probably listed for transplant. My recommendation would still be for a patient who is on the list and waiting for transplant, if you're embarking on sotatercept, I think it's a difficult discussion to have as to whether you should keep them on the list or explore whether they do respond to that medication or not. And I think time will hopefully tell, and perhaps work using proteomics or genomics will identify those patients who will or may not respond to those treatments, which I think is critical.
One more brief question. There was a discussion on genetics this morning. Will there be a time where we will look at organs with regards to what we transplant there?
I hope so. People smarter than I hopefully are working on that, and we will be able to, I think we discussed about prognostic markers, but I think more identifying patients who are likely to respond to different treatments so that you can move those treatments earlier on and not waste time with ineffective therapies to see if they migrate out into a lower risk category. Oh, sorry, I missed that.
It's almost like there may be genes that are not, they cause PH or cause lung disease or in the further course. So do you think that this will never be an issue because survival is limited? If I get a BMPR2 lung, its likelihood is low, but if there's more genes discovered, will there be a screening phase for organs before they will be transplanted?
Oh, I see. So more matching of organs?
Well, making sure that you don't have an inherited disease that you pass on.
Screening the donor.
Oh, so you get the disease in the graft.
Yes.
That uncommonly happens, I must say. There's very few conditions where you see.
It's not an issue, basically.
Beg your pardon?
It is not an issue.
It's not an issue. Sadly, patients die before they ever see that. And the biggest Achilles heel for transplant is chronic lung allograft dysfunction, either the obstructive phenotype or the more deadly restrictive phenotype. And I think that if we're able to address that, and that's always been the reason for the shorter survival in patients who receive lungs. It's of all the grafts has the shortest lifespan. I think that's the critical point. And there may be genetic issues in those areas or epigenetic which can be modified to prevent clad from happening.
Thank you so much, Professor. Dr. Fatma, just for the sake of the time. Yeah.
Sorry. So I heard cardiomems so many times in PH, but I haven't heard cardiomems in HFpEF talk. And we know that like the data of cardiomem in HFpEF is actually way better than any data of cardiomems in pulmonary hypertension. And probably maybe there is no dedicated data of cardiomems in improving PVR or improving precapillary component of pulmonary hypertension in patients with HFpEF, but they do reduce rehospitalization due to heart failure. So probably that would be something that we should be careful in, like in treating patients with HFpEF, especially those who have PH, combined pre and post capillary pulmonary hypertension with HFpEF.
Thank you, Dr.
Can I just
Yes,
address that? Because I think this is an important point. I think we need to be cautious with what cardiomems are telling us in terms of changes in hemodynamics and patient status. You're absolutely right. In heart failure, it's extremely powerful just because what you're measuring is the wedge pressure through the diastolic PA pressure. And that's why it's working so well. When you start to have a precapillary component, you probably lose that benefit in terms of clinical application, which is, I think, one of the limitations in the field of pulmonary hypertension. And also, one of the issues is to have an accuracy of measurement that would allow you to make the proper assumption that you have an effect on pulmonary vascular resistance. And I'm not even talking about cost, which is quite significant.
Okay, Dr. Faiz.
Thank you for the great session. My question for Dr. Granton. What's your recommendation regarding the ECMO use in pulmonary hypertension patients and in general for the other patients during transplant? So do you put your patients, all of your patients on ECMO during the OR, post-OR? What's your recommendation?
Right. It's a great question. I think I've again learned from Dr. Saval and colleagues is to understand that you shouldn't put them on too late. Even though patients on ECMO or put on ECMO as a bridge to transplantation still have good outcomes, it's clear that patients who come in late and are put on ECMO have worse outcomes. So I think you have to anticipate, particularly in a patient who comes with a decompensated right ventricle, that you may try some pharmacological acrobatics for a short period of time, but if you're not achieving the endpoints and you're starting to see end organ dysfunction, that you probably should if they have an exit strategy, put them on extracorporeal support. Usually veno arterial ECMO, there's different configurations. It depends on the center's experience. As far as support after transplantation, certainly the Vienna group was very aggressive in putting a lot of their patients on ECMO after transplant, but even they are now starting to pick and choose a little bit more and be more highly selective in who they put on ECMO after transplantation. I think our own center underutilizes ECMO after transplantation. It certainly makes life easier as an intensivist. There's less primary graft dysfunction, less pulmonary edema, less hemodynamic consequences, less issues with respect to loading a naive left ventricle, which hasn't seen that degree of venous return, so you less pulmonary edema as well. So I think we generally make an assessment in the OR and then watch them for a while and then determine whether they should either remain on ECMO or go on ECMO.
Thank you.
Yes, Dr. Khalid.
Thanks all speakers, excellent talk. My question is to Prof. Majdi. In terms of pulmonary hypertension in pregnancy, do you have any special recommendation when you discuss the mortality risk, the abortion in patients who are newly diagnosed during pregnancy or patients who are already known PH and proceeded with pregnancy?
Thank you, Khalid. So I think it is definitely once those patients, because we do have few patients who clearly said we want to be pregnant, and whether we like it or not, they're gonna really become pregnant. So I think if you look at the data said that those patients who are at low risk before a pregnancy, low risk basically based on really risk strata, they do have a good outcome. Those people having very, very low mortality. So I think the goal is trying to avoid that the patient accidentally became pregnant. So really plan that very carefully, keep them at low risk. And then once that happens, the chance that they're gonna really survive this pregnancy is extremely high. So, however, we noticed that some patients, the nature of the pulmonary hypertension became more aggressive after a pregnancy. So I think Ragda and I, we have really one patient who was doing very well on oral therapy. She was, I think, on macitentan and sildenafil, and she was doing great with that. after pregnancy, she survived the pregnancy, but her disease became very aggressive, and now she is on 3 drugs, including subcutaneous prostanoid, and we just started her on sotatercept. So now she is on 4 drugs, including sotatercept, for a patient who before pregnancy, she was doing very well. So those category of patients are there, and the patient should be aware that you're probably gonna survive the pregnancy if they are at low risk, but there's a chance that your disease might be very, very aggressive after the pregnancy.
Yeah, but my actual question is regarding the patients who are newly diagnosed. So you face them, they are already pregnant, and they had underlying BH who no one knows about it. So the mortality risk will remain the same, or it will be much higher on those?
Well, I think if you look at the data, those are at really high risk of death are those who have really congenital heart disease with something is a menager, or those who have rheumatic heart disease, really group 2 and group 3. Those patients who belong to group 1, even if they are discovered during pregnancy, for example, our patient, unfortunately, she came with a florid RV failure, and if that happened, then probably mortality gonna be high anyhow, because whether she's pregnant or not, she's not gonna be having really good outcome with florid acute RV failure in undiagnosed patient. But I think pregnancy nowadays, what we are trying to do, even if they became really pregnant or they're becoming aware that they are pregnant while they are on treatment, of course, if they are on, for example, ERAs or Sigewat, they have to be aware that they might have already some toxicity on this patient. So I think it is really a multidisciplinary team approach. The risk, there is no simple answer to what to do to those patients, but you need to take it really step by step. As long as they are at low risk, they're probably gonna do be well.
Thank you.
I think you have...
I was just going to add to that. I think the important thing to emphasize when you're counseling patients is that when you do a risk assessment, they have to be on the drugs that they're going to be on during pregnancy. So if they're on an ERA or a soluble guanine cyclase agonist, they cannot stay on that drug. So either they're going to be on a PD-5 alone, or they're going to be on that plus a prostanoid. So when you're doing that risk stratification, you realize they have to be on monotherapy. And so they have to go into this with all eyes open, and the destabilization afterwards as well. We do exercise as well to see if they have RV reserve. So we do hemodynamics with exercise to further inform whether they have some RV response to exercise as well.
Okay, Dr. Khalid?
Yeah, thank you. This is for Dr. Ghofrani. So I have a question. The ILD is a huge, as you mentioned, like there are several hundred different ILD. At least we need, what do you think about at least separating the connective tissue disease ILD from the non-connective tissue disease ILD? So if you have somebody with the equal severity of fibrosis, but you have a scleroderma versus non-scleroderma, do you think we need to be more granular in trying to figure out the treatment plan?
Yeah, Khalid, thanks for that question. Very important clinically as well. I think that we need to be more precise and we need to be more granular. However, what we were lacking so far are reliable large-scale registries or databases upon which we can derive specific recommendations or specific observations for all those subgroups. This is now overcome by the presence and the existence of these kind of meta-registries, and they will help informing us and become more and more granular. The numbers you saw actually are limited by the fact that towards the end analysis, obviously those patients were depicted or picked who had baseline and follow-up right heart cath, because hemodynamics was a question in that paper. If we look at the entire group that was registered and then just look at outcomes, we would enlarge the groups, the subgroups as well. And there is a lot of information still inside the database. And as I said, it's growing and it will allow us to become much more granular in future.
And similar to your data, the extreme minority of pulmonary hypertension patients are actually making it the way for lung transplantation. We also noticed some resistance from our colleague in lung transplant to transplant these patients or giving them priority. So I was wondering, what is your technique, or how do you approach this? And what will be, if you want to pick up one, let's say, bad prognostic sign for this patient to convince your colleague to get this patient transplanted. We have this patient waiting until they get really very sick, and then the outcome will not be good, and then they will refuse later on to transplant patients with similar cases. So it's becoming a vicious circle.
Right. That's an important question. I mean, some jurisdictions are bound by lung allocation scores or the composite allocation score in the United States and the Eurotransplant score, the Eurotransplant score. We basically just prioritize 3 rapidly deteriorating, which a lot of the pulmonary hypertension patients would fall under. I think our program is comfortable transplanting those patients, and they recognize that doing them earlier, much like the French data, translates into better outcomes. So I think if you wait too long, you start to have bad experiences, and then you become gun-shy in not wanting to transplant these individuals. What does a sick person look like? I think we all know what they look like, and they may have different markers of badness, but certainly patients who have cardiorenal syndrome, patients who start to have a congested liver, you know, if you're starting to look at ascites and those other things, that's a very late patient and probably should be looking at doing something a lot sooner. And certainly those high-risk conditions that you know their trajectory is very poor, that there's probably no advantage to holding off and waiting, exposing them to the risk of death and morbidity from their underlying condition, which can be unpredictable. So people with significant pulmonary hypertension and group 3 disease, patients with PVOD, those patients should probably be transplanted quite early while they're quote-unquote healthy.
Well, that's basically what we do. And last question for RD. I don't want him to feel lonely. Hi, RD.
Hi, Abdullah. Good to see you. You're looking good.
Thank you. You too. Now, the risk stratification for PAH patients with lung comorbidities, you know, the one we have is probably not optimal for these kind of patients. How do you risk assess your patients who are falling in this category?
Well, actually, what this latest interrogation of the database shows that actually all those PAH risk scores, though not being fully validated yet, but they roughly give you a very good assessment of the risk also in patients outside group 1. So the risk scores work in group 2 and group 3 and group 4. This is the bottom line reading from the large-scale registries. Of course, it needs a final validation step. We are on our way to do so with different partners in the consortium. But I think as a kind of rough approach to risk assessment, they work quite well.
Conclude the session. I would like to thank all the panel for your great talk and for all the audience to stay late with us on the Friday afternoon.
So last announcement. First of all, I would like to thank all the speakers, for also the organizing committees. I would like to thank also the attendees, really remarkable. We have attended since morning till the 4 p.m. and this is really appreciated. Last announcement for our SAPH guests, we will have gala dinner. Please, we will meet at 7 p.m. in the lobby and the buses will be outside waiting for you. Thank you so much.